Let me know if my comment through there. Should be working. All right. Good afternoon, everybody. Tamam. How audio levels is? If you're all okay with the audio, if you need anything, raise the Lord. Let me see how everybody. Let me know. Right, so hope all you don't mind the little background music. That just so you do, you know, you have a little vibe to it. Alright, that's now all you need to see me. Alright, that's now right. Perfect. Okay. Nice. Okay, so the first thing I want you all to do is to check the description of the video. When you check the description of the video, you're gonna see a link to a file. The file is this manual that we're using here. Feel free to use it. You don't need to, honestly. So you know 100% that you need it. I'll link it here. Somebody post the actually no I could get. Tell me if you all getting access to the file. It's supposed to be a PDF. As I said, not absolutely necessary, it's just who wants it. You don't download it? Okay, good. So you can have it open if you want, but you don't need to because I'm showing it on my screen, right? So let me tell you, well, first of all, anybody I say who you know they feel like they're getting a 4 or 5, right? At least they'll get 3, hopefully at least a 3 by watching this video, right? As I say, if you're in a real bad state and you never went to any class, you would at least get a pass by watching this video. I can't say I won. You have to at least go some classes during the year to get the 1. But just to let you all know up front, we're going until 10 o'clock, right? We will take a break sometime in between. We'll take an hour break or hour and a half break. But we're going until 10 o'clock. I understand that the exam in the morning is a AM exam. So we don't want to go too late because all you have to get all the rest. Make sure you don't drink no milk and eat no curry today, right? Because tomorrow you have 2.5 hours to get 6 questions done. Right, so another thing, I don't give lessons. I'm a teacher, right? I just here, I'm a good Samaritan here to help all you out. So anybody you know, send the link to them, the thing free. I don't usually post on YouTube or anything, but I'm doing a paper 1 review the day before the exam. So I can't remember the date I put, but the channel. I'm doing a physics 1 too, a physics paper 2 review, and I'm doing a physics paper 1 review. You watch those and I say you guarantee to at least get 3 if you never went any Zoom classes. But those of you who feel like you're in a 3 or 2, you're going to get, right? Once you get 3 here, you get a 1. I don't know how to use a computer, so I can't guarantee that the live stream will save. YouTube just tend to pull down all kind of thing. The Cape Biology one that I did, all I do was post a GIF, an animated GIF of a man scoring a goal, and United European Football Association take down the whole 9-hour stream. So I can't guarantee that it'll stay up because I just tend to pull all kind of shoppiness on the screen. Right? So and if you get taken down, I don't really care if you get taken down. He's not a YouTuber, he's not my teacher. As I say, this is just a conduit for me to here to get through to all you. It is how some people always so used to online classes. You don't have to ask to go, right? Just go if you want to go. If your mother calling you to sweep the yard, just go. You don't have to ask. I have no idea. Like why some people just ask if they have to go. I don't really care if you're going or you're staying. So you want to go and use the toilet, right? Just go. You want to go and sleep? Just go. You don't need to announce it. Alright. So no, I can save. I just can't stop YouTube from taking it down. I try my best to use non-copyright material, but as I say, something as small as a chip could get a thing taken down. Alright, all you. So let me press on. So what we doing today? So what I'll do is I will go through the topics as we go in, right? And sometime a little later on, if it's like a difficult topic, I will go through some past papers with it. So I won't be doing past papers the whole time. The paper ones here, I'm going to be doing past papers, right? Now remember, for CXC, they give all the broad topics. So I'm not reviewing everything. I'm not reviewing things like expression. I don't think you have to know osmoregulation. I think as somebody asked. So I only go into the topics that you have to know for paper 2. For paper 1, I'll probably go into a little more depth in the stuff that I didn't do, but that'll be our next day. So that's one where we're just doing past papers. Alright, all here. So there we go. So the first topic that we have on the list, right, is nutrition. So nutrition here is based on exactly a pretty big topic. I don't think, let me double check and see if excretion on the list. I had it all written down. Oh yes, excretion is on the list. Then yes, I have to go through that. Yeah, so let me call out the topics here for all you. So we have nutrition. Nutrition is one. Nutrition, biotic and abiotic factors, food chains, predator-prey relationships, decomposers, treatment and control of diseases, transport, excretion, reproduction, and continuity and variation, which is the whole last blast to the syllabus. So that means we got to spend some considerable time going through that. Luckily, some of the other things, they kind of break it down. So we have a lot to go through. As I say, we're going until 10 p.m. Those who now come in, see the description and you'll see the links to the notes, right? So again, they're there, but I'll be showing it on my screen. So let me start with nutrition now. And I say, as we go in, if we encounter, if we bounce up some difficult stuff, what we'll do is I'll go through some past papers. Right, Ollie? So nutrition encompasses a lot of different things. Nutrition encompasses diet, enzymes, photosynthesis because it's autotrophic nutrition, and also the alimentary canal, like food going through and absorption and assimilation and things like that. So we're going in order the broad topics, right? Assuming that's the order of the questions that I get in CXC. So assuming that CXC did not lie and say they bring in things that they say they end up bringing things that not on the list. But I think according to how some exams went so far, they're lying. In fact, CXC does be rather upfront about the dotishness, right? I don't think they need to lie. Alright. Let me start first with photosynthesis. Alright, so let me actually let me switch here, right? So photosynthesis, trying to do this in a way that I could still see chat and thing. Right. First thing you have to know about photosynthesis is the formula. So we have the word formula and we have the chemical formula. So we have the raw materials for photosynthesis. The raw materials are carbon dioxide. Let me make this a little smaller. The whole doc not showing. Okay, I'm going to re-upload it just now and change the description, right? Alright, I'm going to change it just now. let me link already let's now go on the Google Drive. Okay, I understand. That's probably my bad. As I end up uploading the old versions of the document. Okay. Somebody check, I post a thing there, somebody check that and tell me if that's the whole doc. It's not PDF, though, it's a Word document, right? Right, so we have the raw materials for photosynthesis. I'm just change this font a little bit there. Remember to set your thing to remember to set your thing to 720p, right? Alright, so we have carbon dioxide plus water, and that's going to give you glucose plus oxygen. So these are what you call the raw materials, right? So these here are the raw materials. Put in the chat quick what our arrow represents now. Like what would be the other factors that this arrow would represent. Put in the chat for me. Alright, so these are what you call the raw materials for photosynthesis. Or only the corner was showing. Oh, yeah. Sorry about that. Right, sunlight and chlorophyll. Sorry about that. I fixed it up. I say I don't know how to use a computer. Right. So this here would be the other factors. So this would be basically the solar energy, right? So this is the sunlight and the chlorophyll that this represents. And the chlorophyll, as you remember, comes from, they are found in chloroplasts. That's really all you need to know about it. So we have to know the chemical formula as well. All you know all this thing already. So this is the chemical formula is going to be 6 CO2 plus 6 H2O. So there's 6 molecules of each, right? Sometimes you'll see this written as 12 H2O, but that's if you're taking into account the next thing. Right now we're doing 6, 6, 6, the mark of the devil, right? So we have 6 oxygen, 6 carbon dioxide, and 6 water, right? 6, 6, 6, mark of the devil. And that's gonna create glucose, which is C6H12O6. There's no need to put the states of matter. So as I say, it's not chemistry. Chemistry is the devil itself. But there's no need to actually put that. So having this here is fine enough. I'll make this guy a little smaller in the corner here. So yeah, it could be too much anyway. Right. So there we go. That is the chemical formula for photosynthesis. So that there's not too interesting. What is interesting is the next thing that you'd have to know is what happened, what the fate of these products. So what happens to carbon dioxide and what happens to water, right? So if I here was to draw, let me say I was to draw a leaf, and to go into this here, hold on now, right? So let me say that this here is my leaf. Now when we say nutrition is also the adaptations for these things, right? Yes. So let me take a look at the raw materials again, right? So we have 6 CO2 and we have 6 H2O. These are our raw materials. This is what CO2 is coming from the atmosphere, and the CO2 is going to enter through. Put it in the chat what's the names of these little things under the leaf. So the CO2 is going to enter through there, and the H2O, which is water, that's going to be taken up through the roots, and that's going to be pushed up through the xylem, and the xylem is going to take it into the leaf itself, right? So you're going to get the water through these little veins here. But the question is, how do we get C6H12O6? Well, the other factor here is going to be sunlight, and all of this is going to take place in the chlorophyll. So if we think about what adaptations a leaf must have, right? So what adaptations a leaf must have in order to undergo photosynthesis. One of the main things is geared towards making sure that the leaf gets enough sunlight. So the leaf in this case here has to be broad. It has to be very broad. So it has to have a broad surface area. It has to be highly vascularized, which means that, you know, it has a lot of veins and vascular bundles to transport that water. And then we'll get a little deeper into what inside the cross section. But then you have a petiole, which is a stalk to orient the leaf towards sunlight. So if they ask if for three external factors that assist with photosynthesis, those are the three answers that we could look at. That the leaf would have a broad surface area. Of course, that would make sure that it has more exposure to the sunlight. It's highly vascularized or highly venuated, either one. Basically, it has a lot of veins. And it has a petiole, and the petiole sort of does this thing to bend it towards the sunlight. Not bad. Now, what is the fate of these products? So why do we need sunlight? Well, what sunlight does, right, is that when the sunlight comes here and interacts with these water molecules, and I'm going to actually draw a molecule here. Let me say that this represents one water molecule. So this is going to be H2O. So this is going to be a O, and this is going to be a H and a H, right? So what the sunlight is going to do is going to do a process that is called photolysis. Now I'd be like, well, what is that? What is photolysis? Not photosynthesis. Well, photo means light. Lysis, to lyse is the split. So it literally means to split using light. In this case here, we split in water. So we're going to have the photolysis of water. Now if we look at what glucose is. Glucose is C6H12O6. So you see here these hydrogens. These hydrogens are coming from when this splits. So when this water molecule splits, remember it's 6H2O, right? 6 of these. Now if you think about it, we have 12 hydrogens. We have 12. So this is where this 12 coming from. So we're going to get the 12 hydrogens from the glucose from the water, right? Remember that. We're going to get the 12 hydrogens from the water after it splits. So what happens to this oxygen here? Put in the chat what you all think happens to these 6 oxygen molecules. And so these carbons now, this comes from the carbon dioxide. So the carbon dioxide is going to lead to these carbons here. And some of these oxygens are going to come here. So just now I'm going to draw like a little flow chart to show everything. So far, let me see what we have here. We have 12 water molecules. Well, sorry, 6 water molecules. The sunlight is being used to split those water molecules, yeah, into hydrogen and oxygen. The hydrogen forms part of the glucose molecule that combines with the carbon from the carbon dioxide and the oxygen from the carbon dioxide. Nice. Okay, so we are going to say waste product here. Now, sometimes it will say waste product, but instead we're going to use the word byproduct. So what's going to happen to this here? This oxygen, well, this is going to diffuse into the atmosphere. So this is going to diffuse into the atmosphere as a byproduct. So I guess it's not really considered waste because this same oxygen could be reused for respiration. So this is why we say byproduct instead of waste product. And some of it is used in the oxygen molecules. Nice. Okay, so what we've done here is talk about the light dependent reaction, right? So we have the light dependent reaction. So let me actually give a little overview of what is what here. What is the light dependent and what is the light independent reaction? So long time they said just call it the light reaction and the dark reaction, but they said that they're a little primitive, you know what I mean? So let me look here and see what happens, right? And I'll put up a little flow chart here. Alright. Alright. Hopefully the thing's showing good now, I believe so. So if I look here, the light dependent reaction, right? Light energy is captured from sunlight. Sunlight is captured by chlorophyll in the chloroplast and converted into a usable energy called ATP. So if you remember, ATP stands for adenosine triphosphate. So this is basically our fuel. We don't plug in each other, we don't put in AA batteries, and for we to work, we don't hook up to our charging port. We use ATP. And most of our ATP comes from carbohydrates. So light energy causes photolysis, which splits water molecules into hydrogen and oxygen and is released as a byproduct. The hydrogen produced in photolysis is used to reduce carbon dioxide to photosynthesis. Reduce carbon dioxide to glucose. So the ATP required for this process, this shouldn't be hydrogen, it should be oxygen. The ATP required for this process comes from the light independent stage. So let me look at this. No oxygen, sorry. Thinking of myself, right? So let me look here. Alright. So what we have to do is be able to not just state a formula, not just to state formulas and things like that, but we have to be able to describe what happens to the fate of the products. So let me look here now. I know it look a little confusing, but if you look here, so light energy that's from the sun is absorbed by chlorophyll. The light splits the water into hydrogen and oxygen. That's photolysis, splitting using water. The oxygen diffuses out the stomata and goes back into the atmosphere. Some of it would be used for glucose, right? So the light energy is converted to ATP. That happens with a series of reactions that take place in something called photosystems inside it. Yeah, I need to know that. So the ATP now, from this reaction, from the sunlight, this converted ATP provides energy for this reaction to occur. So this is the light independent reaction. The hydrogen from the water combines with CO2, which is reduced to form glucose. So what we have here now is an enzyme reaction. We have photosynthetic enzymes, right? And you'll learn about those photosynthetic enzymes later on. But one of the main ones is called rubisco, and rubisco stands for ribulose 1,5-biphosphate carboxylase oxygenase, right? And don't worry, I don't have to know that, but there's photosynthetic enzymes. So without that, basically all life on Earth will perish. Now, so these photosynthetic enzymes, if you remember what enzymes do, enzymes catalyze reactions. So enzymes basically allow reactions to occur much faster than they usually would. So they lower the amount of what we call activation energy that's needed. That basically means that if you had to imagine it, right? If you imagine it, imagine that. So imagine that this is the amount of energy that's needed for a reaction, right? So you can think of this as the activation energy needed. So imagine you're trying to climb this hill it would be very hard to climb because of how steep it is. So what an enzyme would do is that an enzyme would actually lower the amount of energy that would be needed. So this would be with an enzyme, and this would be without an enzyme. So basically, it would lower the amount of energy needed to actually initiate the reaction. As a result, you can climb this hill faster. Reactants can convert to products at a much faster rate. So imagine this, an enzyme is like a microwave. If I put my food out in the hot sun, yeah, the food will heat up, it'll just take a long time. If I put my food in the microwave, there's a lot more energy, it lowers the amount of time that's needed to convert my cool food into hot food. So that is really what an enzyme does. And all enzymes are made out of proteins, and all of them we describe as biological catalysts. Right. So hopefully we're okay so far. I say I'm going too fast for you. Now, coming back here. So far, this is what we have to understand. There's a light-dependent reaction, right? Which is here. And there's a light-independent reaction. These involve enzymes. So what does that mean? That means if we think about, list for me some factors that affect enzyme activity. If you all remember, what affects enzyme activity? And this is where the light-independent reaction will get affected. So what we have now with photosynthesis are these factors that are called limiting factors. So limiting factors basically, they limit the rate of photosynthesis. pH is a good one, temperature is a good one, light intensity, right? Okay, good. So we have limiting factors, and let me go through some of those. Alright, all they should know how to test for starch and thing, eh? Water availability is a good one. Alright, so we have some good ones here. Anushka, Surya, Petal, Hope. Very good. Nice. So there are several requirements for photosynthesis. So if one of these were absent, photosynthesis will not occur. And glucose, you wouldn't get any glucose, right? And if you don't have any glucose, you wouldn't get any starch. So these factors would be like light intensity, CO2 concentration, temperature, water availability. So all these things here, if we have one of them that is not present, or pH would be 1, 2. Let me say that the temperature is too high, or the temperature is too low. Then we have a problem. The enzymes won't work. If the temperature is too high, by the way, tell me what happens to enzymes. Give me that word. Give me that word that happens to enzymes. Temperature is too high. What happens? So when temperature is too low, there's not enough energy to initiate the reaction. If the temperature is too high, something happens and the enzyme doesn't work. So we need all of these here. And so if you look at each one here, the effect of light intensity on photosynthesis, you'll see here that very good. So it denatures. I'm going to fix it up. Right. So what we could see here, we could see that the higher the light intensity that we have is the higher the rate of photosynthesis. High light intensity, higher the rate of photosynthesis. So, but this doesn't continue forever. This happens up to a certain point that we call the saturation point, because what happens, there's only so many chloroplasts and so much molecules of chlorophyll we could stimulate to convert sunlight to ATP. There's only so much we could stimulate. So if I subject it to a normal light bulb, normal, medium or high wattage light bulb, it's fine. If I put a whole stadium floodlight on it, it mightn't change the rate, because again, you only have so much chlorophyll it could stimulate. So this is how it looks. It goes up to a point, but then it reaches a saturation point where you have no further increase. So if it gets too bright, it just kind of wasting energy. In fact, it could be bad, because the more light that it produces, more likely the more heat, and more heat will denate your enzymes. Okay, so I explain that again just now. So what is a good experiment? And this is something that could come here. What is a good experiment to show that light is needed for photosynthesis? So this is where we would use an underwater plant, right? We would use an underwater plant, something that is called the Lodia plant, in order to demonstrate that photosynthesis is an easy way to show that light could affect the rate of photosynthesis. So if I have a test tube, or let me say a little flask or something, right? A measuring cylinder. So if you have a measuring cylinder here, okay, so if you have a measuring cylinder, or you could have a setup with an inverted test tube too, and we have one of these plants in here, this Elodia plant, right? It's kind of like seaweed, and we submerge it in water. This would be inside of a flask, right? And you know, we have a certain amount of water in here up to this point, right? So this is all this is in water. Now if we subject this to light, right? So if we subject this to light, we are going to see, you're going to see bubbles coming out. Put here for me quick what those bubbles are made out of. We're going to see bubbles forming on the leaves, and we're going to see bubbles rising to the top. So we're going to see bubbles, and these bubbles here could actually form a little column. Yes, good. So this is oxygen, because remember, oxygen is the byproduct. So we could see a little oxygen column there. We could see O2 bubbles coming out. So, alright, I don't know how to pronounce him, Anyawa Adams. You could always go back to the beginning of the live stream before if you want. Not now, but you know, when we catch a little break. Alright, so we have these oxygen bubbles that are coming out. And you could actually measure this column, and you could use the amount of light as the manipulated variable, or you could just count the bubbles. You could count how much bubbles emerge in 30 seconds or something. And for example, if we get 10 bubbles for one, and you get 20 bubbles for one, and you get 30 bubbles for one, increasing light intensity would mean that we'll get more bubbles because we have a higher rate of photosynthesis. Very, very easy baby thing. So by counting the amount of bubbles, we will be able to see an oxygen column being developed here. Now past a certain light intensity, the oxygen level will be the same throughout. Alright, so Microsoft Word kinda doing poop here. Alright, I think it normalized, right? Okay, so our next thing they could ask here is the necessity of carbon dioxide in photosynthesis. Won't this plant stop producing eventually because of the lack of carbon dioxide? That is true, Michael. So that means that this experiment is only good to measure the initial rate of reaction. It's not good for measuring the rate over, let me say, a long period of time. So you might only conduct this experiment and test it for the first 30 seconds. First 30 seconds is fine. So that's one of the limitations for the experiment, that you would only be able to test it for like a certain beginning of it. So you wouldn't carry that. They wouldn't do this experiment over an hour time or something. Say just like a minute for the most. Alright, so remember the factors that we need for photosynthesis. The raw materials is carbon dioxide, water, and then the other things that we need is sunlight, we need chlorophyll, and we also need for the plant to be at a certain temperature. So for photosynthesis to occur, we need carbon dioxide. So how can we test that? So what we're doing, we're sucking out all the carbon dioxide from the atmosphere. So we can't do that, but we can actually put part of the plant in, you could put it in like a little bag or something, and you have KOH in there, potassium hydroxide solution. Potassium hydroxide solution, what that does is that it actually removes the carbon dioxide from the sample. So any iodine tests for this would come up negative, because if you have no photosynthesis, you have no glucose. If you have no glucose, you can't have starch. So that's the necessity of carbon dioxide for photosynthesis. Alright, so we went through this already. What are the external factors that help with photosynthesis for the leaf? Oh, let me go through one other thing here. So one way to see if chlorophyll is necessary for photosynthesis, you're gonna have to use a something leaf. A leaf that have spot, like little white spots on the spots that don't have chlorophyll. Put in the chat there was the name of that type of leaf. So if we have a leaf, I might actually see if I can just get a picture of it. If I leave this up, I don't know how to use a computer, so I don't know. Right, so this is a variegated leaf. And what's happening here is I don't know how to use a computer, so you can't really depend on me to do anything. I'm not a teacher. So here we have the non-chlorophyll parts, right? And here we have the chlorophyll parts. Right? We have the chlorophyll dense parts. So these here won't produce glucose or starch. And these here will. I put product instead, right? All you understand. Right? That's would be I feel too lazy to fix it. So this will produce glucose or starch. I will go through some Karishma. I'm just going through some revision for now. We have until 10 o'clock tonight. Alright, so this will produce glucose or starch, right? So with an iodine test, this will test negative for starch. And this will be like a yellow-brown color. And this here will test positive for starch. And this will be a blue-black color. Right? So, easy. Me and for the pick leaf and thing during the pandemic, or if they do it in form for, I don't know, or they probably don't know iodine and thing homes have. I don't know. Hopefully a teacher show you a video or something. I don't know what they do for labs and thing, nah? Hard luck for you, yes? Alright. So, they shouldn't even be writing no exams now. CSEC, real bad boy. Them better than the government, you know. Them better than everybody. I tell you, the only people who CSEC not better than is that guard who they stand up outside the health clinic and tell you to social distance when they go in for your vaccine. That's the only person who CSEC not better than everybody else. Them could tell everybody what to do because they have to kneel down and do it. CSEC say jump, they'll say how high. Nice. So how exactly is photosynthesis maximized? Well, large surface area, as I said, more exposure to sunlight. Petiole, right? The petiole is the stalk, and that helps orient the leaf towards the sunlight. Highly vascularized, very, very dense network of veins. And remember, veins have the xylem in it that transport water, and they have the phloem, which will transport the sucrose. And there are many stomata which help with maximizing gaseous exchange. Yeah, CXC, I gotta lie, CXC is a huge fraud operation. Too bad all you have to write it. Hard luck, yes? I say, why all you do, why just to pull on all your mask and cough in the exam? Just cough in the exam, what they gonna do? Kick you out? So the place dusty, so the mask dusty, what they gonna do? All is a bunch of cowards, all should just cough in the exam, the examiner and the invigilator, and them just be coughing. In fact, all of them probably have COVID. You probably have COVID. So, a huge fraud, and they're more powerful than anybody else. They're more powerful than COVID itself. Alright. So let me look here. So here we have the internal thing in the leaf, right? So I'm just making a rough drawing here. So we're gonna have the upper part, that's the upper epidermis. That girl next to us, because she probably had COVID, that means you probably have it too. If I had COVID, I still going for my exams. 15 months or whatever of Zoom classes and Google Classrooms and thing, and them want to tell me I can't come for the exam? Nah man, I go and I go give everybody COVID. Doesn't matter what. If my mother and thing get COVID, I'll tell she, hey, don't say nothing, you know, don't say nothing until exam's done. Me know was the last exam. Next thing, like all your brother or auntie or something get COVID, what they gonna do? My auntie get COVID as a primary contact, my mother get COVID. You can't say nothing, don't say nothing. Just finish your exams and hush your mouth. That's what I want you to do. Right. And I'm just drawing. Alright, so I'll just draw in some parts here, right? Okay. Alright. Here we have the internal structure of the leaf, right? For legal reasons, hey, I not teacher, you know. I tell them all you, pull on all your mask and cough during the thing. What I want to see what they will do. What they will do. They'll kick you out. What they'll do, they put in all their in health hazards to do stupid exams. Alright, so if you look here now, here we have a cross section, the internal parts of the leaf, right? So this is the upper epidermis. This is what we call the palisade mesophyll layer. This is what we call the spongy mesophyll layer, and this here is what we call the lower epidermis. I don't really think this asks you to draw this, but you should still, it's easy enough to draw a basic diagram of it. Okay, so the upper epidermis, what you should know about it is that it is transparent. Put in the chat where you think is the big advantage of it being transparent. So you might be saying, well, what's these words? What is palisade? Like, where the hell that word come from? A palisade is actually a plank of wood. So you know, you ever see them picket fences and thing? You just call it a palisade fence. So they actually name the cells after that because it does kind of look like a picket fence. It does look like a row of very chlorophyll dense cells that, you know, this is basically where the party is happening, right? So this is where the party is. You can't have no party right now, right? I know some people still be having birthday party, but this is where the party is. And this is where so this has a lot of chlorophyll dense layer. Some plants have a double palisade layer, which means that they have to absorb as much, they make as much glucose as possible. Usually, like in the jungle, let's have a double layer of this. Right, so this is to allow sunlight to reach the transparent, so sunlight could come in, right? Because if you think we have in a party, we have in a photosynthesis party, right? Who are the guests? Who are the invited guests for this photosynthesis party? We need sunlight, we need chlorophyll, we need carbon dioxide, we need water. So those are the 4 invited guests. We're gonna have a small little birthday party because you only could gather in 5, right? So we need sunlight, carbon dioxide, water, and chlorophyll. We need those 4 guests to show up to this party. So what we need now is, well, where the carbon dioxide coming from? The carbon dioxide is coming through this here. And the carbon dioxide is coming through, right? Like coming in this thing here. So this is a guard cell. So the guard cells is kind of like the bouncers, right? So there's the guard cells, and this here is a stoma. You have 1 stoma, 2 stomata, right? 1 stoma, 2 stomata. So the singular is stoma. So we have one more thing I didn't put in here, by the way, which I'll put in just now. So the spongy mesophyll has a lot of air spaces in it. There's a lot of air spaces, so that this CO2 could come in. The last thing that we have here is this here. This is called the vascular bundle. The vascular bundle is basically the xylem and the phloem. And mainly what we're going to need is water. So water is going to come through here and go there. So it looked like a mess that we have here, but the point is we have in a party and we need water to show up, we need CO2 to show up, we need chlorophyll to be there, and we need sunlight to show up. The other thing is this: water flows from the vascular bundle to the palisade cells, right? But we don't want the water escaping through the top. So we don't want this water to be leaving through this top layer here. So we're going to have something on top here. Put in the chat there what's the name of this something on top here. So we're going to have a layer that is going to prevent water loss on top here. So most plants are going to have this layer. You're going to notice that we don't really have any chloroplasts up here. It is have, but not much. Right? So there's be like a thick layer on top here, especially for plants that have to retain water. So like xerophytes, nice. So we have a cuticle on top there. So the name for this is a waxy cuticle. So there's a thick waxy cuticle at the top. And what that is doing is that is preventing water from escaping through the upper epidermis. Right? So in a nutshell, let me go over again. Going from top to bottom, right? Going from top to bottom. We have a waxy cuticle at the top that's preventing water from coming out through the top. We only want water to go to the party. We only want it to go to the palisade cells. We want them to cross that fence and go in there. The upper epidermis, which is 1 cell, right? Which is 1 cell thick, has to be transparent. You can think of this as like there's a roof, but the roof is a glass roof. And that's allowing sunlight to come in as a glass roof. Right, so you have a nice car and the car have a glass roof and the sunlight coming in. So it's a daytime party, you understand what I mean, right? So this is the palisade mesophyll here. So the palisade mesophyll is chlorophyll dense. This is basically where the reactions are going to take place. The light dependent and light independent. We have the spongy mesophyll. These are very they're not dense at all. Right, so they have a lot of air spaces. So this allows the CO2 and the water to pass through to get to the palisade mesophyll. The lower epidermis is filled with guard cells and stomata. Guard cells basically open and close to allow, we'll get more into those when we're doing transport. They basically open and close when to allow CO2 in and also to let water vapor out. And between the little opening is called a stoma, plural stomata. Easy, very easy. All right. Don't worry, you're doing past papers as soon as we clear up something. Smart fellow, fat smeller. And that is it. That is plant nutrition. So let me look at animal nutrition now. So animal nutrition All right. Firstly, we took at enzyme. They take a look at enzymes. Now, if we break down digestion, animal nutrition is basically digestion. You also call it holozoic nutrition. You also call it heterotrophic nutrition, which basically means that that is something eating something else. Whether you're eating a plant, whether you're eating a chicken, whether you're eating a duck, whether you're eating goat, whether you're eating KFC, that is heterotrophic nutrition. It basically means contrasted to autotrophic nutrition, which is the thing making its own food. All of us are heterotrophs, right? All mammals are heterotrophs, which means that we cannot make our own food because we don't have chloroplast. There's no way to actually take in sunlight and manufacture ATP from that. So we must eat other things, right? We must cook pilau, we must cook soup, we must go on the road and not gather to El Pecos and get some food when things reopen and we have to go to all the bars and all the restaurants that convert themselves into markets because we all need to eat. We can't just stand up in the sun and make food. So enzymes now. So let me give a brief little thing about enzymes here, right? So enzymes, what enzymes are. So let's imagine that we have this very, very funny shaped molecule in the body, and you have a number of them, right? So we have this molecule here. Okay. I'm not supporting KFC. I tell you why I'm not supporting KFC, because when KFC let the workers go, they give them an aloo pie and a Coca-Cola. I can't support that. I will fry my own blasted chicken. I can't support a company that have millions of dollars giving the workers an aloo pie and a Coca-Cola to survive on. Okay, so this is what we call an enzyme. So what an enzyme is a biological catalyst. I just explained that, right? And what an enzyme does is that it converts substrates to products. What is a substrate? A substrate is basically a molecule that either has to be anabolized or catabolized. What does that mean? Anabolized means made into something bigger. Catabolized means broken down. Basically, it's a molecule that has to become something greater or be broken down into something smaller. So, how does it do that? Well, an enzyme is a protein, and you'll learn about proteins a little more later on. But proteins, they could form very, very specific shapes. So each enzyme is specific. So, and their enzymes are involved in numerous reactions, not just food. So we even have things that could block enzymes from operating. So in fact, if you ever spray a cockroach with death or BUP, what you're actually doing is when you spray them, you're deactivating the enzymes that help with the nervous system. So that's why they see them flailing around. I mean, some cockroaches don't dead with it, but that's why you see them flailing around and all kind of thing, because they actually can't control the nervous system. So you have enzymes that control many things, not just digestion. You have enzymes that control sensitivity. You have enzymes that control blood circulation. You have enzymes that control photosynthesis. You have enzymes that control movement. You have enzymes that help with reproduction. You have a bunch of them. The ones that you focus on at the CSEC level is only the ones that focus on the digestive system. So they are very specific. When I say they are very specific, is that they are very specific in shape. They are very specific in shape, and some of them are very specific in function. So what do I mean specific in shape? Well, they only interact with really one molecule. So imagine that we have 3 molecules here, right? So we have 3 molecules here, and let me say that this enzyme is called amylase. And we have 3 molecules here: molecule A, molecule B, and molecule C. Put in the chat for me which molecule you think is going to work here. Now, what's going to happen is that the molecule has to be able to fit into this area here. So this area is called the active site. So the active site is basically the point at which the metabolic reaction would occur, you are going to get something called an enzyme substrate complex being formed. Basically, the thing had to fit into there to initiate the reaction, right? Good. So that's a baby question. So right, so enzyme B, sorry, substrate B is the one that's gonna fit in here. So this is going to fit into something called a lock and key system. So the lock and key system or lock and key mechanism is going to allow the substrate to fit into the enzyme. Later on, you'll learn that the enzyme is actually not like a lock and key. So we're basically lying to you right now. It's not like a lock and key. It's more like a hand in a glove. It's kind of like, you call it induced fit. And it's kind of like, you know, the glove, this kind of shape itself a little bit to form into the hand. So, but as it's kind of like that. So they must fit. So this is what you need for the reaction to occur. So the reaction could basically be that this substrate here is going to get broken down into smaller products, and then another substrate could end up going. So the enzyme actually doesn't change shape when this happens, right? So but other things could happen as well. You could actually take two substrates and make a bigger product from it. Most of the digestive enzymes, however, break down things. So if this is amylase, that means that this substrate is starch. So amylase breaks down starch into a disaccharide that is called maltose. So maltose is still a reducing sugar, by the way, but it is a simpler version of starch. Starch is a very, very big it's a very big molecule. So I just drawing something here, and you don't actually have to know this, but just to show you here, this here is a glucose molecule, right? This is a glucose molecule, and here imagine this is one molecule. Just now. Alright, start with this one. Alright, so here's how to do this and keep. Those who want to do keep. So you do ho ho oh ho. That's how I know it. Ho ho oh ho. Alright, there's a C and there's a O. Right. So imagine this is one glucose molecule, right? And this is CH2OH. I know I bring in chem in bio. And starch is a very long molecule comprised of numerous of these linked together. What the enzyme does is that the enzyme breaks these bonds here. So it breaks these bonds and separates them into different molecules. So it breaks the bonds between the molecules so that they can become simpler molecules. Now, if you think about it, what is the point of doing this? Well, if I go to KFC and I buy a zinger, that zinger has bread, it has chicken, and the chicken is fried in a batter, so it has fats, and it might, I don't know if it's come with a little lettuce and things, it might have some lettuce. So I need the food, the nutrition from that zinger to go into my bloodstream. So it is too big. The starch is too big, the proteins are too big to actually go into my bloodstream. I need enzymes to break it down into something small in order for it to go in there. So it has to be small, it has to be simple, and it has to be soluble. There we go. And that is what happens there. That is what enzymes do. They ensure that molecules are small enough so that they can actually move into the bloodstream. Okay, so now something that they like to bring, and let me actually see if I could pop up a little diagram here, right? Let's know. something that they like to bring is a graph that's involving enzymes, and I could kind of bet that that's something all they're gonna get. So if they say nutrition, it could be a graph. So let me show you here. Alright, that's now I'm gonna link it up. Alright, so let me look here. I'll paste it on this thing here. Alright, let's see how this is showing. It's still a little small. Okay. Let me move it up. Make this a little smaller. Alright, all in. Hopefully all you can see this graph okay. And here we have two types of enzymes, right? Two types of proteases, those break down proteins. I don't know how well y'all could see it, but give me what is the optimum temperature. The optimum temperature is temperature at which you have the highest rate of reaction. Give me the optimum temperature of mammalian protease, that's the red. And give me the optimum temperature of the prokaryote protease, that's the black. So this is very easy to determine. All you have to do is look at the peak. Where's the rate the highest? And that there will actually show at what rate the substrates are moving and breaking down into products, so anabolizing into products. Nice. Very good feat. So we have 94 degrees Celsius. About 94, 95 degrees Celsius. I myself can't really tell from the graph, but let me say 94, 95. And the other one is 100 degrees Celsius. Now, what you'll notice here, now when I ask you to draw a graph in bio, and it is highly likely that all you get in a graph, right? Let me tell all of the things that you must remember when you're drawing a graph, right? So when drawing a graph, you need to remember the following. I can write it down if you, I'll put it in the chat actually. So you need to remember the following. Remember when drawing a graph. A graph is usually be like 4 marks, right? So you need to label the axes. You need to plot points correctly. You need to make sure that graph occupies at least half of the space provided. I have to make sure to put a title. So the title is always a descriptive title. So you can't do it, you know, like physics. You just put physics. You just put x versus y or y versus x. You have to put a descriptive one. So like for this graph here, for this particular one, I would title it like this. So I would say graph showing, right, percentage max efficiency for proteases across temperature. So that's what was meant by a descriptive, like a descriptive. All right, so I know this come off there. So right, so that's how you write a descriptive thing. So anytime you're drawing a table and anytime you're doing a graph, you have to make sure that it's descriptive. Okay, so when you're doing a table, by the way, oh shucks, I peeped the wrong thing, yes? I saw watching here. Yeah, it could be 40. I starting to watch here. It could be 40 degrees. And this could be 65. Yeah, sorry about that. I starting to watch here is the temperature. Oh yeah, I'm not too smart, eh? It's not teacher, eh? Anyway, so yeah, 40 degrees. Because all you have to do is come down here. Not 65 now, 70. Yeah, so this will be 70. So when you're coming down here, so that will be 70. So if you look at the next one, I'm get our next graph for it. Let me say we do this one. Let's draw one here. Yeah, 70. Good thing I ain't writing bio tomorrow, right? Alright, so let me say that this is a graph that we have here for pH. So here we have pH. And we have 0, we have 1, we have 2, we have 3, we have 4, we have 5, we have 6, and so on, right? And this here is percentage activity. Alright, so what's the optimum pH of this? What's the optimum pH of this? And which enzyme will you feel it is? So this could be, so this is the optimum here. All we have to do is come down. Right, so somewhere between, so about between 2 and 3. You're looking for the highest point. So the optimum will be about 2. Which enzyme will you feel this is then? Right. So this is most likely this if it's adult, it will most likely be pepsin. Where is pepsin located? So pepsin works best in acidic pH. So if we have, right, so this will be found in the stomach because we have a lot of HCl in the stomach. HCl provides a good medium for pepsin to work. Nice. So this is enzymes, right? And I say, or they probably draw enough enzymes that you're fed up already. So let me go a little more into the alimentary canal. Alright. Tell me, is it fish or beans? Pick one for me, fish or beans? Alright, I see in fish. Alright. So if you look here, see what the fish contains. The fish has protein, fat, water, some vitamins and some minerals. The fish wouldn't have carbohydrates really. It might have some glycogen, but it wouldn't have starch. Remember, glycogen is kind of like animal starch, right? Alright, first let me see what has happened during the whole alimentary canal. So first what we're gonna have is, let's lower the volume at this time. Alright, so what we're gonna have here is physical digestion occurring. The actual breakdown physically of the fish. We cooking the fish first, right? You said we cook the fish. But for me in the chat, what type of digestion is that? When you physically break down the food to increase the surface area. So this is going to happen with your teeth. So your teeth is going to break down the food. Teeth are going to break down the food. And you know, you're going to cut the food with your incisors, tear off any flesh with the canines, and you're going to grind it up into small pieces and that small surface area using the premolars and molars. Very good. So that's mechanical digestion. Alright, so your saliva now is going to have salivary amylase in it. But since we don't have starch, we're not going to do anything. So there's a neutral pH here. And chewing the food is called mastication, right? Mastication, not an X word, but mastication. Alright, just telling you in case you see the word and you want to know what kind of word is that. Alright, so this is going down the esophagus. So when it goes down the esophagus, and I'm going to show you something here. When it goes down the esophagus, it kind of looks like this. Alright, so if all you see here, right? So this is called peristalsis that you're seeing happening here. So that's peristalsis. So peristalsis is basically the esophageal muscles contracting and relaxing. The food is going to be rolled into a ball called a bolus, and it's going to be pushed down the esophagus from the mouth to the stomach. This is actually what is going to happen that is what is going to happen along your intestines as well, and is also what happens, you know, when you're ready to go in the toilet and do number 2. So this is peristalsis that you're seeing here. So the food just do fall along your esophagus. So let me see what happens here in the stomach. So in the stomach, you're going to have a bunch of digestive juices, basically hydrochloric acid and other things, and they're going to mix it up into something called chyme, C-H-Y-M-E. Some people say chyme, the real thing is pronounced chyme, C-H-Y-M-E. And what's going to happen here is that you're going to mix with proteases, mainly pepsin, because this is a acidic pH, acidic medium. And what the pepsin is going to do is going to break down the proteins into polypeptides. And polypeptides are eventually going to be digested into amino acids. So the food only stays here for about 2 hours, or hour to 2 hours, depends on what you eat. If you eat KFC, it might be less time. If you eat some salad, it might be more time. But there's actually mechanical digestion and chemical digestion happening here, because the stomach actually contracts to allow this food to churn and turn into chyme. Pepsin breaks down the proteins, and what happens here is that the amylase that is found in your saliva actually gets denatured when it goes to your stomach, because amylase gets denatured in an acidic pH. Alright, so what's happening now? So now we're going to go down to the duodenum. So the duodenum now is connected to 2 other organs, 2 main other organs. What are those 2 other organs that the duodenum is connected to? So the food now, which has been churned up and some of the proteins have been digested, right, are going to get further broken down. So here we have proteins. We would also have starch here, but since we don't have any starch in fish, we don't have it here. But we're going to have protein enzymes, and we're going to have this big boy here. So this big boy here is fats, right? Or lipids. Lipid is the overall word for fats and oils. Now, enzymes tend to have names similar to what they're breaking down. So, for example, proteases break down proteins. Lipases break down lipids. Amylases breaks down starch. I might be like, well, amylase, that does sound like starch. Well, actually, the name for starch sugars is amylose. So that makes sense. Amylase breaks down amylose. Protease breaks down proteins as pepsin and trypsin. Lipase breaks down lipids. Sucrase breaks down sucrose. Maltase breaks down maltose. Polyolting breaks down lactose. And people who lactose intolerant does lack that enzyme. Right, so we have the pancreas. Well, the bile duct connected to the liver. So the pancreas is the big boy organ here, because the pancreas actually secretes 3 different enzymes. So the pancreas secretes, well, more than 3, but it secretes pancreatic amylase, lipase, and trypsin. So the difference between trypsin and pepsin is that trypsin functions best at a neutral alkaline pH. Yeah, lactase is what breaks down lactose. So trypsin is kind of like the cousin for pepsin. They do the same thing, but they live in different places. Bile. So bile is not an enzyme. Bile is something that assists enzyme, right? So bile comes from the liver, is produced by the liver, and is stored in the gallbladder and is released through the bile duct. So what bile does, right? Bile emulsifies the fats. So how I say to depict emulsification, how to think of it is like this. Think of emulsification like this. It's like taking bread or mayonnaise and spreading it out on butter. Spreading it out on bread. It's like taking butter, and spreading it out on bread. Think of that as emulsification. It does not break down the butter, but it makes it easier. Like it turns it into this big glob and breaks it down into smaller droplets. It basically increases the surface area. It's like taking some butter and spreading it out on bread. It doesn't break it down, but it increases the surface area. That's kind of like emulsification. So, come in here now. Bile is going to emulsify the fats. Basically make them into smaller droplets, make them kind of softer and more spread out. And these fat enzymes, these are lipases. These lipases are secreted by the pancreas and some by the intestines itself. Now I'm going to break it down. So what happens now? Well, now that most of the food has been broken down, we are going to send it to the ileum. So the ileum is basically renin. What renin? What is a baby? Only babies have renin. So the ileum is what does the absorption of the foods. So this will happen through mainly 2 processes here, which is diffusion, simple or facilitated diffusion, and active transport. Those are the main 2 things that are going to take this food, these particles here, these broken down particles. Remember, we're taking with big zinger, we're breaking down the starch into glucose, and we're breaking down the proteins into amino acids, and the fats into fatty acids and glycerol. We're basically making them into monomers. We're making them real simple and soluble and small so that could go into the bloodstream. Anyway, it was not zinger we're eating here, it's a fish. So the fish, the proteins into amino acids, the fats into fatty acid and glycerol. Alright, since we boy asked here about renin, renin is used to curdle milk. So renin is actually an anabolic enzyme. So babies don't eat nothing, right? but you still have to go and buy real diapers and Huggies in the grocery, because when you have a baby, it is poop every day. And then you have to clean that poop and you have to wipe the baby clean and make sure the baby don't get any rash. But the baby are eating, and like the baby going and the baby eating pale or something. So how come the baby pooping? Well, milk is constituted of curds and whey. And the baby now, they has to use the nutrients in the milk. That's the proteins, the fats, and the lactose in it, in order to get energy and for growth and the vitamin D and the calcium and whatever, right? But the thing is, the baby digestive system is still very primal in its state. It's not fully developed. So what you need to do is that what renin would do is make those milk molecules bigger. It would curdle them, and it allows the molecules to go more slowly through the ileum. So it can be properly absorbed. That's what renin does. That's why babies poop. That's why you have to buy so much diapers, and make sure that when you're having a baby, make sure you have enough money to do so. So the ileum now is going to help absorb these nutrients, and this is going to happen through these projections that are called villi. So these villi here are filled with a very highly vascularized. They have a lot of capillaries in them. They have a large surface area, and they also have microvilli. They have smaller versions on them. So that's going to take all of these and put it into the bloodstream. There's also something called a lacteal that's going to absorb the lipids. So what we left with? What we left with is poop. We left with water, and we left with roughage. Roughage is fiber. Humans don't have cellulase, so we can't break down cellulose. So in order to break down cellulose, you typically need cellulase, and you need 4 stomachs like a cow. We don't have that. When is the first break? That's up to all you. So what we need to do with this poop now, what we need to do with it is remove the water. So this is going to take actually a good few hours to pass through here, depends on what you eat. Then it passes into the large intestine. The large intestine here is made up of the colon and the rectum. So the colon is used mainly for one thing, to absorb any water and to absorb any vitamins that are dissolved in that water, namely vitamins B and C. So it's going to pass through here the colon, and not all the water, because if you absorb all the water, you'd have a very bad time at the toilet, right? And this is usually going to be due to osmosis, osmoregulation, thing like that. So you're going to absorb some, because you don't want to be constipated. And then finally, we're going to go into the rectum. So the rectum now is where the indigestible solid waste is going to be released from the body. It's going to be stored and it's going to be released through the anus. Give me what's the word for when feces is expelled from the anus. Right? Give me the proper word. Give me the proper word. Don't give me the word you just use home. Give me the word you write in the exam. When you remove any solid indigestible waste. Very good. So that is called egestion. So egestion is the removal of, right? So not excretion, because excretion is removal of metabolic waste. That's like urine and carbon dioxide and calcium oxalate, which will give you kidney stones. That's like things like that. Egestion is removal of solid waste. Okay. So let me look at this list of enzymes here. Amylase, which is found in the salivary glands and the pancreas, breaks down starch into maltose. Pepsin, which is found in the stomach, breaks down proteins into polypeptides into amino acids. Trypsin does the same thing, but trypsin is secreted by the pancreas, not the stomach. So trypsin is alkaline, pepsin is acidic. Now they had a question that come once. They say, well, why do we need multiple enzymes to digest proteins? So this is because proteins are quite complex, and they require they have several structures of proteins to be broken down. And Kerwin. Well, I don't know. Maths tomorrow or something. Anyways, only me who does them last minute exam. Them last minute exam reviews. Alright, so this is why I need multiple enzymes to digest proteins, because proteases tend to be very specific in the pH that they work at, and different parts of the body have different pHs. So most of the alimentary canal is neutral or alkaline, right? And only the stomach is acidic. There's some other acidic parts of the body, so like the vagina is acidic. So like when sperm is in the vagina, they're basically having an acid bath. And only the ones, could survive is the ones that will go forward. Lipase breaks down fats into fatty acids and glycerol. The last one here is catalase. Catalase is produced by the liver. Catalase is actually what's making hair gray. Not catalase is making hair gray, sorry. Hydrogen peroxide, which builds up in your body as a waste product, is what's making hair gray. I have a few grays on my beard right now. And as I get older, you produce less and less catalase. It breaks down hydrogen peroxide into hydrogen and water. So if you actually take a little piece of liver and you put some potato, you put some you put some hydrogen peroxide on it, it will actually break down into little bubbles, hydrogen and water. Potatoes also produce a little bit of catalase. So if you take some hydrogen peroxide and you put a piece of potato in it, you will see bubbles forming up. You see bubbles coming up. Those bubbles are the hydrogen. You see like little foams that's be coming up. Right. Now I ain't about to go through diet and thing because I feel like everybody should know that. But it's one thing that you should take note of that other people forget. The two plant nutrients that you have to know. So the nutrients nitrates or nitrogen is used for growth of shoots and leaves, because nitrogen is actually one of the molecules that's in proteins. So usually nitrogen is used in a nitrate form, and nitrate is basically kind of like ammonia, right? It's not ammonia, but is ammonia would turn into nitrates. Magnesium is used for chlorophyll production. So it is actually what makes the leaves green. When plants don't have magnesium, the leaves turn yellow, and that is called chlorosis. I wouldn't bother go through food tests because I assume everybody do food tests in a lab. And that's nutrition. The only other thing I guess you should look at is how the villi is adapted for absorption. So the villi has these little microvilli on them that again increases the surface area. It has a very, very thin layer of epithelial cells that allows easy diffusion. And there's a little lacteal in the middle. It's kind of like a blood vessel, but it's a lymphatic vessel that absorbs fats, dietary fats. Okay, so I go about to go through teeth and thing, but all you should learn how to draw the tooth, right? Now tell me this. Put it in the chat here. After absorption, we need to take this food, we need to take these nutrients into the cells. Give me the word for that. So after we absorb this stuff into the bloodstream, we need to take the nutrients into the cells. Give me the word for that one. Nice. So that's called assimilation. So assimilation is basically this. So let me open my palm. So if you look here, imagine this, right? So blood is being pumped out of the heart, And this is the digestive system here, right? So let me say that this is like the ileum down here. So the ileum now is going to take in a bunch of nutrients. So what we need to do is take that nutrients and send it into the liver. So the name of that blood vessel that takes the nutrients and sends it from the ileum to the liver is called the hepatic portal vein. The word hepatic means liver, of the liver. So what happens here now is that, why do we need to do that? Why do we need to send it to the liver first? Well, the liver has a function that it removes toxins. It removes any waste material that includes things like alcohol from the bloodstream before it gets sent back to the heart or before it gets assimilated by the rest of the body. So the liver there has many functions. It stores glycogen, it releases glucagon, it produces bile, but one of the other things is that it removes toxins. And so this is why, and this has happened throughout time. So when granny given you, or when auntie given you, or mommy given you a flush out from Traversiers or some kind of thing to buy in the market, and they say, well, this can help cleanse your blood and flush you out and things like that. Once you have working kidneys and once you have a working liver, you don't need need that, right? So once you have a functioning liver, that's gonna remove as much waste as possible from your body. Okay, so that wraps up nutrition. So the next thing, I feel like some of the things here I don't need to really go through. I need to go through biotic and abiotic factors, I think. I feel like we're okay with that. But let me just touch on it like real briefly. Abiotic factors is non-living factors, right? So factors that is of the ecosystem, but it doesn't involve living things. So it doesn't involve plants, it doesn't involve animals, it doesn't involve decomposers. So put some abiotic factors for me in the chat now. And see if anybody could name this one for me. So you know you have water, and sometimes the water just be like real muddy. It have a T word to describe that water. To describe the factor of that. Alright, let me go through it briefly then. CO2 concentration is one. Well, soil a little vague because soil could contain living things. But if you say things like soil, pH, water salinity, that's a good one. Water salinity, temperature, sunlight availability, water availability, nice. And the one that the one with like how the muddiness of the water. So like how Cascade could only really live in. Alright, so not turgidity, turbidity. So turbidity is our next one. Also give me the word for when we have water that is only slightly salty. So like where a river would meet the ocean, we just call that something water. What's the word for that? Topography could work too. We just call it something water. Like when a river meets the ocean. Where fresh water does meet fresh water does meet seawater, salt water. Alright, hope get correct there. So brackish water is called it. So brackish. Alright, so do we look here? I'll just briefly go through it. Alright. An ecosystem is a combination of two factors, right? So you have biotic factors and you have abiotic factors. Biotic factors are basically the living things. The word bio means alive. So biotic factors produce their own food, they eat organisms. This is like a predator, your prey, your plants, your bacteria, your animals. All these living things are in the organism, in the ecosystem. All living organisms. Basically things that comprise a food web. Abiotic factors are the physical features of the environment, the non-living factors. So these are things that though present, they could affect the numbers of the species populations. They could affect the symbiotic relationships between these organisms. It could cause separations between two different species. So like it could be like if there's a big pond or something between two organisms and they can't meet, you know, that's an abiotic factor. If there's a area with very little water, that's an abiotic factor. So that's all it is. Okay, so not bad. That is what it is. So the other things I had to know is with soil, and the abiotic factors in soil would be things like the air spaces in the soil, the water availability of the soil, the pH of the soil. And I guess the biotic factors would be like the amount of decomposers at present, the amount of larger organisms like earthworms at present. So nothing difficult. How much like our next abiotic factor would be like the mineral nutrients at present, things like that. That's it. Food chains. I feel like I don't need to go through food chains earlier. That is wasting time. So food chains, that real easy. All you have to do is make sure with a food chain is that the producer does go first. Whoever consumer is the last one, whichever one is the biggest one, the one that receives the least energy, that one does go last. So is 10% our energies be transferred through each level. So we always start with the producers, and the producers get energy from the sun. So we do say we say that the sun, they say, is not a cycle, it's an energy flow. It's not a cycle because none of this energy actually returns to the sun. So producers undergo photosynthesis, primary consumers eat producers, secondary consumers eat primary consumers, so on and so on and so on. We need to go through food chains. All are good with that? I feel like predator-prey relationships, I don't know if we and decomposers, I think we're okay with that. But let me just explain something here with predator-prey. Alright, so predator prey. Hey, all don't worry now. We have until 10 o'clock today. We have until 10 p.m. to go through everything. All go and sleep now. If you want continuity and variation, go and sleep and wake up 8 o'clock. And you'll come back. Maybe go and sleep now. Go and sleep and come back 6 o'clock if you have diseases. All you have to do is watch Terrence Dial sing. And you'll learn plenty about diseases. Alright, so you look at predator-prey relationships. So here we have population size, right? So this is something that has come sometimes. What we notice here, we have this kind of sinusoidal shape here for both of them. But what assumptions we could make here? The assumptions we could make here is this. Prey populations are almost always higher than predator populations, right? So prey populations have to be more, because if you don't have enough food, you wouldn't be able to survive. So predators need food. But what happens to alright, so let me explain what happens here. So I want you to imagine this. The prey is how much food it have at the party. The predators is how much people it have at the party, right? The prey is how much food it have at the party, and the predators is how much people it have at the party. So it have plenty food at the party. So that means more people going to come to the party. If we have a high prey population, we're going to get more predators, because it have more food. Now look at what happening here now. When we have more people coming to the party, we have more people eating food at the party. So when predator population increases, prey population going to decrease. Right? So when we have predator population goes up, prey population is going to go down, because we have more people at the party, so we have less food at the party. So what's going to happen now? When prey population decreases, right? So when prey population decreases, predator population is going to decrease. Right? So when prey population decreases, predator population is going to decrease, because it has less food. So if you go to a party and have no food and have nothing to eat, well, you go on, right? So, but look what happened now. Because there's less predators, there's less people to eat the food, there will be more food. So these prey now could come out and party and say, well, you know, it had nobody around to eat with, so we're going to reproduce. So what's going to happen here is when the predator population drops, right? So when the predator population drops, the prey population is going to go back up. So it's a cycle. So when the prey population drops, the prey population is going to go back up. It's a very simple cycle. If there's more prey, there's going to be more predators. If there's more predators, there's going to be less prey. If there's going to be less prey, there's going to be less predators. And if there's less predators, there's going to be more prey. So that is what the cycle is. So that's the predator-prey cycle. Okay, and that's it. That's really why you have to know about predators and prey. So we have our next thing here. That's about biological controls. Yeah, chill out now. Okay. So I remember one year they brought this biological control question and almost everybody get it wrong. So let me go through it here. Like real people get trouble. People didn't even know what it's talking about. What's a biological control? I say something that could actually come. Alright, so if you look here. Jokes. So what's a biological control? So I'll give you a little history here. In the 1990s, right? Trinidad had something called the mealybug. And let me put it in paint here. Okay, I picked it. So they had this kind of white, fluffyish looking thing called a mealybug. There's a mealybug. And this mealybug not really found in Trinidad and not really found in the Caribbean, actually found in the Philippines or so. And we people when they're bringing in the fruits and things from away, that's why they just get on so dread when they're bringing in stuff from away. You have to make sure and check it on it because all kind of pests and things could come in. So what happened now is that this mealybug now, when it come here, it start to obliterate a lot of the crops that was in the Caribbean. It start to eat a whole lot of the crops. It have a name for that type of species earlier. Give me the name for that type of species. So like when something that don't really belong somewhere, they end up there, but they end up eating out a bunch of the organisms and disrupting the whole food web, the whole ecological balance. So it have a name for that type of species. So the mealybug basically come in here and they didn't know what to do about it because it will really only have so much yeah, it still have it here, they still have it, but they're not in as large numbers now. So they didn't know what to do. Alright, very good. So they are invasive species. So kind of like the lionfish, there's an invasive species. And what happened now is that there's only so much pesticides and so much chemicals they could spray. And what they go to spray the whole Caribbean, they try to spray the place for mosquito and it still has so much mosquito. So what happened now is that they say we need to find a next way to get rid of this thing. Well, a good way of population control is, well, not the COVID virus, but a good way of population control is to have a predator, to introduce a predator, or at least to increase the population of a predator. But that could be a little dangerous. If we increase in our predator population, we run the risk of the predator itself that's going to eat out all these mealybugs. We're going to run into the risk that the predator itself become a pest. So they say, well, a good predator that is not really a pest, and one that is a natural predator of the mealybug, is this one here. I'm not posted it. So all it know is the name. What else? Eh? Right. Time what was the name of this one here? So they say that if they bring in plenty of these insects here, this is going to be a good natural predator. And this predator here, we're going to do is that they're going to feed on the mealybugs. Yes, this is an example of a biological control. So you're going to feed on the mealybugs and you're going to control the population. So you understand the term now is a biological control. It is something that is a natural predator that we're going to put out there to control the population of a pest. So this is a ladybug. So the ladybug is going to be the biological control. And we could still see mealybugs and ladybugs today, but they are very, very controlled levels now. They're basically in a kind of equilibrium now. So the ladybugs don't really have much other things that is eat here. So once all the mealybugs they get eat out, the ladybug population will drop because the ones that could not find any food, they would have died out. So yeah, it's kind of like that, Maya. I guess the cat could be, except if you imagine you have a country full of rats and you release a bunch of cats to get rid of them. Yes. So a biological control, what we have to know is the advantages and disadvantages. So let me look here and see what's the advantages and disadvantages of this. Now all of this falls under predator and prey. Okay, so if you look here, the thing about a biological control is that you could target a specific pest with it. So if you in this case here, the ladybug could have targeted the mealybug and nothing else, because the problem with releasing a biological control is that what if it end up targeting a bunch of other things that we need? What if it end up targeting butterflies, or it end up targeting bees or something like that, that's needed for pollination? So biological controls don't result in any pollution, because it's really just an organism that you release. It's not a chemical that you're spraying. The other thing is that it's a long-term solution. Once the populations balance out, you don't need to hire anybody, and you don't need no government contract and thing paying every year to go and spray and go and take care of this thing. So basically, is an ecological solution. The thing will take care of itself. Disadvantages is this: the research expensive. So you would need to put in, you need to hire people who actually know what they're doing. You need people to be able to research food webs and see what exactly this biological controllers eat. Because it have cases where, they release something, and that something now is a pest of its own. It takes a long time to work properly. So it's not like it can work tomorrow. It might take a couple years, a couple months to a couple years to work, because populations have to build up naturally in order for it to work. So it takes a long time. If you just want to spray the thing, it can work in a short term. It can only work temporarily. The other thing by using chemicals, by the way, is that natural selection. So when you're spraying these insects, the strongest ones can survive, and those are the ones that are going to repopulate, and they're going to get stronger. The cockroach, basically. If you had a nuclear fallout and then basically everything mashed up in the world, the one thing that's going to survive is the cockroach, because it basically get killed so much times. It know how to come back. It know how to survive any number of things, including radiation. Alright, and I say it could cause disruptions to the food web if you don't know what you're doing. So this is something that could come. You never know. Let me paste a little thing there in the chat. But all of this here I could find in the little study document that link, right? Right, so we have, that's like the advantages. And there's the disadvantages. Alright. Alright, so let me go on to decomposers. Yeah, well, take a nap if you want. Alright, so decomposers. Oh, Candace is probably a man looking for attention, right? So they don't get chained up. Let me put on something else. Let me not put up nothing that go get the stream takedown. YouTube real dread now. Alright. Let me go to decomposers. So first is, well, what exactly is a decomposer? Alright, that's not it. I'll make sure and see what's showing there, right? I feel decomposers could just be like a smaller part of the paper. So decomposers basically is organisms that break down the dead remains of other organisms. The one thing that you shouldn't mix it up with is carrion feeders. So carrion feeders is like kobo, well, vultures now, or like earthworms or hyenas that eat dead organisms. So it's not the same thing. Decomposers chemically break down. So they chemically break down dead matter, and mostly into carbon dioxide. So they actually is an integral part of the carbon cycle. So organisms obtain nutrition by breaking down the dead remains of other organisms. So these is usually bacteria and fungi. So it have a name, it have a next names for them. Start with S. So like fungi that break down dead organisms. It have a word that start with S. Give me that word there. Something that lives on dead or decaying matter. Yes, keyword is chemically, so not mechanically. Alright, very good, Surya. So saprophytes. So saprophytic nutrition. Right, so saprophytes. We should be here in Let's Talk from Talash. Right now, Talash on a 5. Hopefully, we could get him up to a 4 or a 3. But we're looking for something higher than a 3. Eh? I mean, all I want all this CXC slip to look like a forest and have a bunch of trees in it. We want to get 1s and 2s. Okay. So yeah, you don't really have to know much about decomposer. You have to know that temperature affects the rate of decomposition. So decomposers won't work in very cold temperatures. So that's why when you put stuff in the freezer, it don't decompose. When you put stuff in the fridge, it decomposes very, very slowly. You can't leave it in the fridge for a year and expect it to be good. So decomposers like dark, warm, and moist. That's usually the 3 conditions. But of course you need a food source too. Alright. Let me go into let me take a 5 minutes. Let me take a 5 minutes and then I'll start diseases. Diseases is topic number 6. I don't do lessons for Cape as not teacher. You would pay $1 million. It would probably have to be more than that. Because I mean, I have much more than that. Alright, we started back in 2 minutes. Rahul, I don't think you're again sampling methods. They're exactly pretty specific about what you're getting in ecology. From what I see. You get into draw food chains, you could do what you want, buddy. It's your life. If you want to draw a food chain, draw one. We're coming back from the break now. Just taking our 5 minutes. Alright, what I do, I'm a writer. We do have to do cells, Marisha. Alright, Talia. So let me go through diseases. Okay. Let's now, diseases is quite easy though. So they have diseases before transport and all kind of thing. That's why I say I feel like they're giving you the questions, like how they put the topics is in the order all again. At least I think so. Alright, Olia, so you should be seeing nice. Dislike ratio. I don't know like what this I don't know how much people on here. I don't even know how to use a computer. There's like how much people in here? Like 10? Not a teacher, I don't know how to use a computer. I don't even know why I'm doing here all this Sunday afternoon, but I say I'll come on and help all you out. Because as a CXE real bad, but them is the baddest I tell you to still have exams in this time. And I have the knowledge. I ain't doing nothing with the knowledge because I was not a teacher, but I might as well share it with all you. Remember, as I say, I'm doing a physics one too. A day on the channel, I can't remember what date it is. It's like the Sunday before physics. Alright. So we have 161. Okay. That's cool. So let me go through diseases now. Diseases is a topic that they tend to bring. I tell you the types of questions that they like to bring. They like to bring what are the different types of diseases and how you can manage them, and to give examples. There's also like to bring stuff about vaccination, which is very, very relevant today. So basically stuff about active and passive immunity. What else I like to bring is linking it to diet, which is nutritional deficiency diseases. Yeah, don't worry, I know physics and biology and thing inside out. Don't ask me nothing about chem. I know nothing about chemistry. Chemistry is the devil. Not me and that. I only know chemistry that relate to biology. I know all them other stupidness and thing. I could barely balance an equation. Okay. So let me see if we can name some diseases. So let me try to go alphabetical, Post some diseases they're familiar. We're going alphabetical order. Alright, so let me see. So A, let me try to name one disease from each one, right? So A, there's a disease where you have a low iron. You have a low iron in your diet. And because of that, your red blood cell count is very low. The disease that we have for B, this is when your tubes that lead into your alveoli, they get inflamed. Tell me what's that one that started with B. Alright. C, this is so not COVID, but this is like if you go on and drink drain water, basically water that infected with pathogens, not AIDS only, not AIDS. If you have a low iron count, anemia, right? So if you go on and drink drain water, you could get C. Alright, D, if an Aedes aegypti mosquito flying in the night and they bite somebody else who have this disease, and they bite you and you get this disease. Tell me what's that one? Alright, E, this is where, let me see with E, boy. Alright, so E is where, give me one for E. Alright, B is bronchitis. For some reason, E is a skin condition. A skin condition that, it's got a lot of bumps and a very itchy, could come there. F. Alright, so F, I go put this one as cystic fibrosis. Alright, G is the big word. G is the big word for stomach flu. G is the big one for stomach flu. Alright. H is where there's a little cool sores on your lips. You're really taking on you have multiple partners and you can end up getting Alright. So E is eczema. I, the big name for flu. The big name for flu. Game was I. The big name, right? So G is gastroenteritis. I is the big name for flu. Alright, so J, I don't really have one for J, so I'm going to leave out there. I know you have like jaundice something, but that's not disease. Alright, so K, when you have a protein deficiency, so H is herpes. When you have a protein deficiency is J. Alright, L is get from rats. It's like a rat pee in your food. So like in Price Plaza, when the rat did pee in the woman pizza and she still eat it and then she dead. I mean, she didn't know it had it, but yeah. Alright, so I is influenza. Hope no, again no influenza. I think it's COVID. Alright, so M is where Anopheles mosquito bite you. That infected with this. K is quassia. I put in jaundice because jaundice is not actually a disease. It's a symptom for a disease. Alright, L is leptospirosis. Alright, so N, let me leave out N. O is when you have a BMI of over 30. Actually, I could put one for O, so I put one for N. N is I'm trying to remember what it's called. Liver. Right. So we have I lost the stream there. Right. Okay. So N, O is obesity, M is malaria. Well, one nearby. Alright. Okay, so M is malaria, O is obesity. Alright, then we get one for P. So P is like so what's one for P, boy? I'm trying to find one. I want to use palsy. Alright, so P is when a flu get real bad. When a flu get real bad. You guys end up getting this one with P. When a flu get real bad. Leave out Q. R when he's get this kind of mark on his skin is a fungus and it could result in a rash. What's that one? You could get a rash. What's that one? Pneumonia. Good. Alright, S is a sexually transmitted disease, and you know it could get a nervous system shut down. It can lead to death actually if you leave it for too long. You get like a high rash that go on anybody. T, long time they used to call it consumption. You call it consumption, but nowadays you know you don't really see it again because the bacterium not that present anymore, but it's basically coughing up blood. You have a real bad cough. Again, it's not COVID. Alright, so leave out U. Alright, V is what way? Oh, V is this thing that your skin is turn white as a result. Your skin is turn white as a result. And Michael Jackson had it. You just see people in Trinidad, let's call it Juta. T, tuberculosis, tetanus could work too. Alright, and if I could put one for W, you know. Forget X. A, somebody good. Vitiligo. Alright, so W, me for W. Alright, X, leave out X, leave out Y, leave out Z, and Z could be Zika virus, right? Okay. So here we have a list of diseases. So there's 4 types of disease. Right, so you have 4 types of disease. We have infectious diseases or pathogenic diseases. We have nutritional deficiency diseases. We have hereditary diseases. And we also have the last one is physiological or lifestyle diseases. So we have 4 types of disease here, right? And what we're gonna do is we're gonna put a little I by the ones that are infectious. We're gonna put a one with N that deals with nutritional deficiency, a H with hereditary, and a L for the lifestyle ones. So just to explain each one first. Infectious diseases or pathogenic diseases are ones that are caused by pathogens. That's bacteria, viruses, or fungi. Nutritional deficiency diseases are when you lack a certain nutrient. So like if you're lacking proteins or you're lacking vitamin A or something like that. Oh, I can actually, let me take out this one. Speaking of that. Right. So hereditary diseases are the ones that are due to mutations. So due to genetic mutations. And physiological or lifestyle diseases are the ones due to the malfunction of an organ. Okay. So anemia. Anemia is a nutritional deficiency disease. It is due to the lack of iron. Anybody? what happens is that you have a low red blood cell count, a low hemoglobin count, so you can't absorb as much oxygen as before. You tend to feel very faint. Bronchitis is a pathogenic or infectious disease. It could actually be pathogenic or it could be lifestyle. It depends on how you get. But a lot of it is due to infection, like due to breathing in smoke or particles, and you get inflammation in your lungs. By the way, anything that ends in itis means inflammation. So gastroenteritis is inflammation of the stomach lining. Hepatitis, you have dermatitis, inflammation of the skin. Gingivitis, inflammation of the gums. Hey, xerophthalmia, boy, we have a, let me put that one. Xerophthalmia is kind of like night blindness. It's like when the front layer of the eye gets highly keratinized. Alright, cholera is caused by a bacterium, so this is an infectious disease. Dengue is caused by the dengue virus, but the Aedes aegypti mosquito does carry the dengue virus. What's the put in the chat there? What's the name of the type of organism that could carry a pathogen but not suffer the consequences of the pathogen? So these are infectious. Alright, so eczema. Eczema is like when you have a skin condition, you tend to have like a lot of inflammation and things. The thing about eczema is that usually you don't really get through a bacterium or something like that. You exactly get eczema. Right, so that's a vector. Very good. So eczema is actually a hereditary disease. It's due to a mutation in a gene, and because of that you end up with your immune system kind of fighting itself, and you end up with all this bumps on your skin and rash and things like that. Cystic fibrosis is a hereditary disease. So cystic fibrosis is basically this. When you imagine this, there's a protein in a cell membrane that shuts off, and what happens is that it produces mucus instead. So because it produces mucus, what happens there is that the lungs fill up with fluid very, very quickly. There's a high risk of infection in the alveoli, but it's a hereditary disease. So that means if somebody have it, you can't get it from them, I guess, unless it's a mother. I have no idea who is Mr. Singh. Gastroenteritis is caused by a bacterium, so there's an infectious. Herpes is a STI, sexually transmitted infection, so there's an infectious or pathogenic disease. Herpes is caused by a virus. Influenza again is infectious. Kwashiorkor is a nutritional deficiency disease. Put in the chat there when somebody has kwashiorkor what they lacking. Leptospirosis is caused by the Leptospira bacteria. So as a result there, this is an infectious disease. The rats is the vector. Malaria is caused by a protist that is called Plasmodium falciparum. So again, it is an infectious disease. Jaundice is not really a disease. Jaundice is more of a symptom or a sign for a disease. A sign for a disease. So that's why I put jaundice. Proteins, good. So kwashiorkor is when you're lacking proteins. Night blindness is a nutritional deficiency disease. You're lacking retinol or vitamin E. Alright, so we come into our first lifestyle disease here. So obesity is when you have a BMI of more than 30, and what happens is that the adipocytes, which are the fat cells, tend to produce a lot of chemicals that cause inflammation. So it is considered a disease. And opportunistic infections like pancreatitis, liver failure, and so on could result from it. So it is a lifestyle disease, which usually is caused by lack of exercise and high carbohydrate or high lipid diet. Pneumonia is an infectious disease because again it's caused by the influenza virus, and when it becomes more powerful than anybody. Ringworm is an infectious disease which is caused by a fungus. Syphilis is an infectious disease because it's caused by a bacterium called Treponema pallidum, which is right. So it transfers from one person to the next. Tuberculosis is infectious because it's caused by bacteria. Vitiligo, if you don't know what vitiligo is, what that is, when you see people have like spots that don't have melanin, and the melanin tends to wear off over time. A lot of people think Michael Jackson bleached his skin, so he not like Vibes Cartel and use blue soap and try to rub off the melanin, but he had vitiligo and eventually his whole, epidermis turned very pale because he all the melanin was lost. So vitiligo is hereditary. And Zika, well, since it's transferred by the mosquito, this is infectious. So we only had like one lifestyle disease here though. So let me see if we could name some more lifestyle diseases. So let me go through the notes here. If you want halftime, you're free to go halftime, Hey, that's a good one, and Sarah. Yellow fever, yeah, and yellow fever would be infectious because it's transmitted by the Culex mosquito. Right. So like stuff like diabetes, atherosclerosis, hypertension, all of those there are lifestyle diseases. Those are due to the failure of an organ. Right, the failure of tissues and organs. So cholesterol is not a disease, but high amounts of cholesterol can lead to those things. Coronary heart disease is one, or xerophthalmia. I'm gonna come back there. Xerophthalmia is a nutritional deficiency disease, lack of vitamin A. Why yellow fever will be infectious? We do plenty. So there's really only a handful that you really have to elaborate on. Okay, so let me focus on what exactly you have to know about these diseases. Albinism is a hereditary disease, Johnny from the 8. So pathogenic or infectious is caused by pathogens, bacteria, viruses, and fungi, and some protists. Both of those are hereditary. Alright, so, and Sarah, to answer your question there, cancer is kind of like its own class of diseases. You'd have to consider each individual one, but cancer is usually considered physiological because it is due to the failure of an organ. But there's exceptions. So for example, cervical cancer is caused by a virus that's called HPV, human papilloma virus. So that one would be considered pathogenic. So basically, if you're a man who do have a cervix, right? But a man could give a woman HPV. So he could pick up HPV from one woman, have sex with another woman and give it. But he himself can't get cervical cancer because he don't have a cervix. So HPV causes cervical cancer. So that one is infectious. Basically, if the man don't bathe and he don't wash himself properly, is high risk of transmission for it. Right. So pathogenic or infectious. What is the prevention methods or treatment? So quarantine, we well aware of that. Face masks, well aware of that too. Vaccination and anti-sera. Anti-sera is basically, we'll get into that just now. Anti-sera is the plural of anti-serum. Antibiotics, antiviral drugs, and antifungals. Nutritional deficiency, all you're well aware of this. There's like your rickets, your night blindness, kwashiorkor. If you don't have vitamin A, you get night blindness. You don't have vitamin B, you get beriberi. You don't have iron, you get anemia. So easy fix, you just need a balanced diet or take nutritional supplements. I'll give you an example of this. A lot of people in Indonesia suffer from night blindness. So this is because they don't have a lot of vitamin A in their food. And what happens as a result is that they can't see in dim light very easily. Not that they go blind or anything, but they genetically engineer the rice there to contain beta carotene. They call it golden rice, and the rice itself, the rice plant can make vitamin A, and the people could eat it, and they could get not a lot, but they could get a decent dose of vitamin A in it. So just having nutritional supplements and getting dating could hopefully, once it's not too far gone, reverse the supplement and reverse the symptoms. Hereditary diseases, right? So hereditary diseases are those that we'll get into these a little later on when we're doing continuity and variation. So hereditary are diseases that are passed on from parent to offspring. It has to happen through the genes. So this doesn't include diabetes. You know, like if your mother have diabetes or your father have diabetes, the chances are that you will have diabetes. It don't involve that, because that is not them passing on diabetes to you. That is them passing on the risk of diabetes. These hereditary diseases are due to if you inherit a recessive allele or two recessive alleles, usually from your parents, or there's a mutation that leads to it. So for example, hemophilia occurs when the child inherits a recessive H allele or two recessive H alleles. Cystic fibrosis, when they inherit two recessive alleles, and that leads to fluid building up in the lungs. So this is where we'll talk more about this when we get into here, but these things can't be fixed with medication or anything like that. The only way to really treat it is through gene therapy. Gene therapy is where they actually replace the DNA, and usually they would use a virus to carry the DNA into the body, or genetic screening, which is they could tell from before, they could get a little bit of amniotic fluid from the mother, and they will do a process called amniocentesis, and from that they could actually sequence the DNA from the amniotic fluid, and they could tell if the child would have so and so disease. So that's genetic screening. Physiological diseases they can't fix with medication. So it have no pill. They could make the symptoms lesser. It could reduce the symptoms, like by using things like blood thinners and metformin to control blood sugar. But there's no antibiotic or anything you could take to cure physiological disease. So typically, most physiological diseases are managed by having a balanced diet, having regular exercise, by reducing substance abuse, so like reducing alcohol and nicotine, and reducing carbohydrate intake, reducing lipid intake, things like that. So that's usually it there. But I think at this point, all you should know the four difference. So the remedy 4 is pathogenic, nutritional deficiency, hereditary, and physiological. Well, that could fix anything, eh? Okay, so the next thing now you have to know is the two types of diseases, communicable and non-communicable. Communicable diseases is like COVID. It can be spread from one person to another. So it either could be spread through contact, it could be spread through droplets, which is like sneezing and coughing, which is what all you should do during the exam, or it could be spread through vectors. So vectors like animals or insects that carry pathogens. Well, diabetes is not caused by a virus. Diabetes is caused by malfunction of the pancreas, or we'll actually get into that later. So, or the body not responding to insulin. So it's not like it could rise from its dormant state. It's just that you have an increased chance of the pancreas, going bad or getting affected by it. So some people, they do have that chance, or they have very low chance. Some people have higher chance. Non-communicable diseases are basically everything that is not pathogenic. These are deficiency, physiological, and hereditary diseases. There's diabetes, arteriosclerosis, hemophilia, sickle cell anemia, scurvy. All of these here are non-communicable, because if they will say I have diabetes, I cannot give you diabetes. as a result. If I have coronary heart disease, I cannot give you that coronary heart disease. Yeah, it's set up by like I'll get into that just now, like doing going through the two types of diabetes. Okay. One thing that frequently comes up is vectors. I say, well, what is a vector? A vector is an animal or an insect that can transport a pathogen, right, without itself feeling the symptoms of the pathogen. So, for example, a mosquito is able to transport, Anopheles mosquito is able to transport malaria. But malaria itself, the mosquito itself doesn't suffer from malaria. So the mosquito don't have headaches and damaged tissues and things like that. It basically just living inside it. So same thing with rats. So rats could transport the leptospira bacteria. But the rats themselves don't suffer from leptospirosis. So they wouldn't get nervous system shut down and things like that. So this is not the same as, let me say, a HIV positive person. So a HIV positive person is not a vector for AIDS. A HIV positive person is just a carrier for the disease, because a HIV positive person will suffer from the disease. A COVID positive person is not a vector. A COVID positive person is a carrier, whether or not they're symptomatic or asymptomatic. So that is the difference between a carrier and a vector. A vector doesn't feel the consequences of having the disease. They don't have the signs or the symptoms of it. Carriers do, vectors don't. So the life cycle of the life cycle here, you usually have to know it for a housefly and a mosquito. Both of them are very similar, but usually it's the one for the mosquito comes up more. So here we have the eggs. The eggs hatch and turn into larvae. The larvae turn into pupae, and then the pupa hatches and becomes an adult. Then the adult lays eggs and you have a cycle. Well, they learned this in primary school already. So what you usually have to learn next is what are the yeah, that's correct answer. Is how to control each one. So eggs now, very easy control method. All you have to do is drain any stagnant water sources. So once the adult mosquitoes don't have a place to lay their eggs, you wouldn't have any eggs. And that's probably the easiest fix, because if you don't have eggs, you won't have any of the other stages. Because the egg is a dormant phase, which means that it's not moving. So it's the easiest to get rid of and the easiest to prevent. The larva, however, is an active phase, which means that the larva actually that's a good question, Matthew. Are they a vector? They probably would be. Honestly. That's a good point to bring up there. Larva, basically you need something that like a predator. It's kind of like you need a biological control. So if you have a pond that's full of mosquitoes, introducing like fish, like koi fish, they would be able to eat all the larvae that are there. The other thing, which is not really the thing that you want to do, is spraying larvicide. Basically like a pesticide on the water, but if you have fish in there and you have other organisms, it could be toxic to those organisms. The pupa, same thing. Since it's a dormant phase, it could drain the water sources. But at this phase here, they're basically in a protective layer. So using pesticides and things won't work at this phase. The adult, which is what we've accustomed most to dealing with, well, spraying bop and death and fish and whatever else it have. Using electrical zappers, and this don't kill them, but sleeping under a net to prevent infection. It wouldn't kill the mosquitoes, obviously, but it could prevent the spread of the infection. We reach diseases, Dion. Right. Other things that there's asked about is impact of diseases on society. Look, they just have to look at Trinidad. How much money we burn on COVID already? The economy plummeting. Nobody could afford nothing. When we starving, we have nothing. we tourism sector would be zero if we still have COVID. So we have a lot of things. We have reduced household income because people have to pay the medical bills, if they have to try to get into the hospital, if they have to pay for quarantine, if you basically own a bar, you're working in a bar, or you're working in a restaurant, or where again, boy? If you own a gym or you're a gym instructor, well, hard luck there, yes. Yeah, your household income drop right down. Food, yeah, reduce food availability because if farmers and so on get sick, agricultural sectors are affected. You also would have restaurants closing down. I mean, what we have here is basically, I write these notes 8 years ago, right? And everything that you're seeing here happened. We have reduced income from tourism sectors. Who the hell want to come Trinidad? And economy, state-funded medical care. We have all these step-down facilities here. We have all this COVID tests that are performed. We have all these vaccines that had to give out. We had to pay China for all this Sinopharm. We had to take whoever expiring from Grenada and Bermuda. All of that there, quarantine can result in businesses closing down. We have a reduced labor force. We have all these people getting COVID and they can't work. And of course, we have the quality of life itself. The emotional toll of illness is nothing to really laugh at. I'm not joking about all you getting COVID from CSEC, which is probably true. But the quality of life will go straight down because being quarantined for lengthy. I read this thing 8 years ago. Being quarantined for lengthy periods, I'm sure you're feeling mad, like all you're going mad. The deaths, the fact that you can have funerals. Well, not funeral, you can have usual funeral, but people who died from COVID, they can't really have a funeral. You just have 5 people, you do the rites real quick and you move on, you basically schedule. So these are the 5 things if they ask, well, how does disease affect a country? So you have economy, you have tourism or trade relations, you have the food sector, you have household income, and you have quality of life. Very easy. Okay. HIV and AIDS. Alright, HIV and AIDS. Honestly, they don't bring so much about HIV and AIDS. You're trying to sue CSEC? CSEC will counter you and they will win. CSEC is more powerful than you, and you just have to take it. CSEC have a much higher power level, over 9,000. Your power level is probably 2 compared to them. Nah, the best lawyers can't, the better than the best lawyers. CSEC is basically immune from everything. CSEC not forcing you to have exams. That's your fault that you want to write exams. They could have deferred. But you so want to write exams, you so love exams, that you're writing it. And that's why you're here today. So you can't sue them. Hard luck day, boy. Alright. So, first, before I go into HIV and AIDS, right? We have to understand what's the difference between bacteria and viruses. Bacteria are much bigger than viruses. So you can actually see bacteria using the proper microscope, like using an electron microscope, you can see bacteria. Viruses, you can't really detect them using a microscope. You could, but only the bigger ones. So they're very, 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 very small. So let me say this. You're going for AIDS test, right? To see, if you're HIV positive. They don't really take a cell and a microscope and see if you have, see if they could see the HIV, That's not what it does do. So you see what HIV does do is that it is infect your lymphocytes. If you remember, your lymphocytes are your white blood cells that secrete antibodies. So HIV, when it goes into your antibodies, imagine the antibodies is like the police of your body. Alright, your lymphocytes are like the police of your body, and the antibodies is the guns. So what AIDS do, or what HIV does, is that it allows the police to kill each other. So it trains the guns on each other. So what happens is that white blood cells don't do the job anymore. They don't fight any bacteria or viruses anymore. Infected white blood cells, what they instead do is they end up killing other white blood cells. So if white blood cells was the police of your body, HIV scrambling the brain and telling white blood cells kill other white blood cells, telling police kill other police. So the antibodies is like the guns that the police have. So it basically is shooting out antibodies to other white blood cells and killing them. So why is there no cure for it? Well, this is because antibiotics don't work against viruses. Antibiotics only work against bacteria. So an antibiotic could work against tuberculosis. An antibiotic could work against cholera. An antibiotic could work against syphilis. Antibiotics could work against dysentery. But antibiotics cannot work against the flu. Antibiotics cannot work against dengue fever. Antibiotics cannot work against COVID because those things are caused by viruses. So, for example, malaria is caused by a protist, which is kind of like a bacteria, just more complex version. But you have an antibiotic called quinine that could be used against it. Popular antibiotic is penicillin, which could be used against a lot of diseases. A virus mutates constantly. So as you know, for COVID, we have the alpha, the beta, the gamma, the delta variants. And what happens is that a lot of the antiviral drugs or the vaccines could be rendered useless against them. The thing about AIDS is that it mutates a lot. Or HIV is that it mutates a lot. And it's very, very difficult to detect as a result of that. So there's 4 ways in which HIV is transmitted. It's through semen, vaginal fluids, breast milk. So that means it's likely that a mother could give it to the baby. And blood transfusions. So if one person's blood mixes with the next person's blood, some infected white blood cells could get transported across. This would be like if you're sharing intravenous needles, or if you have a blood bank that get contaminated. It has happened. You could get HIV from that or any other bloodborne diseases. So how can your spread be prevented or reduced? This is common sense. Use condoms. You get regularly tested. As Marshall say, Marshall, just get tested. Education. You have a lot of people who think having sex with virgins is this cure AIDS. Of course, that is not true. And you have anti-HIV drugs for pregnant women so that the babies don't get. It basically blocks the HIV virus from crossing the placenta. You do have drugs such as AZT that is minimize the impact of AIDS. So like if you're real rich, you could buy a whole set of AZT and you could live for a long time if you have HIV. So like Magic Johnson or Charlie Sheen. So yeah. Now there's a thing I'll tell you. It have nobody in the world who ever died of AIDS. So that might sound weird. I say nobody in the world ever died of AIDS. Nobody's died of AIDS. AIDS is a state where your white blood cell count is so low, your T lymphocyte cell count is so low that if you get a flu, that flu could turn into pneumonia and kill you. So nobody ever died of AIDS, but people die of complications due to AIDS. So people could die of opportunistic infections. So if you have full-blown AIDS and you get the flu, well, chances are you're never going to recover from that flu. But you're not going to die of AIDS, you're going to die of the flu. So yeah. Alright. Alright, that's now, Let me just make sure that Okay. I don't know if I'm trying to remember if I don't think I sort of learned that much about drug impact and things like that. But it's kind of like COVID, you know. You don't really die of COVID, it's die of complications due to COVID. Hey, speaking of that, I wonder how much COVID that's it at right now. Let me check. Somebody give me the numbers now. Give me the numbers like it's playway numbers now. Give me the numbers there. What's the COVID numbers? I check the numbers like it's playway now. What it is? Alright, I see it. There's 188. 188 and 7 deaths. 2 deaths. Not bad. Time to open back doubles. Okay. Alright. Next thing we want to look at is going a little more into diabetes. So diabetes, we have to know how exactly what is the difference between diabetes type 1 and type 2. Is 188 cases, 2 deaths. Alright. I'm going to use an analogy here. To explain diabetes. YouTube going to take it on though, so you could bet your life on it. Alright, Olia. Let me get a little thing to play there in the meantime. Alright, that's now it. Alright. I putting up music so you'll get taken down. So if you had to join in, join in. Alright. I want you to imagine this. I want you to imagine your body is a hotel. That's a very poor hotel. I want you to imagine your body is a hotel. If your body is a hotel, right? And that hotel is actually filled with billions or trillions of rooms. What is one room in a hotel? Alright, so if your body is a hotel, what would be one room in the hotel? And now you have guests coming into the hotel. Right, so each room is a cell. Right, so you imagine like each room is a cell, right? Now you have the entrance, and I want you to think of the entrance as like, this is like the alimentary canal. That's like the mouth, the esophagus, the stomach, everything, right? Now, there's front desk at the hotel. So this is inside the hotel now, right? And we have a bunch of people coming into the hotel. Right? So you have a bunch of people coming into the hotel. These are the guests. So the guest is going to be the food. Right? So the guest is going to be the glucose. So there's the glucose. So the people at the front desk now, there's the clerks. So what we have, the hotel is the body. The rooms are cells. We have the front desk. So the front desk is actually going to be the pancreas. And here we have the clerks. So the clerks, right? So the clerks going to be pancreatic cells. So you know when you go in a hotel, you have to check in. And when you check in now, the clerks, you have to go and tell the clerk, give the clerks the information and things like that. So we have the glucose here, right? We have a line of glucose waiting to come in. Right? So you have a bunch of glucose waiting to come in. Where you getting this glucose from? Where you getting it from? Like your bread, you getting it from when starch breaks down now, you getting it from fruits, everything. So where you need to enter the room. So the clerk, after you're done signing, the clerk going to give you something so you could go in your room. What the clerk going to give you? What do you think the clerk going to give you? The clerk have to give you a key. So you need the key in order to get into the room. Right? So you need the key to get into the room. And so that key, in this case here, is going to be a hormone that is called insulin. And so the pancreas is going to give out insulin, right? So you're going to have a key that you're going to need to get into the room. And the key is insulin. So let me break it down here. The glucose requires insulin, right, to get into the cells. So what the insulin does, insulin converts glucose into glycogen. Glycogen is a stored form of glucose in animals. Glycogen is basically like animal starch. Okay, so we go it again. Think, we have a hotel. The hotel is the body. The hotel has rooms, many rooms. Each room is a cell. We have guests constantly coming into the hotel. Basically, like you're always eating food. Some of those guests are glucose. They're going to go up to the front desk. The front desk is the pancreas. Because now this glucose can't just remain in the bloodstream. It has to go from the bloodstream into the cells. So the pancreatic cells are going to give it a key. They're going to give each glucose molecule a key. And that glucose molecule is going to act to open the cell door, basically to allow the glucose to go through the cell membrane and get into the cell. So it can be stored as something called glycogen. That is basically what is happening. Now, diabetes is a failure of this process occurring. Is a failure of one of two things. Now, when we come back here, diabetes type 1 is where there are not enough keys to be given out, but there are many guests coming in. So basically what this means is that you have high glucose levels, low insulin levels, right? So what do you need in order to solve this problem? Well, we need to call somebody we need to get more keys into the body. But how are we going to do that? Well, that's easy. We just take an insulin shot. So if you take an insulin shot, we get in the keys. So diabetes type 1, we don't have enough keys. We have plenty guests coming in, but we don't have enough keys. We have the same number of rooms, but we don't have enough keys. So that there's a problem. So it's either that or the pancreas is working way too slow to give out the keys. So it's kind of like you have a Trinidadian running the hotel. You know, they're always on the phone. They're always doing something else. They come to work. They're never there. That's kind of like diabetes type 1. Diabetes type 1 is the pancreas is working too slow, or there's simply not enough insulin. So there's a next way we could solve this, and it's easy. All we need to do is limit how much gas coming in. So it's kind of like, you know, when people line up to go for the pension, or they line up to go in the bank, and when Terrence say come out for your vaccine, or everybody just walk in and a whole set of people come in, I didn't have enough people, you didn't have enough vaccines. That's kind of like diabetes type 1. Terrence say, hey, everybody come for the vaccine, but only have 50 vaccines per health center. What do you feel going to happen? So what's something that's more sensible? Well, if you have 50 vaccines, tell 50 people to come. So if you have diabetes type 1, do eat plenty sugar. If you have diabetes type 1, cut down on the fats, cut down on the glucose. So that's diabetes type 1. Diabetes type 2 is this. Diabetes type 2 is there are enough keys, but they don't work. So they don't work to open the doors. So basically what this means is that the cells are resistant to insulin, and exercise can help the cells be less resistant. So the cells resistant to insulin, and the cells become less resistant as a like if you exercise or if you just manage a diet. So diabetes type 1, you have high glucose levels because they are low insulin levels. Diabetes type 2, it don't make sense taking a shot because it's kind of like the key, you have enough keys, but the keys not working. The insulin not working. So the cells are not responding to the insulin. That is diabetes type 2. So diabetes type 2 is where you get later on in life. So I'm going to draw a little thing there. Hypertension, okay. So imagine this. There's a bloodstream, right? There's a capillary. And these are cells. Alright, so these are body cells. Yeah, so you basically had to, your key card is working, so you have to like swipe it number of times. It's kind of like that's like diabetes type 2. We need to get the glucose into the body cells and convert the glucose to glycogen for storage. So what's going to happen here is that these glucose molecules have to eventually go into here. Now, what happens if the glucose cannot get into the body cells? Well, the blood glucose level would remain high. If the glucose ends up going into the body cells, the blood glucose level drops. That is what you want to happen. You want the glucose level to drop. Because what happens there, if the glucose ends up staying in your bloodstream, right, what happens is that you could have a lot of bacterial growth that happens in the tissues. Because bacteria love sugar. They love to feed on sugar. And if respiration is not happening in the cells die. And then bacteria would invade those cells, right? Those cells that have necrotized, that have died. And you end up getting something called gangrene. So, or you get very little sensation in those areas. So this is why people who have diabetes, they tend to feel numb in their feet. And then if the foot get infected, right? So basically this is gangrene. Right, so it's dead, infected tissue. And what would happen is that this would have to be amputated to avoid further decomposition. So this is what happens when the glucose remains in the bloodstream and it doesn't go into the cells. But anybody could tell me how, if you have excess glucose in your bloodstream, how you eventually get rid of it? Tell me how you eventually get rid of the extra glucose in the bloodstream. Yeah, eventually is excrete the glucose. So is urinate it out, is come out, is pass through your kidneys, through into your bladder, and come out through the urine. So this is why one of the symptoms for diabetes is having glucose in urine. Fun fact, now you could actually use a Benedict solution test to test how much reducing sugar, basically how much glucose there is in the urine. And but long time they start to actually take a little sip of it, to see if it tastes sweet. But I mean now you have Benedict's reagent that is turning orange, red, everything there. So the person who said dialysis there, so dialysis would be used to regulate it here. So eventually a person might have to use dialysis if they have kidney failure because of it. Okay. Let me go into immunity and immunization. yeah, just Jessica, well, as they say, we on diseases. Alright, so let me before I go into that here, let me go through glucagon one time. Alright, so I'm going to show you a typical graph here that involving diabetes. Let me see if I can find. Okay. Okay. So let me look here. Let me trace what's happening here, right? So let me look for a normal person. You wake up. When you wake up, your active blood sugar level is at its lowest. This is why people, when they take in blood sugar test, they take it right after they wake up. So you wake up and you eat breakfast. That sugar that you had in your breakfast, maybe you eat some frosted flakes, maybe you eat some leftover, maybe you eat a doubles, maybe you eat some leftover fries from your KFC. I have no idea. So you eat and that's going to get assimilated into your cells, right? So it's going to get absorbed into your bloodstream. This is why your blood sugar level is going to go up after you eat. Maybe eat some pancakes with syrup, maybe eat some fry bake, but whatever sugars that it had in there, whatever starches that get broken down and gets put into your bloodstream, so your blood sugar level is going to go up. Now at this point here, right, where I have this label A, right, where I have A, this is where insulin is secreted to allow glucose to enter cells as glycogen. So what happens is that your blood sugar level will drop. Your blood sugar level is going to drop now. You're going to eat lunch and it's going to go back up, and you're going to secrete insulin and it's going to go back down. This is your typical thing. A person who has diabetes, right, so a person has diabetes, what's going to happen is that their blood sugar level is going to go higher. And it's going to take a longer time to decrease back to normal. So basically a person who has diabetes, right, so a diabetic, either does not have enough insulin, or the insulin or the cells are resistant to the insulin. So as a result now, blood glucose levels are higher. Okay, I don't charge anything because I was not a teacher. Right, so here we go. This is what it would look like. So the blood glucose levels would be higher. But there's another hormone that is secreted by the pancreas. So if you look here, let's see if I can get this to fit. Right. So what happens if so your blood glucose level is too high? Your pancreas is going to secrete insulin. Insulin is going to make it drop and return to normal. That's where we just went. But what if your blood glucose level is too low? Basically, if you have hypoglycemia. Well, a way to think of this, right? I don't know if we have anybody here who ever fast for anything, whether it's Shivatri or whether it's Lent or whether it's Ramadan or whether it's just feeling to eat nothing or whether it's just you're out whole day and you get to eat nothing because all the restaurants close. I want to think about the last time you didn't eat nothing for the whole day. And think about the last time you eat was probably 6 o'clock, 7 o'clock in the morning. They will say it's fasting. 1 o'clock, 12 o'clock reach you're real hungry, right? You're real hungry, but it had no food. It had no food around. You're real hungry, but it had no food. Now, I want you to think of this now. You probably didn't feel so hungry for like hours after that. The next time you eat is probably in the night. So, I feel streamer a little behind with the stream. Alright. So, what happened here is that you have very, very low blood glucose level because the last time you eat was hours ago. So, all of a sudden, you're not feeling so hungry again, even though you're feeling real hungry, like at lunchtime. 2 o'clock, 3 o'clock, reach you're feeling normal. Well, not normal, but you're feeling so hungry again. Well, what happened is that this next hormone called glucagon get released. So what does glucagon do? Glucagon turns stored glycogen from the liver and converts it to glucose. Glucose is then put back into the bloodstream and cells absorb it and use it for respiration. Okay, so that is what glucagon does. So whereas insulin converts glucose to glycogen. Glucagon does the opposite. Both of them are secreted by the pancreas. Glucagon converts glycogen to glucose. So you could think of the liver as kind of like, imagine your body had a little fridge. It could go into, you're feeling hungry, let me go in the fridge and get some food now. That's like your liver. So, but the fridge locked down now. The fridge have a lock on it. So glucagon is kind of like the key to open the fridge. I don't know who does put a padlock on a fridge, but imagine you have a padlock on your fridge. So glucagon is like the key to open your fridge so you can get some food when you're feeling hungry. So basically what it means is this. Imagine you didn't eat all day, you don't want to fall on and dead by the end of the day. You have stored stuff, you have stored sugar in your liver. Just open up the liver and get some, put it back in the bloodstream. That's what glucagon does. Hopefully that makes sense. Justin, I just going through notes for now. I'll probably go through past paper questions a little later. This is just notes and theory for now. Alright. What we're going to do now is immunity and immunization. And then we could take a little break. We could take a little hour break after that. Alright, I'm gonna put it in the chat. You don't study when I doing past papers now. You don't study that. It will come when it come. You just come on people's stream and ask them, what are you doing for a whole breakdown of the timetable there? You just study that, huh? Again, free thing, but let me tell you, free thing not good and good thing not free, boy. You should have paid for lessons. There's the free bootleg lessons again here. You don't study that. I don't even know why I don't even know how to use a computer. You're asking me when I even pass papers? What's that? Alright. We're gonna do it when we do it. You're real time. There's a last minute thing, there's last bag ass kind of thing. Only should have pay for a actual teacher to teach all you. only studying to ask for thing. This free thing real trash, bootleg, last minute kind of revision here. Only making demands, yes? Wait. There's a desperation kind of thing not happening here. So don't study it, just sit back and enjoy. Okay, let me go through immunity and immunization there. Alright, take out all this nonsense. Alright, okay. So let me break down immunity into some categories here, right? But first let me define immunity. I don't feel I have to go through. I think somebody's asking about irritability and thing. I don't really think I have to go through that because I don't think that's one of the broad topics. Free thing not good, good thing not free, but they giving out. I bet you if you can go and pay for vaccines, you can see how well run that thing would be. If you can go and pay for it, it can be well organized, but because the thing free is a free for all for everybody. Alright, where are we? Alright, going to immunity. Okay. Alright, so first, let me come back here first. So what's immunity? Immunity is the organism ability to resist infection, right? Immunity is an organism's ability to resist infection. So some people who just get sick real fast, you know, a raindrop falling and get sick, they have a low immunity. People who go and walk in a crowd and don't get COVID, they have a high immunity. So what exactly happens when you get sick? So when you get sick, your body stimulates the production of white blood cells to basically fight off to fight off whatever pathogen that gets into you. So the problem with using COVID as an example, right? The problem with COVID is that COVID overstimulates your immune system. So you overproduce white blood cells to the point that you start secreting a whole sort of mucus, you start getting massive amounts of inflammation, and then you're dead. Lungs start filling with fluid and things like that. So we could break down immunity into these categories here. We could break it down into being natural or artificial. So what do I mean by natural or artificial? So by natural this means that there's no, imagine this, there's no external factor, there's no external factor or external chemical or drug that is involved in the process, right? Nothing external to the human body, so to speak. Artificial means that there's an external chemical or drug involved in the process. So now we could break this down into each one. We could break it down into passive and active. So we have natural passive and we have naturally acquired active. Same thing here, we have artificial passive and we have artificially acquired active. Those who came in a little late, by the way, all my notes you could find on the video description, right? Alright, so natural passive is basically this. You get sick, unaided, you get stronger. Actually, no. That's the wrong thing. That'll be this. Natural passive is this. Your mother antibodies make you stronger. Artificial passive is this. You get a little dose of a serum to cure a little infection, right? And the key word here is cure. Cure or treat. Artificially acquired is this. You get a little dose of a pathogen to prevent the infection. So there's your Sinopharm, your Pfizer, etc. Basically a vaccine. Okay. So I'll give the proper explanations right now, but let me understand what the difference is, right? Natural passive is this. Natural passive is what you're born with. So you're born with antibodies that have been given to you by your mother, and you just get it through either the placenta, like you get through the blood transfer through the placenta, or you get through the breast milk. Naturally acquired active is where, I'll give you an example. Chickenpox. I don't know how much all you have had chickenpox, but chickenpox is caused by a virus. And if you get that virus, like if you get infected with chickenpox, you develop immunity to it. So, which means you're very unlikely to get reinfected with chickenpox. So, what happens there is that but chickenpox could mutate into shingles, but it reduces your chances of getting reinfected. In fact, most strains of the common flu work that way. So what happens is that if you get a flu in January and it's the same strain of the flu, it's very unlikely for you to get reinfected in February unless your immunity really going through. Okay. Artificial passive is this. So when we say artificial is where whatever you're getting doesn't come from your own body. It comes instead from some external source. It is done through a clinic procedure. It is done through a physician. There's the introduction of something else into the body. So artificial passive, right, is where I want to imagine this with artificial passive. Now, assuming nobody here ever gets tetanus. Tetanus is like when you step on a rusty nail and you get infected with the pathogen on the rusty nail. Now, imagine tetanus as this. Try to flex all your muscles at once and imagine you can't unflex it. It's very painful. So, it's kind of like antibiotics, but it is there to it is an anti-serum or serum. So artificial passive is like this. Imagine like you never get a tetanus vaccine, and you step on a nail, and you be like, oh God, but I feel like again tetanus. I feel like again, I don't want to get tetanus because I don't want all my muscles to seize up. I don't want to get lockjaw, right? So you go to the clinic, and they're gonna give you a shot of something called an anti-serum. And the anti-serum is going to clear out, it's going to kill, kind of like antibiotics, it's going to kill all of the pathogens in the blood. It is only a short-term cure though, because it can't prevent you from getting tetanus the next time. Because if you go out there now, and you end up stepping on a next nail, that anti-serum done, the white blood cells and thing done dead and thing from fighting the thing. So that's artificial passive. It's a cure, but it is not a prevention method. Artificially acquired active is a vaccine. So this one is your vaccine. This is your Sinopharm and your Moderna and your AstraZeneca and all kind of thing. There's your MMR, rubella shots and all kind of thing that you get in primary school. So this works in very different ways. A lot of the vaccines that we know are called live attenuated vaccines, which you basically introduce a dead version or deactivated version of a virus into the bloodstream. What happens is that you'll have an immune reaction to it, and when you have an immune reaction to it, that's basically your body training its white blood cells to fight for the real thing when it comes. So let me use Rowley as an example, right? So Rowley get COVID, and he don't wear a mask during the press conferences because I assume he think he have natural acquired active immunity. He would have get natural passive immunity from his mother 70 years ago, right? And that would have keep him from dying like when he was like 1 year old or something. That's just the first set of immune system. So let me say, he get COVID, so his body already recognize what COVID is. It wouldn't be able to recognize the variants and things, but it recognize the spike proteins and the actual thing of COVID. So what happened there is that if a next COVID virus were to enter his body, it is likely that the body say, hey, we know who's you already, we see you already, so we could fight you off. So since COVID is a virus, there's no antibacterium, there's no antibiotic for it. So what happens is that artificial passive immunity can't really work against it. So you get COVID. I mean, it have drugs that could help treat it, and that'll probably be the artificial passive. You get COVID, and they say, look, this drug keep your lungs from disintegrating or whatever. That would be like artificial passive. Is a treatment. It's like, well, I never get the vaccine, and you just want a little flush out scene now. So that's artificial passive. Artificial active, acquired active is this. So now Rowley say he getting the vaccine next week, right? So which one he getting, I don't know, but he say he getting it. So what happened now is that what they going to, let me assume it's Sinopharm. So what happened there is that what they're going to inject him with is basically they're going to give him COVID, but a deactivated version of it, or they're going to give him like a little spike protein from the COVID. And what's going to happen there is that one day what's going to happen is that the body is going to recognize that even though it's like a dead version, it's kind of like if I imagine it's like this. Imagine like it have like a wolf. Now wolf was like a real dread animal, but imagine it have like let me say a wolf. Imagine it have a wolf, a plague of wolves. Oh no, let me say rats. I hate rats. So imagine have a plague of rats. And I never see a rat. All I know is that a rat is something that would kill me. But imagine now you bring a dead rat for me and be like, that's a rat. Well, at least I would know it was a rat. So that's what a vaccine is. It's showing your body the corpse of a virus, or a very, very weak, sick in the hospital, in ICU version of the virus. Basically, it giving you COVID, but a very, very weak version of it, or a dead version of it. Other vaccines like Pfizer don't work like that. What Pfizer is actually giving you is a little piece of RNA, kind of like DNA, and it actually building piece of the virus inside you. So it's not actually giving you the virus, it's giving you instructions on how to build the virus, and as a result you would know how to build the cure against it, how to develop white blood cells. So that there's the four of them. Now let me look at the actual definitions. I know I use like real bootleg definitions there, but that's just to help you understand. So let's look at the actual definitions. Now, as I say, usually, like if they say diseases is a topic, the usual thing is to bring immunization. That's a big part of this topic. Alright, so immunity and immunization. The key thing here is to produce these cells here that are called memory cells. Memory cells is basically what would remember the disease, remember the metabolic reactions of the disease. So it's kind of like this. It's kind of like I remember you are gonna fight you off now. So was the stupid Game Boy like Among Us? I know who's the imposter, so I ain't giving you any chance to do anything. You're gonna vote you out right now. So that is what when you're immunized against something, you have developed those memory cells, those specialized cells called memory cells. You don't get that during passive immunity. You only get them through long-term immunity. So let me look at the actual definitions, not, if you get sick and you're dead kind of thing, right? So natural passive occurs when a child prenatally receives antibodies from its mother. So basically while the child is a fetus, or through breast milk postnatally, this is going to form the beginning of the immune system. This is what's going to prevent you from getting sick, from being sick and dead when it's 2 days old. That is natural passive, and if you really want to think about it, it's natural because is developed in your body. It's not due to anything external from a human cell. So it's not like it's an anti-serum or a vaccine. Is human transferred, and passive means that you don't expend any energy to acquire the immunity. So you can think of passive as it is passed on to you. You don't have to spend any energy to actually acquire the immunity. Naturally acquired active is basically occurs when a person is exposed to a pathogen which stimulates the production of antibodies. It could prevent a future infection. So let me say, for example, you get sick, you will start to produce a massive, your white blood cells undergo mitosis, calling the whole army. They will make the whole army and they will fight it off. So that is actually the high metabolic reaction is what's giving the fever. Artificial passive is this. So when an anti-serum containing antibodies is administered to the bloodstream or the body to treat an infection. So again, this is like you don't have a tetanus vaccine, but you step on a nail. I'd be like, oh God, I don't want to get tetanus. You take an anti-serum for it, and it would get rid of the viruses or the pathogens or whatever. It is just a short-term thing. It's basically like an antibiotic. It don't prevent it long-term, but it prevents you from getting sick right now. And the last one is artificially acquired active, which is, so you have a vaccine, and that vaccine would contain a weakened or deactivated version of a pathogen. So this stimulates the production of antibodies and memory cells to prevent future infection. So now you would also have to know some advantages or disadvantages of it, right? So artificial passive, the advantage is this: it can actually treat an infection. So it could treat it, but the disadvantage is that it can't be used as a preventative measure. It don't make sense to say, hey, I don't want to get COVID, I don't want to get tetanus, inject me with this anti-serum. No, that don't make sense because I get it yet. It also don't make sense to imagine this, right? You get COVID, and now you're going for the vaccine. That makes no sense because a vaccine is not a cure. A vaccine is there to prevent it in the first place. So, let me say when Ian Allen get COVID, right? It wouldn't make sense for he to go and say, I want a COVID vaccine right now. I mean, people like he might actually do that. But a vaccine is a preventative measure, it's a long-term preventative measure, not a cure. So if you get COVID, you can't go and get a vaccine. This is why when Keith Rowley get COVID, he couldn't go and take the vaccine. It had a lot of people who didn't understand that. Like when Rowley taking the vaccine, everybody else take the vaccine, but the man had COVID. It's right, he can't take it because it's not a cure, right? So as little as I would defend politicians and things, I mean, that just makes sense. That's following the science. So a vaccine is long-term. It stimulates memory cells, but the downside is you can't use it as a treatment. You can't use it to treat the disease because it's there for your body to recognize the disease. That's what a vaccine is. So artificially acquired active. Okay. So that would wrap up immunity and immunization. And it would mostly wrap up diseases and things. Yeah, Among Us going through. That was like good for like 2 days. Okay, so we're gonna take an hour break here, right? When we come back, we're going to do transport, excretion, reproduction, and continuity. And in this bootleg lessons that we're given here, if we write some past papers into some of the more difficult towards the end. So we more than halfway through here. Alright. Well, I'm gonna talk about hypertension when I'm doing transport, because transport is the heart and thing. So all them thing atherosclerosis and thing I'll talk about that. Okay, so let me take an hour break and then we'll come back at 6:10 p.m. right? So you said this day. So I'm gonna leave the stream running. I don't know if you have anybody outside of Trinidad here, but talking about Eastern time, right? Because the last time we had a host of the Jamaicans and thing coming, and all in all, our difference. So let me put EST there just in case. If all your Bajans get out of here, Bajans invent CXE. All right. Good evening, everybody. We'll start back in a minute or two. So we're here for round 2 of this thing, and the topics we're going to do is transport, excretion, reproduction, and continuity and variation. So let me start with transport. Alright, give me a little second there, right? Alright, let me know if everything's showing there. Let me ease up the volume a little bit. Alright, let me know if everything's showing there. You're supposed to see like a pulmonary systemic circulation thing. Nice. Well, let me look at a basic thing here first, right? I'm gonna switch this to all right. Let me look at something here. Actually, let me see if I could get a non-markup version of that. All right. Oh. Eh? Right. Don't worry about reading the labels, right? Just follow me. Alright, I think everything looking alright there. Okay. So first, let me understand the human circulatory system, but before we do that, let me understand the need for a circulatory system. If we have something like an amoeba, a single cell organism with a nucleus, a few mitochondria, this doesn't need a circulatory system. Instead, what they could do is that they can simply absorb all their oxygen and do their gaseous exchange. They could just use diffusion, simple diffusion, in order to get materials through. And if it's things like water, they could just use osmosis. So if it's like a protist, like an amoeba, it doesn't need a circulatory system because, and I'll tell you why. So this is because single-cell organisms, right, have a high surface area to volume ratio. So basically what that means is that their volume compared to their surface area is or their surface area compared to their volume is very high. So they have a lot more surface area, so to speak, than compared to their volume. But if we look at a human now, a human surface area is really just what on the outside is what you can see, is what exposed to the environment. So we have a lot more going on inside than we have outside, right? Well, physically and metaphorically. But talking about the physical side now, if you think about it, if you could unravel all of your alveoli in your lungs, it would occupy the space of a tennis court. If you could unravel all of your neurons, it would basically span the equator, the earth, multiple times. If you could unravel all of the non-neurons, nephrons, right, like in the kidneys, it would span the equator, the earth, multiple times. Neurons is an even bigger deal because those would stretch on for miles and miles. If you just unravel them, we have a lot of stuff going on inside. So humans now, and well, we should say mammals, mammals have a low surface area to volume ratio. So this is why we need a circulatory system. We have a lot more going on inside than, let me say, a protist. Now, we're gonna focus first on the human circulatory system, the mammalian one, and then we're gonna look at the plant one. Alright, so let me take a peep here. The first thing you're gonna see here is the heart. So the heart now, if we take a better look at it, and let me add a little. Let me take a look at the heart here. So if you look at this here, you could see that the heart is made up of 4 chambers. Yeah, so the heart has 2 upper chambers and 2 lower chambers. Now you have to remember that the heart is facing you. So when you write in the direction, when you're labeling it, hopefully you don't have to draw it. That'd be real sickness if you had to draw it. But if you're labeling it, it's not that bad. Basically, if you have to label it, there's 4 chambers that you have to learn, and there's 4 main blood vessels that you have to learn. Now, in terms of the valves, you don't have to really know the names of the individual valves. That really only comes up in multiple choice. So let me take a look at the names of the chambers. Now, it's very important, as I say, to remember that the heart is facing you. This is why left and right is flipped. So you don't want to make a mistake like this case I did hear about in the States, not Trinidad, or it feel like it could happen in Trinidad, where a man had testicular cancer in his left testicle, right? And the doctor do the x-ray and the examination and thing, and when he went to put it up, he forget that he's supposed to flip left and right. He forget that the thing was facing him. So he had the cancer in the left testicle. The man ended up taking out the right testicle instead. And after that, he realized that he's supposed to take out the left one. So he had to take out the left one after and try to reattach the right one. Well, in the end, the man sued and got a whole set of money for something that, biology students supposed to not be able to chain up. But I say it could be high up and still chain it up. So you remember to flip it because you have to remember it facing you. So here we can see the 2 upper chambers are called the right atrium and the left atrium. And atrium is actually a room that is like a chamber that is high up in a building. So it actually, think of it like the attic of a building. So you have the right atrium, the left atrium, and the lower chambers, basically ventricles, which comes from the word vent. It's kind of like the chambers venting the blood downwards. So we have the right and the left ventricle. Out of these 4 chambers here, by the way, which chamber has the thickest muscle? Put it in the chat. Which chamber has the thickest muscle? So there we have the heart. So we have the right atrium and the left atrium, the right ventricle and the left ventricle. Yeah, you probably have to, well, chances are you wouldn't have to draw it, but the arcs already. So I would recommend that you practice it tonight or something. Just, but usually there's asks for a flow chart of it, which I'll show you. So there are 4 chambers here. There are 4, blood vessels here that we have to take note of. Now this is an easy way to remember this, right? Alright, so Faith is correct there. The left ventricle has the most muscle. Because as you notice here, the left ventricle is the one actually pumping blood to the aorta. The aorta is the one that carrying blood away from the heart and towards the body. So the left ventricle is the one that has the most muscle. Okay, now let me tell you how to remember what is a vein and what is an artery, right? How to remember what's a vein and what's an artery. Alright, so arteries always carry blood away from the heart. Right? So arteries always carry blood away from the heart, right? Think of A far away. And veins always carry blood towards or into the heart. So remember this. Vein has in, so they carry blood into the heart. Right? So for example now, the pulmonary artery carries blood away from the heart and into the lungs or towards the lungs. Easy way to remember it. Arteries always carry blood away. Veins always carry blood in or towards. Vein in, artery away. Vein in, artery away. Easy. So any type of artery will carry blood away from the heart. So for example now, the hepatic artery carries blood away from the heart, but that's going away from the heart and towards the liver. The pulmonary artery is carrying blood away from the heart and towards the lungs. The mesenteric artery is carrying blood away from the heart and towards the intestines. The jugular artery is carrying blood away from the heart and towards the cranial area. So you have plenty different arteries. You really only one you have to concentrate on is the pulmonary. So let me take a look at the notes here, right? When it comes to the heart. And let me draw a little schematic here for you. So if they ask you to draw a little flow chart for the heart, this is how we're going to do this, right? So watch me. So you're going to draw a little box, divide it into 4, and the 4 here going to be the 4 here, this is going to be the 4 chambers. So this here going to be the right atrium and the right ventricle, and this here going to be the left atrium and the left ventricle. Yeah. So let me start with blood leaving the heart. So when blood leaves the heart to go to the body, it's gonna go out the left ventricle. And I'm calling it red because it's oxygenated. So this is going to be the aorta. The name of this is going to be the aorta. And this is going to take blood to the body cells. So this is going to be to the intestines, it's going to be to the liver, it's going to be to the legs, it's going to be to the brain, wherever, right? So this is now going to re-enter as deoxygenated blood. Right? And it's going to come into the right atrium. Okay? So you're going to come into the right atrium, and the name of the blood vessel that is bringing it back in, remember, in is a vein, So the main vein that we have is the vena cava. So you actually have two vena cavae. They are the top one that comes from the upper body, that's called the superior vena cava, and you have the lower one, which is called the inferior vena cava. Okay, so this, you see this type of circulation here, this is called systemic circulation. And basically this is between the heart and the body. So it's a double circulatory system, because after this blood comes back into the heart, right? The blood now has to leave the heart again, because it has to regain oxygen. It has this is deoxygenated. So this blood now is going to get pushed down to the right ventricle, and this blood now is going to go to the lungs, the alveoli and thing now. You're going to get reoxygenated, and you're going to come back through the left atrium and then go to the left ventricle. So you don't necessarily have to draw a heart, you know, unless they say specifically draw a heart. But you don't necessarily have to draw a heart to show the blood flow. All you have to do is just draw a little flow chart to show where the blood is flowing. And it's not hard, honestly. You just need to visualize it. You need to visualize, and I'll pop back to a picture just now. So what y'all think is the name of this blood vessel here that I label X? What is the name of that one? And if you think about it, it is taking blood away from the heart and to the lungs. So drop that name for me in the chat. And then we have this blood vessel here, Y, that is taking blood into the heart from the lungs. So remember, away and in, away and in. And it's not hard at all. Well, which one X could be? Nah, so X is not the pulmonary vein, because remember, going away from the heart. Remember, going away, and when you're going away is an artery. Right, so this is going to be a pulmonary artery. So you're going away. So you have to look very, very carefully to see if it's going away. Now, remember something here that particular about the pulmonary artery is one of the few arteries, not the only one, but it's one of the few that actually carries deoxygenated blood. Arteries usually carry oxygenated blood, but pulmonary arteries carry deoxygenated blood. Veins usually carry deoxygenated blood, but the pulmonary vein carries oxygenated blood, because remember, coming back from the lungs and into the heart. Okay, so let me take a look at something here. Let me get a little picture of the heart there. Alright. So remember, arteries always go away, veins always go in. Okay, so here again, we have the left atrium, not the left atrium, sorry. We have the right atrium, we have the left atrium, we have the right ventricle, and we have the left ventricle. So let me trace the blood, what's happening here, right? So let me start from the left ventricle. So from the left ventricle, the blood is flowing away from the heart, and this here is the aorta. That's the aorta, and that's carrying blood away from the heart and towards the body. So this is going towards the body. So this is going to go all the way to the body, and then they're going to come back right. So this is coming from the body, well, never mind the diagram, but that's how it's supposed to be. Alright, so from the body, so this here is the vena cava, the superior on top, the inferior below. I got to differentiate here. So this is the vena cava. So the vena cava is bringing blood back from the body into the heart, into the vein, into, right? So this blood now going to flow down here, and then it's going to flow through here and here. So this here, remember, it's going away from the heart, it's going away. So it's going away from the heart. That would make this, right? That would make this a pulmonary artery. You have more than one pulmonary artery, by the way, as you see in there, right? So that would make this a pulmonary artery, because they're going to the lungs and going away from the heart. It's deoxygenated. So this is going to go all the way to the lungs, and this is called pulmonary circulation, going all the way to the lungs, and then this oxygenated blood is going to come right back through here, and then the process is going to repeat. So this blood vessel here, this one here, this one is called the pulmonary veins. Right. So I know this looking like mess here, but these are the 4 chambers you have to learn, are the 4 blood vessels. So as I said, just remember, arteries always go away, veins always go in or towards. Arteries always go away, veins always go in. It doesn't matter what type of artery, it doesn't matter what type of vein, arteries always go away and veins always go in. There's other differences, of course, but we'll get into that just now. Alright, so the last thing that we have to get into here now when labeling this is, I'll use a fresh diagram. These things here, so these things here, okay, so these things here, these are called, right, these are called valves or semilunar valves. So there's a name for each one, but you don't really have to know the names, but you should still know it, right? Because it's usually come in multiple choice. Okay, so this one here, this one here, this one is called the tricuspid valve. Right, so this one here is called the tricuspid, and the one on the opposite is called the bicuspid, or sometimes it's called the mitral valve. These two here, so this one here, this one is called the pulmonic valve because it relates to the pulmonary artery, and this one here, since it links up with the aorta, this is called the aortic valve. Right? So, but you don't really have to know those two, but you should know tricuspid and bicuspid. So I don't know if you could come up with a way to remember it. I just remember like the one with the R, like tricuspid, like R now, like it's on the right, but on the right side of the heart. That right side, tri, like right. I don't know. It's up to you. So, also remember this. If it ever come up, just say, oh Lord, I can't remember nothing in biology. So remember Lord, left oxygenated, right deoxygenated. Okay, so remember Lord, left oxygenated, right deoxygenated. Okay, so let me look at the notes here. But usually I say the worst they could do is ask you to draw it, but as a drawing it is sometimes it's common, but usually just ask you to describe it or describe the flow, or they might bring it like an oxygen molecule enters the, we'll do some past papers just now, you can see. Alright, so so somebody wanted to have a little explanation of hypertension. There's really not much to describe with hypertension, but you should know the factors that affect blood pressure. So the factors that affect blood pressure is like a fatty diet, excess sodium, and age. So all these things here could lead to atherosclerosis or arteriosclerosis. Arteriosclerosis is the hardening of the arteries. They become more stiff. Atherosclerosis is the clogging of the arteries and restriction of blood flow through those arteries. So if we had, let me say we had a blood vessel. Yeah, change my clothes. This room cool. So if I was to draw an artery and a vein, right? So we have here so there's an artery, and there's a vein. So there's really two things we have to look at. One is this. There's a thick muscular wall. Thick muscular and elastic wall. The second thing is this. There's a small lumen. The lumen is basically the passageway for the blood to pass. So this thing I color in gray here, this is the wall. Right, that's the muscular wall. So arteries have a much thicker one, no matter what type of artery. Veins on the other hand is kind of reversed. So a vein now has a very thin muscular wall. Very, very thin muscular and fibrous wall. And veins have a larger lumen. Now if all you remember anything from all the physics, all you know pressure is force over surface area. So because now, yes, Abiana, aortic and pulmonary valves are semilunar valves, that is correct. Because these have a larger area, right? They have a lower pressure. And because arteries have a smaller area, they have a higher pressure. So with arteries, you'd actually feel a pulse. So you'd actually feel a pulse through an artery, but you don't really feel a pulse in a vein. So there's like some differences that there. So what exactly is hypertension and what is coronary heart disease or atherosclerosis? Well, let me draw artery lengthways here, right? if I was to draw an artery, and this is like a side view through it, right? So imagine like here we have blood vessels, not blood vessels, red blood cells flowing, but there's a build-up. Alright, so there's a build-up of plaque. So this is a atherosclerotic plaque, or just a plaque. So imagine a plaque is like a traffic jam. No, not traffic jam. Imagine a plaque is like road work. Like when Ministry of Transport, Ministry of Works block up half the road to, or when WASA fix the road and now that to fix it. Right? So that's a plaque. But what has happened during a plaque is get traffic. Because all of a sudden now these red blood cells that was flowing real good here don't need that kind of squeeze and tips past now. Right? So what happens here is that you have a build up. You have a restricted flow of red blood cells. No, that P equals rho GH formula is correct. That could work as well. I won't get into that now, but yeah. Gravitational potential energy is mgh. Rho GH is pressure. Alright, so you have a restricted flow here. Well, not there, but over here. So here we have an even smaller area, right? So you have a smaller area over here. So you could end up having a much larger pressure over here, and that could even lead to hypertensive conditions. But this is the other thing that could happen here. These red blood cells, let me say one of them end up getting stick up here. They could end up forming a little clump over here. This is what you don't want to happen, because it basically is like a car accident. So you know, like if you have a truck that block up the whole pathway, you have an accident, and well, you know, you have even more traffic now. So this here, when this happens, right, this is called a clot, or more correctly, a thrombus. And this could lead to what is called a cardiac infarction, which is more commonly known to lead to a heart attack or lead to cardiac arrest. So this clot, which is called a thrombus, is a result of coronary artery disease and hypertension. So you would need a chemical to kind of clear out these things, or you would need to use a stent, which you don't need to learn about now. But a stent is like they just put a little wire inside here, and then they just have a balloon that it is inflate in this thing to clear out the plaque. You don't have to learn about that. Yes, could end up with a pulmonary embolism if the clot flows to the lungs. So yeah, you have a lot of things that can happen. But this is basically hypertension and coronary artery disease. Okay, let me look back at my notes here. But as I said, there's a number of factors that could affect this, which is lack of exercise, of course. Things like diabetes, which actually restricts blood flow and damages the nerves in the heart, the pacemaker in the heart. And the age, because when you get older, these arteries become less elastic, so they're less liable to be stretchy, so clots and things could form more easily. Just in thromboplastin, I'll get into that later on. A thrombus is a blood clot. A thrombus is basically what go kill you, yeah. And stroke is caused by a coronary thrombus, not coronary one, a cranial thrombus when you have one in the brain. Okay. Let me come back to the notes. Okay. So let me find a past paper here that deal with like blood clotting, but actually, all right. So I kind of afraid to show the clip here because I'm afraid the thing get taken down. But I had a little clip to show you like with blood clots. But let me see if I could find it on YouTube. I'll link it up with all you. If all you know this cell, or if all you know an anime called Cells at Work. Right, so I want to show the thing here because it copyright now. I want to take the chance. So it's like 3 minutes. So let me give all you a little 3 minutes to watch it. Alright, nice. So let me surely have asked people a question involving it, right? Just give me a little while, I'm gonna find it. Alright. Alright, if only watch here. Let me look at this here. There's Jan 2017, right? No, Cohen Spring on a whole different level from me. I not really planning to do this on a daily basis because I mean I have a life, but I only decided to do this for this year because this pandemic was so hard. I can't imagine being in all the, I mean, not joking, but I can't imagine being in all the shoes right now. I know when I was writing CXC, it was the worst. Um, and make I imagine that being multiplied and added to online classes and virtual labs and all kind of stupidness. So I'll say I have the expertise, I can share. All right, so let me look at this question here. Figure 1 shows components of blood. Let me actually hold on. Let me capture my window instead of the display. Alright, so A is platelets. So there's platelets, and platelets is what's going to help form the blood clots, right? Alright, so B is going to be a phagocyte, not phagocyte, a lymphocyte. So, I'm tell you how you know it's a lymphocyte. You have to look at the large nucleus. You have to look at the big nucleus that it have. A phagocyte is not going to be round like that. A phagocyte is going to be more irregular. Oh, no. Nah, I forgot to resize it. Sorry about that. And right, so you're looking at the nucleus. So a phagocyte looks more like this, right? So this could be a phagocyte. Right, and C could be a erythrocyte or a red blood cell. Right. The component for blood clotting, right? So that's going to be A, the platelet. Okay. Oh, shucks. The thing mess up. Just now. So if you look here, alright, so this question here is saying, alright, just I know this kind of thing up there. Right. So they say complete the flow diagram shown in figure 2 by filling in the missing words at positions D, E, and F. So if you look at it here, it's saying clotting factors released from D, plasma protein synthesized in liver and circulating in an active form. This is going to allow prothrombin circulating in the plasma to allow the formation of something, and that's going to activate fibrinogen into forming something else. See what it could put as D, E, and F. Yeah, sorry about that all here. The browser messed up there, so everything did freeze. Okay. So clotting factors are usually released by the liver, right, or actually damaged tissue. So E now, so any clotting factors are usually synthesized in the liver. Prothrombin converts into thrombin, into something called thrombin, and fibrinogen converts to fibrin, or fibrin. Now, let me explain exactly what happened here, right? So I want you to imagine now that you get a cut. So imagine here we have a blood vessel. Right, so here we have a blood vessel. And you get a little cut on that blood vessel, right? So you get a little boo-boo on it. So what has to happen here in this blood vessel? So what happens here is this. You'll end up with these things called clotting factors. And what the clotting factors are going to do is that, so these clotting factors are also called coagulation factors, right? And these clotting factors here so in the presence of these clotting factors, and in the little anime clip all you see there, that's what the platelets was carrying with them. So the platelets kind of get, you can think of it like they get activated by these clotting factors. So example of these clotting factors is thromboplastin and thrombokinase. So those would be those would be like coagulation factors. So if you ever hear about thromboplastin, that's what it is. So in the presence of that, it's like a metabolic pathway. It's kind of like, well, in the presence of that, you're gonna get an inactive thing called prothrombin is gonna convert to thrombin, right? So prothrombin is gonna convert to something called thrombin. Prothrombin literally means before thrombin. So in the presence of thrombin, and I say you don't have to know that much about it, right? But in the presence of thrombin, you're gonna get fibrinogen, which is soluble, is gonna get converted to fibrin, which is insoluble. So what exactly is fibrinogen and what is fibrin? The fibrin is like a net. You feeling the question? Let me go through it real easy. Let me go through it real simple, right? Much all the thing I bigger. All of this and the thing might name come. Right. So you get a cut. There's the cut. There's the wound. Right? That's the damage tissue. So you're gonna have here is you're gonna have clotting factors, platelets, and that's gonna convert prothrombin to thrombin. That's what we're doing. So what it's doing is that you have this thing called fibrinogen. Fibrinogen is just like, imagine like a liquidy form in the blood. They're in the blood already, but inactive. So the whole point of it is this. You want to form a little net in this cut, right? You want to form a little net here. Right? Where you want to form a little fibrin mesh. Right? So fibrin is only formed when thrombin is present. Think of thrombin like an enzyme. Right? Fibrin is insoluble and forms a net to trap the red blood cells. The red blood cells then form a scab. So this is why I told you to watch the little clip, so you can actually see it. I mean, it's anime, but you can still see it happening. So basically, yes, now, this here, if I let it watch the thing, that there, that's the fibrin. And that was converted from fibrinogen. These are the platelets. And this is the scab trapping red blood cells. I mean, other than that, it really, that's what it is. I'm gonna type out that thing there. Let me link it. Alright, I'm typing out something. right? So read what right there. Hopefully that simplifies it a little bit. If I simplify it, check now. I'm coming back to the thing there. Check it, eh? We have to pray. So you have to sacrifice some things so that it don't come tomorrow, right? Alright, so we have to sacrifice, let me see. Alright, so we have to sacrifice some things. We have to pray that the easy things come. Alright, so sacrifice. You have to make a blood sacrifice. Pray to the CSEC God and them. The last thing. Alright. So we had to pray some of these things do come. So all these things are the sacrifice for tomorrow. Hopefully it come. Somebody said it come Jan, so if it come Jan, it will come. It most likely it will come now. You can sacrifice KFC. And I tell her I not supporting KFC when they reopen. If them have millions of dollars and them paying the workers in aloo pie and warm soft drink, I not supporting that. I will go and buy fried chicken down the road or something. Alright, let me get my contract there. Okay, but I mean really and truly, the other stuff here that's in circulatory system, the main thing you understand is how the heart does work and how if you have to imagine the journey of a red blood cell coming from the heart back into the heart. So if we have to visualize it, right? So if you imagine now blood flow in the body, and we start with the left ventricle, right? It will flow to the aorta, then the body. Then from the body, it's gonna flow from the vena cava to the right atrium, to the right ventricle, and that's going to go to the lungs. Well, pulmonary artery, lungs, pulmonary vein, left atrium, left ventricle. Babies could do it. But just remember the basic thing. Heart, body, heart, lungs, heart. Then again, heart, body, heart, lungs, heart, right? Heart, body, heart, lungs, heart. So you could name the blood vessels once you remember that. It has to flow from the heart to the body, back to the heart, to the lungs, then to the heart again, then to the body, and so on. So the link okay, so let me go into the plants. Alright, Olia, put in the chat here for me. What is some differences between the human circulatory system and the plant circulatory system? And then let me see if we can name some, put some similarities. Now we talk about like the structure. Right, so plants will actually circulate. Okay, so humans have a pump with a heart. Right, plants have no pump. Okay, humans have arteries, veins, and capillaries. The plants have xylem and phloem. Right, humans have blood as transport medium. Plants' transport medium is usually water or water-based. Alright, so one other thing here is that the human bloodstream, well, it spans here, but the plant too, but it is how we could say it involves neurons, but the plant one involves things like capillary action and hormones. Well, not hormones, and what's the word? Concentration gradients. So we have some differences there. Now if you think about similarities, right? Similarities between the two systems is that, well, they both transport food and water, dissolve food and dissolve minerals and things like that. They both have veins. So they both have, they both venuated or vascularized, right? They both have veins. They both have to transport dissolved substances. Yeah, so you can talk about transpiration or pearl and sources and sinks and all kind of things. So all of that there are examples of things you could use. Alright, so let me explain here. So let me say here we have a plant. Alright, so I just drawing like a piece of a plant. Okay, and we're gonna have the roots. They both have veins, but the veins have different structures. They don't have a muscular lining or anything. So all of that there, use your common sense. If big enough you have sense, you could figure it out. Alright, so what we're gonna have here, and if we look at it on a larger level here, larger scale, right, is we're gonna have water vapor leaving through the stomata, right? And a lot of this is gonna get taken up through the roots, a lot of this water. So this is gonna be through osmosis and root pressure that the water is going to come up. So the water now is going to move up a xylem vessel. I'm going to go to the leaf. So this is going to be the xylem. But how exactly does this mechanism work? Well, this mechanism works because of this. If you are to imagine water molecules are actually bonded to each other. So let me actually draw it coming up here. So water molecules are bonded to each other because they are, give me any word in the chat there. What is called when molecules can bind to each other, we say that they are what? Or they experience which force? So when these water molecules leave, is because there's a pull, and they call it transpirational pull. There's a pull. There's a pull up through the roots that happens because of this. The water are polar here, and because they are polar, right, so they're cohesive. Very good. So they experience cohesion and adhesion, which means that they stick to each other, but they also could stick to other surfaces. And this here is kind of like, I want you to imagine like you're taking the end of the water, the long water chain, and you're pulling the end. So the more that you pull here, this is going to come up so. So it's kind of like you're pulling a chain. So what is actually going to pull this? Well, what pulling that is just water being lost through the stomata. And that's because the stomata opening, it could be due to, it's hot, it have plenty sunlight, it have a low humidity, anything, it have plenty wind, and the stomata opening and things, water molecules flowing out. So it's kind of like you're pulling the end of that, and more water molecules come up. What you're doing is that they're creating a water potential gradient, so that water could continuously flow through this. But it has something real particular about water molecules here, about the xylem. Because now it's not really expending any energy to do this, it don't involve ATP or anything like that. This process here is unidirectional, which means that the molecules only travel upwards, they can't travel downwards. Right, so they're only traveling upwards. So it's different with the phloem. So if I had to draw it now, the differences between the two. So imagine here we have the xylem. And there's a little chain of water molecules going up.
Turning on your home computer to read the day's newspaper. Well, it's not as far-fetched as it may seem. In fact, both local San Francisco papers are
Right.
investing a lot of money.
So this going upwards.
Just like that started. Science editor Steve Newman reports on what
So the xylem now,
the brand new system.
there's how you could describe it. So it's hollow, it's continuous, right? So that means that there's no divisions. It's comprised of dead tissue, right? That's called lignin. So it's lignified, and it's unidirectional flow, which means it only goes upwards. So the xylem transports water and anything dissolved in that water. The phloem, on the other hand, doesn't transport water, it transports food. So what draw the phloem? Alright, so what we have here is a bunch of sucrose molecules. So not starch, right? And not glucose. Glucose is too reactive. So what we have here is sucrose. And here we have sieve plates. Now you notice I'm missing something because this actually needs energy to work. You need ATP to actually send the molecules all over the place. So tell me what type of cells I need to put at the sides of this. But this could either move up or down. So I'm missing something here. Right, so I need to put companion cells. So companion cells are what going to basically give it the energy. Alright, so I'm gonna draw in some there. So you need to have some companion cells. The companion cells is what's going to give it ATP in order for it to undergo translocation. Translocation is like the movement of the sucrose. Now, let me look at the differences here. The differences are as such. Right. So is not continuous, right? So it's actually separated into elements. It's living tissue. And it is bidirectional, which means it's down and up. But one of the main differences here is that it transports food. And this here transports water and dissolved mineral salts. And as the differences between the xylem and the phloem, baby thing, real easy. Alright. All they going so far, all they holding up. I mean all the other hour break, but since 1:30 we going. You all feeling okay so far? Alright. We're gonna do some past papers just now. We'll finish up transport, and then what we're gonna do is excretion. But I feel we could tackle reproduction and continuity and think via past papers. Yeah, go and make a coffee now. I mean, the thing you could just scroll back 10 minutes. Alright. So after we finish transport here, we could take a 5 minutes and then do excretion. He's real confident. So you come here, that's false confidence though. That's like when you're drunk and feeling confident. It's good to come here and see the real thing, because you could feel bad when you reach tomorrow and you know. Adrian, 3 more. Excretion, reproduction, and continuity. But reproduction and continuity is I said as the whole last blasted section of the syllabus. So very, very, very broad. Okay. But I feel we go do those in past papers. Like, is only when we doing the past papers and we bounce up some of the more difficult things in it. We don't need to know homeostasis. Homeostasis not in the broad topics. Unless I'm wrong. Double check it and see. Well, homeostasis could be under excretion now. It have a real easy way to dodge this exam, you know. All I had to do, and bio was one of the easiest subjects though. I mean, when all I reach physics and chem and thing, it have a real easy way to dodge this, eh? All you had to do is take off your mask and cough like mad, and cough at the invigilator and cough in the whole room. Like, just spray all over the room. That's all you had to do. And then you would get to avoid the exam, and everybody in the room would get to avoid the exam. That's all you had to do. Just pull on your mask and real cough. Like, cough till you're hacking up. Okay. Alright, so there we go through here factors affecting transpiration, and then we'll take our 5 minutes. Okay, so osmoregulation and homeostasis could be like around where excretion is. So we'll run through there. Okay. So factors that affect transpiration. This don't show up so much, but sometimes it is asked you to label the cells and things, but since you don't have cells, anything, I don't think they ask you to label it. But we have 4 main factors that affect transpiration. Light, temperature, wind speed, and humidity. So light allows the stomata to open wider, because they need to let in more CO2 for photosynthesis now. So as a result, oxygen could exit and water vapor could also exit. Temperature. Well, temperature could have both effects, but usually a plant now, unless you're living in a real dry area, a plant would need to allow water to flow through it in order to cool down. Because water is a cooling mechanism for the plant. So the stomata would open to increase the rate of transpiration. The guard cells would fill with water and get turgid. Wind speed. So every time wind blows, so when if all you ever had to hang up clothes, usually is hang up clothes in weather that's sunny and windy. Because when it's windy, it could actually blow away the water vapor that evaporates and allow more to come out. So the more wind is the more water diffusion that would happen, more osmosis. And humidity, if there's a lot of saturation in the air, that reduces the amount of transpiration because there's a lot of water vapor in the air already. So if the air is already very humid, transpiration slows down. So to speak, everything there basically increases transpiration. So all of this here increases transpiration. Plenty light, more transpiration. High temperature, more transpiration. Plenty wind speed, more transpiration. But so all of this here actually raises it, but humidity lowers it. Well, we're doing physics too. I have a whole thing with physics prepared, so I can't remember what day I put it on. Let me actually check the date. Let me link all of this stream. We're taking a little 5 minutes, right? So we're taking a 5 minutes and then we'll go on to excretion. Let me find the thing for it. As I say, I don't really post anything on YouTube, but you could subscribe just to get the notifications. Don't expect a lot out of me, eh? Don't expect a lot of videos. As I say, I only doing this year. There's the physics one. So I'm doing a paper one too. Nah, you're out of luck there with Kim. Actually, I feel I'm linking already wrong thing, you know. Wait, ignore them thing. Ignore them messages. Ignore them thing. I link already. I tell you, I do not use a computer. I link already studio versions instead just now. Let me get the actual thing. Yeah, we're taking a little 3-minute break again. All right. So let me start back. Excretion, excretion and homeostasis. All right, that's now my computer kind of thing up here. Task manager, stop responding. Yes. All right, that's now me roll up in here. Wait, that's now Hey, all here. Okay, Ole, just making sure everything coming through. The only seeing the screen and hearing me, right? I'm put on the music there. Hey, that's how online learning is. What school is everybody from? While all this thing booting back up. Music coming on, don't worry. Alright. I just had to wait for some things to open here. So again, just give me a little thing. Pain. Well, hopefully this was a smooth experience so far. I real not accustomed to streaming. Alright, so I'm just waiting for some things there. People have 2 exams tomorrow, I see in Spanish, so they're a wicked to do at all, boy. 2 paper 2s in one day.
Like a spirit amazed in a wider paradise.
No, I'm not from Grandy, I'm from Central. I was waiting for it to open up. Alright, so I say I'll go through excretion, but like reproduction and them thing, it take a real time. So I gotta just do those in the past papers. Well, I don't know much Grandy people, to be honest. The ones I know, they're just regular. Alright. This Microsoft Word taking real long, taking longer than everything else. Alright, to come up. Okay. Okay. I think this is it. Alright, just gotta make sure everything's size good. Okay, good. Looking so. But I don't have a whole set of other things open the last time now. That's why I have. Okay. Okay, so we're paying play. Alright, so what I'm gonna do is I'm gonna draw here a kidney nephron. So I'm gonna draw a kidney nephron here, and we're gonna label it, and you're gonna say what happens at each point, right? So hopefully this will come because, in my opinion, is one of the harder things in the syllabus. So audio echoing. Let me know if it's still echoing. So, alright, okay. I think two captures come up at the same time. Alright. Nice. Yeah, no problem. I'll lower the music. Okay, so here we have a kidney nephron. Right? So here we have a kidney nephron. And the kidney nephron now is going to, it basically acts like this. Is one unit of the kidney, right? And what happens here is that it's kind of like how the lungs have alveoli, the small intestine have villi, the brain has neurons, it basically showing here one subunit of the kidney. And let me explain what's happening here. So what's happening here is this. We are going to have a number of things in the bloodstream. So we have a number of materials here in the bloodstream, right? And inside here, we're going to have water, glucose, amino acids, red blood cells, white blood cells, hormones, proteins, urea, and so on, right? So we have a bunch of stuff that's in the bloodstream. So in the bloodstream now, alright, so I know this sound. In the bloodstream, let me think about things that we need to get rid of. So we need to get rid of the following. We need to get rid of excess water, some mineral salts, and urea. So these three here, this is basically urine. So what's going to come out on this side here, this is going to be urine. So we have a number of things here that is going to take place. So this blood vessel here, this is called the afferent renal artery. Right? So remember the things that you want to get rid of. We want to get rid of urea, water, and salts. Not all the salts and not all the water, but the excess ones. These three is what we want to get rid of. These things we don't want to get rid of. We don't want to get rid of glucose. We don't want to get rid of amino acids. We don't want to get rid of anything else. Now these things here, these are way too large. So these are too large to enter the nephron. So they're not even going to go in. So what we have here is a tangle of capillaries that's called a glomerulus. The glomerulus, a gloma by the way, is a ball of yarn. So that's where the word glomerulus comes from. And this here is going to create a filtrate that is coming down the Bowman's capsule. So this is called the Bowman's capsule. So the name of this process here is called ultrafiltration, where it is using a high blood pressure to create a filtrate. So what you're going to have here, you're going to have stuff pushed down here. So your filtrate, right? So your filtrate is going to be urea, water, salts, amino acids, and glucose. We want to reabsorb glucose amino acids. And some water. Tristan, glomerulus. Glomerulus. The word glomerulus is Latin for small ball of yarn. A gloma is a ball of yarn. So glomerulus means small ball of yarn. And it basically, if you was to look at a glomerulus, it looks like a bunch of capillaries tangled up like a ball of yarn. That's why it's called that. So the Bowman's capsule is creating a filtrate. And this filtrate here, we want to reabsorb the following. So let me cut away from here a little bit and let me just put some labels here. This is called the proximal convoluted tubule. And literally what it means is proximal means front or close, convoluted means coil up, tubule is a small tube. So it literally means coil up small tube to the front. Coiled up small tube to the front. This here is where you're going to get this process called selective reabsorption. Basically, what you're going to do is you're going to take back the things that you need. You're going to take back the glucose and the amino acids. But you're not going to take back urea, water, and salts. Not the water, not yet. And not the salts, not yet. So, where some of that water and salt reabsorption would occur is down here. This is called the loop of Henle. You have a good number of things named after Henle in your body. So, for example, this little pink part of your eyes, like in the corner, a part of that is called the crypts of Henle. But anyway, the loop of Henle is inside your kidneys, kidney nephrons. Inside here, this is called the distal convoluted tubule. And literally that just means the convoluted, the small coil up tube to the back. Distal means to the back. And lastly, here we have the collecting duct. So, the collecting duct, what is the job of that? The job of the collecting duct is to collect urine, but also to help to regulate how much water is in that urine. So I know somebody wanted to go through that there. I'll go through that again just now. Now, we're going to make this a lot easier using a simulation. So now I'll have it look like a mess. So let me use a simulation to make this easier, right? Okay, let me look at the molecules here and see what's happening to what. Let me look at urea. Tell me here, put it in the chat, how much urea you want to reabsorb. Right, put in the chat how much urea you want to reabsorb. Also put in the chat how much glucose you want to reabsorb. You think some of it, all of it, a little bit of it, half of it? Tell me how much. Tell me how much urea you want to reabsorb and how much glucose you want to reabsorb. Right. So you don't want to reabsorb any urea, but you want to reabsorb all the glucose, not some, all. There's only one case here really where you're not gonna reabsorb all your glucose, and that's if you have diabetes, and that means you have too much glucose in your blood already. So no urea, but all the glucose, right? No urea, but all glucose. So I want to look at what happens to the urea. The urea molecule rolling like a marble, going through the whole thing. It rolls through the whole nephron. But notice how none of it actually went back into the bloodstream. Right? Look at it again. None of the urea went back into the bloodstream. The whole thing flowed right through the nephron and went right through the collecting duct. Now let me look at glucose. Glucose is this yellow dot that you're going to see here. Glucose. The glucose ends up going in because it's small, but ends up going back into the bloodstream. Right, so the glucose ends up going back into the bloodstream, and you notice that none of it actually makes it out. Let me look at urea and glucose together. Right, both of them could make it in because they're very small, but urea makes it out through the whole nephron, but glucose makes it into the bloodstream because you want to reabsorb all the glucose. You need a glucose for respiration. Now let me look at protein. Not amino acids there, but protein. Protein is really big. So protein can't even go into the kidney nephron. So protein doesn't even go into the kidney nephron, it's too big. So larger molecules like hormones, larger cells like red blood cells, white blood cells, those can't go in. Why is that? Because they're way too big. Larger proteins can't go in. Things like insulin, things like testosterone, estrogen, they can't go in. So let's look at the three of them now. Urea goes all the way into the bladder. Protein never even makes it in to the nephron. And and glucose makes it into the nephron but ends up getting selectively reabsorbed. Then we look at water now. So water is small, so it makes it in. Now notice that water is getting reabsorbed at some places. Even at the collecting duct, it gets reabsorbed. So I'm going to explain that just now. Alright, so look again. Water gets reabsorbed at the proximal tubule. It gets absorbed at the loop of Henle. And it gets reabsorbed at the collecting duct. Now we look at sodium. Sodium is very small. Sodium is basically mineral salts. Some sodium gets reabsorbed and some makes it into the collecting duct. Now what's important here to remember is this. What exactly makes it into the collecting duct? So what's the three things that make it into the collecting duct? Post it for me in the chat. There's three things that make it into the collecting duct. Now we look at all of it. Look at what gets reabsorbed and look what makes it to the end. Right, urea, excess water and excess sodium is what makes it into the collecting duct. That is correct. Glucose, things like glucose and proteins, they either don't make it into the nephron in the first place or they get reabsorbed. Right? So I'll type here. Small molecules gets filtered into the nephron, such as urea and glucose and water. However, only water, mineral salts, and urea reach the collecting duct and the bladder. Useful particles like glucose and amino acids all get reabsorbed and don't end up. Okay. So how does this fit into, right? So a person with diabetes is the only person who's going to end up with glucose in the urine because their blood glucose concentration is already very high. So they can't take in anymore. So the excess glucose ends up in the kidneys instead. Ends up in the kidneys and the urine instead. Okay, so what we'll notice now is that let me get a diagram there. Hold on, We get a simple one. Okay, so this here is the collecting duct. And remember what this does is that it collects urine. But what it also does is that it helps osmoregulate. It helps control, right? So with the help of a hormone called ADH, which stands for antidiuretic hormone, it controls how much water is in the urine and how much is reabsorbed into the bloodstream. Now, first thing we want to do is break down here. We want to break down here what the word antidiuretic means. That actually makes it easy. Is it safe to sell one kidney? Yeah, it's safe to sell one kidney. Unless unless your other kidney go on through, then you're in trouble. Let me break down antidiuretic. Anti means against or not. Diuretic is something that is make you pee. And a hormone, of course, is a chemical secreted by a gland that targets a cell or tissue. So let me break down what is antidiuretic then. So if a diuretic makes you pee more, an antidiuretic makes you pee less. That should be easy. So what an antidiuretic does, so what ADH does is reabsorb water from the collecting duct and put it back into the blood. So I want you to think of this now. All you to think of this. What type of day you would want to have less? You would need more water in your blood. What type of day? A hot day or a cold day? You would need more water in your blood. Right, so a hot day. So ADH is stimulated to be secreted on hot days or warm temperatures. So more water can be reabsorbed. Because what you need the water to do, you want to pee less and sweat more. Right? So basically this. Right? Break it down like this. Osmoregulation. ADH released. Pee less, sweat more. ADH suppressed. Pee more, sweat less. Cold day. Hot day. But what it actually does is that it makes the collecting ducts more permeable to water. Right? So that's what it does. ADH makes the collecting ducts walls more permeable to water. Allows reabsorption to occur. Okay, so that is excretion and osmoregulation. So reproduction and continuity and variation, right? Um, so I'm gonna do past papers for that one, for those ones. And anything that we bounce up, we'll go into it in a little more depth. The thing is this, all you want to go a little later than 10, all you need all is sleep. Hopefully all you wake up a little late today, you know. I say we could go until 10:30 probably. It's up to all you. If you want to go and sleep, go and sleep. If you know it is real, sleep away and thing. Well, if we look at some past paper questions. Alright, so the graph stuff, what you gonna do? Okay, so they say in the box below, draw a label diagram of the female reproductive system and using an arrow, trace the part that a spermatozoon, that's a sperm cell, would have to take to fertilize an ovum. Alright, so we look here. Alright, so keep in mind I'm drawing with a mouse here, right? So it will come out to righteous. Alright, and remember now, I gotta have a line in here. Right? So, yeah, not the best. But, yeah, I'll go draw it better. So, the part that it would take would be through here. Right, and we said that there's the egg. So there's an ovary. What's the possibility of them bringing a drawing? 10%. They'll bring a drawing, don't worry. There's the ovary. This is the oviduct or the fallopian tube. This is the uterus. Cervix. Zoom out. Vagina. And uterus wall. or endometrium. This is where implantation of the fertilized egg would happen. Right? So then drawing this is not too bad. 6 marks. So actually for a drawing, you should also title it, right? So you should always title your drawings as well. Right? So you put your title and underline it. Drawing of, the female reproductive system or something. Right? So okay. So here, since they say just label, you don't need to annotate. So the marks will be like this. Now, there's the thing I just always say, if you have time, because you have 2.5 hours, right? If you have time and you want to put extra stuff, put it. Because you never know if where you draw wrong or you're losing a little mark for something, put it, because you never know if they might overlook something that you put wrong and check that instead. So even though it's, watching the mark scheme, yeah, you want to label as much things as you could. If you want to annotate, but try not to waste no time on that. No, I don't do chem. I did chem, but I know enough to do a thing I had to review it. I'm a writer, game makers. Okay, so a young couple wants to choose between 2 contraceptive methods, barrier or surgical. Discuss why the male may prefer the barrier method rather than the surgical method of contraception. Your answer should include 2 advantages of the barrier method and 3 advantages of the surgical method. Alright, Olia. So let me see if we can answer this. Okay, so young couple. Oh my god, I think they're right. So 2 advantages of barrier. Alright, so first of all, surgical method is a vasectomy, which is cutting the sperm duct, right? Right. So 2 advantages of the barrier is that it also prevents sexually transmitted infections. It is also less expensive. We could also say that is more widely available. And here we have 3 disadvantages. For surgical, is irreversible or very difficult to reverse. Is more expensive. Anesthetic is required. Anesthetic is like, a painkiller. What do you all think is our next disadvantage? For surgical, I suppose our next one would be there's a chance of infection if done incorrectly. Because I mean, what they're doing is that you're actually cutting the sperm duct. So it's is a invasive. It's more invasive than barrier methods, and so yeah, right. So that so that's one too. It doesn't protect against STIs. Right? So easy common sense question. Recovery time is a good one too. All the recovery time is fairly short. But just to show you here. Alright, that's now Alright, shucks, just now Alright, so like they are the vast difference, right? So this is scrotum. And what they're doing is that they're gonna inject the anesthetic into it. And right, so it's a slightly invasive procedure. So if it's done incorrectly, this is the vas deferens, also called the sperm duct. So what they need to do is they need to cut and ligate that. Alright, so what they're going to do is they're going to cut it, which is what you're seeing here. Right, so they're going to tie up the end. It could be reversible, but it is very difficult to undergo. So that'll cut the end of it. And then tie it back. And then they'll do it to the other one. Well, they get the gist. Well, all you feel is YouTuber find himself. Mary, I intentionally doing all that so YouTube will take it on. Okay, you continue. Okay, a biologist discovers that the species of flowers of a species of plants have no reproductive parts, no male reproductive parts. So no male reproductive parts. Suggests one method by which these plants produce offspring, and 3 advantages of this method. So 3 advantages and one method by which these plants produce offspring. Okay, so imagine that they don't have any male reproductive parts, right? So that's one method by which these plants produce offspring. Okay, so this would be... Now they didn't say that the plant had no reproductive parts, they say no male. So there's 2 answers that could work here, right? Now we have all female plants, like if you have a wheat plant and thing, you have male ones and you have female ones. You also have plants that just don't have any reproductive parts on the whole. So it can't be self-pollination, because to self-pollinate you need both male and female plants. So you have 2 things that could work here. So you have asexual reproduction or cross-pollination. Because it has some plants that is female, that could be male and female. So if your answer is asexual, the 3 advantages is this: rapid reproduction of plants, right? It could be cheaper. There's no need for a second parent. And next thing is that it could be done in a human controlled environment easily. Traits can be easily preserved from generation to generation. But I think that the better answer here is actually cross-pollination, because they're talking about male and female plants here. So no gametes required here. That's a good one. But if your answer here is cross-pollination, right? Advantages would be wider gene pool. Well, no variation is a disadvantage, not an advantage. So a wider gene pool. Plants can have a mix of various characteristics and gain desirable ones. And I suppose our third one here could be plants can undergo natural selection easily and eventually evolve. Less susceptible, I should say, to diseases and pests. So basically, when you have sexual reproduction, you could lose some. So let me say that you have a very red, nice flower. If those flowers cross-pollinate with other flowers, it can lose that nice red color, so you can end up losing desirable traits. But if you self-pollinate, it could end up keeping those desirable traits. Asexual reproduction, you don't need an ex-parent, you don't need gametes. You could reproduce rapidly because you only need one parent, as I say. And any advantages traits that it has could be preserved as it goes from generation to generation. The downside is that you don't have variation. So variation is kind of like this. Imagine if everybody on Earth was the same and COVID reached, right? And one person died of COVID. And everybody have the same immune system. Everybody have the same genetic makeup. Well, if one person dead, I mean everybody have the chance of dying. But that's not necessarily so, because everybody has a different immune system and genetic makeup. So when things sexually reproduce, it encourages a wide gene pool, it encourages variation, and is less susceptible to diseases and pests. Yeah, well, cross-pollination could be wind pollination. Alright, so alright, so you look at this one here. Sickle cell anemia is caused by the presence of a recessive allele. Persons who are heterozygous and carry the sickle cell allele are more resistant to a type of severe malaria than persons who are homozygous for the normal hemoglobin allele. State what is meant by each of the following terms. Homo means same, and zygous come from zygote. Hetero means different. So homozygous means, right? So this occurs when the genotype carries two similar alleles. So example would be in this case here, capital A, or common A, common A. Heterozygous occurs when the genotype carries two different alleles. So example, capital A, small a. Very easy, right? So two identical alleles, two similar alleles. Okay, so now just to give all this some definitions here, an allele is an alternate or variant of a gene. So example, blue eyes and brown eyes. And a gene is a sequence of DNA that codes for a protein. So for example, you have a gene to produce melanin. Melanin is a protein, and the more of that gene that you have is the more melanin that you produce, so the darker skin you would be. Proteins, genes and proteins express phenotype. Which are the physical characteristics exhibited by genotypes. Physical or observable. So example of phenotypes would be like blood type A, or blue eyes, or dark hair, or attached earlobes, or having a widow's peak. That is what is a phenotype. So hopefully we get a question like this one here. Yeah, Shiva, that would be fine. With the aid of a genetic diagram, show how a man and a woman who do not have sickle cell anemia can have a child who has the disease. So a lot of the time, for doing these Punnett squares or test crosses, the situation is be this. You have two parents who do have something and the child is have it. So in this case here, both parents have to be heterozygous for the trait. So assuming that the trait is recessive, of course. Pedigree charts, okay, no problem. But you don't really get pedigree charts in as far as I know, in bio. So this is what you want here. So we're gonna use, wait, let me see how much marks this is. So this is 6 marks, right? So we're gonna use A as a dominant healthy allele and common A as the recessive sickle cell allele. Now usually you use HBA and HBS, right? But you can actually use anything that you want. I just using this to make it a little easier here. So you could also use hemoglobin A and hemoglobin S. Right? So you could also use this. Alright. So how are you gonna get your 6 marks for this? Well, you need to show it like this. Parent phenotypes. Healthy. Healthy. So there's the male. That's the symbol for Mars. Mars with his shield and his spear. And there's the symbol for Venus. There's the female with, you know, basically it's supposed to represent holding a mirror. Because apparently that's what women used to do back then. Alright, so parent genotypes. I using A as I said instead of HB. Genes. Well, not genes, sorry, gametes. So we're gonna put capital A, small a, capital A, small a. So in order to get your marks, you have to do it like this, right? Like I had to show all the different steps. And then what we're gonna do is gonna draw the Punnett square. This is going to be put it another. Well, in this case here, we can't tell if it's male or female now because it's not a sex-linked disease. This one, it could be any sex. Anyway, so if we do this here, you just have to match these here. So this will be capital A. Capital A, this one inherits both dominant alleles from the parents. This one will inherit this capital A, common A. Capital A, common A. Always put the dominant one first. And common A, common A. So the next thing you want to do is write a ratio. So the genotypic ratio is going to be, here's one homozygous, one homozygous dominant, two heterozygous, one homozygous recessive. And the phenotypic ratio is going to be 3 healthy, right, to 1 sickle cell. So it's an easy thing once you know what you're doing. But let me give all you the next one here, right? Let me type one. They will say that we have a mother does not carry the hemophilia trait, but the father is a hemophiliac. Hemophilia is an X-linked recessive disease. What are the chances of these parents producing a child who suffers from hemophilia? Right. I'll type anything, right? Okay, so what we need to do is figure out here was the mother's and the father's phenotype. So the parent phenotypes of this. So we have a healthy and I have a hemophiliac father, right? So there's the mother, there's the father. The parent genotypes. So the mother, remember the mother has two X chromosomes. So she's going to be XX and we're going to put the alleles on that, right? And the father has XY. Now the problem is this. When you see the Y chromosome is smaller than the X chromosome, so it lacks a space to have the hemophilia allele. So what happens here is this. So the mother, they say, does not carry the hemophilia trait. So she is X capital H, X capital H. And the father, he is a hemophiliac, so he is going to be, and they say is a recessive disease. So he is X common HY. Now the Y, we're not putting a allele on here because the Y allele doesn't have the hemophilia gene on it. So we're going to have the gametes. So the gametes are going to be the following. So the mother is going to be X capital H, X capital H, and the father's gametes are going to be X common H and Y. Yeah. So drawing the Punnett square now. So drawing the Punnett square now, let me see how we could figure this out. Right, so here we're going to have the father, and he's going to be X common H, Y. And the mother is going to be X, this gamete will go here, X capital H, and this gamete will come here. So, let me fill out the chromosomes first. So XX, XX, XY, XY. So this is going to be X capital common, X capital common, X capital, X capital. So what's the chances of them producing it? Well, outside of a good one, the answer is 0%. Because we could see here that both boys, right, so they don't suffer from hemophilia. And the girls are carriers, but they don't suffer from hemophilia either. So let me go through one more example. Let me say that we have this. Now there's the next thing we have to understand here, right? Is that these people not making 4 children. I mean, they could make 4 children, they could make 3, they could make 2, they could make 10. But this doesn't mean that these 2 parents making 4 children. It is showing the chances or the possibilities per child, right? So it's showing the percentages per child. How to know if they are carriers of this? You'd have to do a genetic sequence of them. But typically carriers don't suffer from the disease. Okay, so let me say we have this. We have a hemophiliac mother. We have a carrier mother, right? And a healthy father. Right? So we have So the father is going to be X capital HY. Remember the Y don't have a allele on it. And the carrier mother is going to be this. Right, so you do the same thing. Healthy girl. Healthy girl. Carrier girl. Still healthy. Healthy boy. Remember, boys can't be carriers because only have chance of getting one allele. And right, so hemophiliac boy. Right, so hemophiliac boy, 25% possibility. So that means if these people had a child, there's a 25% chance it would be a hemophiliac boy, a 25% chance it would be a healthy boy, 25% it will be a carrier girl, and 25% it will be a healthy girl. Well, non-hemophiliac anyway. Yeah, you could put the ratio and still get marked. If they ask for a percentage, per percentage though. It's actually very rare for girls to have hemophilia because they would have to inherit. There's really one of the only cases where you could have a hemophiliac girl. But actually most girls who have hemophilia just die. So, I mean, just die very early on. So if we had this, then this case here you would have a hemophiliac girl. So, but a lot of them die upon childbirth. So it rare. I mean, they could survive, but rare. But they'd have to inherit a recessive from the father and a recessive from the mother. So that means the mother would have to be a carrier and the father would have to have hemophilia. As I said, most hemophiliac girls don't make it to child-rearing age. Some do, but yeah. Alright, so look at the next question. Explain why the sickle cell allele is more common in the population in countries where the severe form of malaria is present. Well, they explained this already. They say that people with the sickle cell allele, they say that people with the sickle cell allele attain partial immunity against malaria and thus have a lower chance, a lower fatality rate against the disease. These people can reproduce and pass on those recessive sickle cell alleles to their children, who would also have a higher chance of survival. So, the sickle cell allele is a recessive allele that if homozygous will give you sickle cell anemia. So, having two sickle cell alleles, right? You have sickle cell anemia. Having one sickle cell allele, you're healthy and strong against malaria. Because the malaria protists can't survive in the erythrocyte. Alright, so look at the next past paper. Yeah, Karishma, it could come because continuity and variation is this whole thing. All right. Let me look at this one. Actually, that's not. Okay, let me look at this one here. Sexual reproduction in both plants and animals requires the movement of male gametes to fertilize female gametes. Meiosis results in the formation of gametes. List 2 important events that occur during meiosis that can lead to genetic variation among offspring. Alright, Ole, so let me explain what is meiosis here, right? Let me get some space here to draw something. Alright. Mitosis is cell division that results in clones. Right? What that means is that daughter cells are identical to parent cells. They have the same number of chromosomes. And they call that a diploid or 2n number. There are two sets of chromosomes. Right? Daughter cells are identical. And only 2 cells are produced. So basically, you have one cell, let me say with 46 chromosomes, that's going to split into 2 cells. Right? So you're going to have 2 identical cells. Meiosis is cell division that is used to form gametes. It produces half the number of chromosomes, or haploid. So with meiosis now, right, you're gonna get this 46, then it's gonna divide, and you're gonna get 1 cell here, 2 daughter cells, and you're gonna put 4 daughter cells in the end. So it's 2 divisions, and these cells each gonna have 23 chromosomes. Basically a sperm or egg. Mitosis could happen in any cell, anybody. It could be an epithelial cell, a neural cell, it could be a cell in the alveoli, a cell in the ileum, it could be a cell inside your kidney nephrons, it could be a cell in your toe, it could be a cell in your eyelid, it could be a cell, a cheek cell, it could be a tongue cell, it could be a cell in the interstitial fluid, it could be any type of cell. But a meiotic cell, a cell that's reproduced by meiosis, would only form a sperm or egg. So these would only be sperm or eggs. They are limited to sperm or eggs. Okay. So, now, let me get into here what happens during meiosis. The difference. So during mitosis, right, we are going to get, during a phase that is called metaphase, we are going to get a bunch of gametes. Not gametes, we're going to get a bunch of chromosomes lining up in the center. So this is metaphase and mitosis. I feel we have a Cape person come here. This is CSEC bio, right? So not Cape bio. So there's not unit 2 or unit 1, there's CSEC. Alright. So metaphase 1 in meiosis. There's actually a line like this. That is pair up. So what happening here is that they have something called crossing over. Right? So you have your spindle fibers and things trying to pull them apart. The difference is that they pair up. In meiosis, the chromosomes pair up during metaphase 1. That's the first. Okay, prophase, metaphase, anaphase, telophase, right? By the way, meiosis 2 is just mitosis. Meiosis 2 is just like it have no pairing up or anything. So the chromosomes pair up and crossing over occurs. So what exactly is crossing over? So crossing over is this. Right? So crossing over is this. Imagine if we have chromosomes hooking up like this, right? What could eventually happen here is, you see this little point here? It actually have a name for this point here where it is meet up. That's called a chiasma. You need to know that at this level. So what happened here is that this here is going to swap genetic information. So you're going to get something not looking like this. So you see now they're swapping information. This is what crossing over mean. Now the thing about this is that crossing over could happen at any point in the chromosome. It could happen at any locus in the chromosome. Locus is like a point. So it could happen at the top, it could happen at the bottom, it could happen at the foot, the head, it could happen anywhere. So you have an almost infinite number of possibilities that could happen. This is why each sperm cell and each egg cell is different from each other. So when a man ejaculates, he lets out 300 million sperm cells, right? And a man ejaculates on average probably 5,000 times in his lifetime, maybe more, maybe less, depends on how active he is. And each of those 5,000 times multiplied by 300 million, it is almost impossible that two sperm cells would be the same. So this is what results in a large amount of genetic variation that occurs in gametes. So not just sperm cells, but also male gametes that are in pollen in plants. So this leads to a very, 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 very wide gene pool, and it leads to a large number of possibilities that occur. So this is where it's called crossing over. So coming back here, now by the way, if you want to remember each thing, so all in Form 5, right? Something that all didn't get is a prom or a graduation. So just the stages of mitosis, but let's go through it quick. Right. So before mitosis, you have something called interphase, where DNA replicates. Right? So then you have prophase, metaphase, anaphase, and telophase. So interphase is not really considered part of mitosis. It happens before mitosis, which is basically the chromatids doubling up. And so this is why when you have chromatids, you have two hook up like an X. So prophase is kind of like this. Prophase is like you have chromosomes forming, right? So is basically a lot of the chromatin, which is like threads. What the threads are doing is that they form in large structures called chromosomes. So it's kind of like Granny knitting a little sweater. She taking the little yarn and she knitting a nice little sweater. The sweater is the chromosomes. You just study when I finishing now. You just study when I finishing. Just enjoy the show. Metaphase is when the chromosomes meet in the equator of the cell. Right, so they meet. So I think metaphase, they meet in the middle of the cell and spindle fibers form. Anaphase. The chromosomes are angry with each other and are pulled apart by the spindle fibers. So when people start to fight and vex with everybody, and then you have people pulling you apart. They say, "Hey, don't fight now, man, don't fight." So say, "Don't fight now, man." So the anaphase is the pulling apart the angry chromosomes, because they're meeting up in the middle and the fighting. So the spindle fibers pulling them apart. The telophase, the chromosomes are so far apart now that they have to call each other on the telephone. Right? So what happens now is that basically nuclear membranes reform as a result, and the cell begins to separate. And lastly, I have cytokinesis, which literally means movement of the cell. Cell splits into cytoplasm, splits into. That is it. It's easy. Or let's treat this like it's some big hard thing. Little babies could do this. Just remember PMAT. You're peeing on the mat. Oh, I'll move you up here. Yeah. So, prophase, right? It forming like the things forming. Metaphase, it meeting in the middle. Anaphase, you're angry and the spindle fibers pulling it apart. The telophase is so far apart now that I have to call on the telephone, and there's two nuclei. The cytokinesis literally means, the Latin for cyto is cell, and kinesis is from kinetic, which means movement. So that means the cytoplasm is splitting in two and they're moving apart. What's happening there is that the nuclear membrane is reforming, the nucleolus is reforming, and the chromatin is going to decondense to the chromosomes. So the whole cell cycle is going to happen again. So basically cell cycle is 95% of the time, and mitosis is only 5% of the time. So it's very easy. But meiosis on the other hand is almost the same thing. Meiosis is almost the same thing. But no, you don't need to know the stages of meiosis. You just need to know that it's different in a way that they cross over. You only need to know in meiosis that the chromosomes pair up during metaphase 1 and they cross over and exchange DNA. Nah, but you don't need to know all of that. I say it's very easy. Okay. So coming back here. They come back here and answer the question. Where the question? They lost the question. Blast out the question. Do. Alright. Let me get back. Right, so the question is meiosis. There's two important events. Real easy. Meiosis. Basically, you have chromosomes pair up during metaphase 1. Chromosomes undergo crossing over and exchange of DNA. That's it. Okay, so this one here could be a little tricky. Describe the process by which the male gamete reaches the female gamete in the flower. I know all this off the top of my head because it's very, very easy. Okay, so let me draw this out. Imagine this is the pistil. This is a carpel of the pistil, right? And we know the pistil is the female part of the flower. Alright, so I could draw that better. Alright, so inside here, what we need to do. So how are we gonna describe what happens to the what happens during double fertilization? So watch here. What we're gonna have is we're gonna have a flower, right? And that flower there is gonna send pollen onto a stigma, whether it's self-pollinating or cross-pollinating. So you're gonna have pollination occurring, right? So you're gonna have pollen moving from an anther to a stigma. This is the stigma. That should be stigma, right? This is the stigma. This is the style. And this is the ovary. And inside here, this is called the embryo sac. Alright, this is a drawing of okay, so what's gonna happen here now? If we was to zoom this out to see what's happening. There's what going on here. This is the pollen grain here, right? There's the pollen. So what's going to happen here is that this is going to grow a pollen tube. And this pollen tube is going to grow all the way down the style, the ovary, and you're going to loop all the way around and come up into the embryo sac. This is the pollen tube. What exactly is the pollen tube? Well, I want you to imagine this. The pollen itself is not the gamete, but the pollen contains two male gametes. So the pollen contains two male gametes, right? These here, these are our two male gametes. Well, there's a zoom up version of it. These are the two male gametes. So unlike men, who does release 300 million, one pollen grain is only released 2. So it might be like, well, why 2? And there's only one flower that we have to fertilize, right? So Why is it called double fertilization? Why is it called double fertilization? Because the two male gametes have to fuse with two structures, right? The egg to form the zygote and the endosperm nucleus or the polar nuclei to form the endosperm. So let me explain here what exactly is happening. So let me start over. This is a carpel of a pistil. The pistil is the female part of the flower. So there's the stigma, the style, and the ovary. A pollen grain is being deposited onto the stigma. Once it is deposited onto the stigma, it is going to release two male gametes. Those two male gametes are going to move all the way down to the ovary where the embryo sac is, right? And one male gamete is going to fertilize the egg, and they're going to get the zygote, and the zygote going to turn into basically a baby flower. The polar nuclei or the endosperm nucleus, either one, is going to form something called the endosperm. So what is that? What is the endosperm? Well, the endosperm is kind of like a plant placenta. So it's kind of like a plant placenta. It's there to give it food while it is a seed. So I'll give you an example. It could be in a liquid form, it could be in a solid form. It is basically plant food. So coconut water is an example of endosperm. The coconut is the fruit, there's a seed inside, and you have coconut water. So if you drink coconut water, you drink real endosperm. So endosperm is just nutrients. You know, people like to drink coconut water and say it has so much nutrients in it. The ovary wall turns into something called the pericarp. And the pericarp is basically the flesh of the fruit. So using coconuts as an example, coconut jelly is the pericarp. Using mangoes as an example, the little flesh of the mango, the sweet part, that's the pericarp. Using an apple as an example, when you bite into an apple, that sweet part, that is the pericarp. So let me do a zoom up on what's happening here now. So there's it step by step. Pollen grain lands on stigma. This is pollination. Pollen grain grows a pollen tube that travels down the style and to the ovary. Two male gametes right. So that is basically what's happening here. So the pollen grain lands on the stigma. That's basically pollination. The pollen grain is going to grow something called a pollen tube. It actually has a cell in it that grows that pollen tube. So that pollen tube goes all the way down and travels to the ovary. Imagine the pollen tube as a tunnel. The two male gametes, right, going to flow right down that pollen tube, and they're going to go to the ovary. One of those male gametes is going to fertilize the egg, and that's going to form the zygote that forming the baby plant, the embryonic plant. That's the thing that's going to form the plumule and the radicle and the whatever, right? The second male gamete is what going to fuse with the central cell, which is also called the polar nuclei. You might know it as the endosperm nucleus, anything, right? But are going to form the food source for the seed. Because guess what? The seed don't hook up to the parent plant. The seed busts out at the parent plant. So imagine is this, right? Imagine a woman having a baby, right? But the woman having the baby outside she uterus. So what's going to happen there? You need to have a food source, right? The plant can't get pregnant. What happens there is that the plant sheds the seeds after fertilization, and those seeds are dispersed, whether it's through wind or whether it's through an animal or whether it's through mechanically or self-explosive or whether it's through water or something like that. So you look at the question. Describe the process by which the male gamete reaches the female gamete in a flower. Okay, so you don't have to talk about double fertilization so much, but basically is this. The pollen grain is transferred to the stigma of a flower either by wind or an insect. A pollen tube then grows down the style of the flower and deposits two male gametes from the pollen grain into the ovary sac. One of those male gametes will fuse with the egg to form the zygote. The other one will form the endosperm, which is actually a triploid nucleus, which means that 3 sets of chromosomes. Okay, so let me look for some natural selection questions or something. Let me look for more genetics questions. Let me see. Hopefully all are holding up. I'm looking for stuff that could be a little tricky. Yeah, and not hard to keep in your head. If you like the subject, well, what probably happens is all the teachers make all they hate the subject. So, but sometimes you have to be independent. I mean, I went to one of the worst schools in Trinidad and Tobago, which is Presentation College Shogunas. Basically a bootleg prestige school, which make you hate every subject and didn't really care about it unless you was getting a scholarship. But I mean, sometimes you have to be the captain of your own ship. You have to be the master of your own soul. I'm looking for genetics questions. A lot of these involve sickle cell anemia, boy. I just know. Irritability is on the syllabus, but it not on the broad topics. Okay, let me look at this one here. That real thing about like blood type and things. Sally and Prez, hard luck there, boy. I don't know how it is now, but back then, I mean, it is a bootleg prestige school. It's not a real prestige school. It just masquerades as one. All right. Wait, but I can't find any natural selection questions, boy, just now, eh? Or any genetic engineering questions or something. Aha, okay. So I find a good one here. Alright, let me look at this one here. Alright, let's look at this one. Mutation and sexual reproduction are two processes which can lead to variation in the population of a species. State one way in which the processes are similar and two ways in which they differ. What y'all think? What's two ways in which mutation and sexual reproduction are similar and two ways in which are different? Or one way in which they're similar. So similarity between sexual reproduction and mutation, and two differences between sexual reproduction and mutation. Okay, so both of them result in genetic variation. Right, so that's the similarity. Differences, right? Okay, so mutation is a random change in the DNA. And alright, so sexual reproduction. Sexual reproduction involves gametes, while mutation does not necessarily involve gametes. It can occur during asexual reproduction. Right, so easy. Both of them basically involve traits passing down, but one involves the random change in a trait, so that's mutation. So for example, Down syndrome. Down syndrome is where you have an extra chromosome being put into one gamete, and that there's a mutation. It's random, and it is not necessarily passed down from the parent's genome. It's a random change, but it is an example of genetic variation. Okay. Explain the differences between variation in a community and variation in a population, and suggest one way in which diversity benefits the community. Okay, so if you remember, a community is a group of different species in an ecosystem, and a population is a group of individuals of the same species. So a community could be termites and ants and grasshoppers living in a tree or something. And a population would just be the group of termites. So this one is not bad. This one is just individuals of a community would genetically vary more than in a population due to being different species. Diversity allows survival of the fittest and symbiotic relationships between various organisms. Alright, Lisa Simpson, I don't know there's a real name, but the Down syndrome is random, so there's really no percentage I could give for it. Environmental variation, I think they talk about genetic variation, but you could have environmental too. So you could talk about either one. Alright, so you look at this one. Alright, that's not it. Ah, okay. We have a nice one here. Name 3 characteristics which distinguish a biological species and 3 factors that lead to the formation of a new species. Okay. Well, y'all can remember doing natural selection. What's the name of the scientist who basically discovered natural selection in a species? Abiana did enzymes already. You could check the live stream like 1,000 hours ago and you'll be able to see it. The also Max, there's something here. So let me basically explain natural selection, right? So natural selection is kind of like this. Imagine that a bear is chasing you, right? So there's a bear that is chasing you. So imagine that there's a bear chasing you and your friend. Right, so Darwin is his name. There's a bear chasing you and your friend. You don't have to be faster than the bear. Why don't you have to be faster than the bear to survive? A bear is chasing you and your friend. You don't have to be faster than the bear to survive. Why is that? You don't have to be faster than the bear. You only have to be faster than your friend. So that's an example of natural selection. So you only need to be faster than your friend. You don't need to be faster than the bear. Because what natural selection is, right, is survival of the fittest. Now the fittest doesn't necessarily mean the strongest or the fastest. The fittest could be a number of things. So it could be the ones that are best able to adapt to the environment. So example, the ones that can camouflage and elude predators. So that is what you have to do there. Well, if you push your friend along and run, Enter is strong. Enter is the fitter in some sense. You're smarter than them at least. So That is example of natural selection. Now, let me explain here what Darwin observed, right? So Darwin observed finches, and he went to this group of islands called the Galapagos Islands. And I'm gonna find it here. Right. So he went to this group of islands called the Galapagos Islands. And on each island, what happened there is that a storm blew some finches, which is a type of bird, onto these islands. So what Darwin observed, right, was that you had one type of finch at first. Well, he didn't observe it, he basically alluded to this, that there was one type of finch at first, right? But that one species of finch had genetic variation in their beaks. So you had some finches that was good at, that basically had thick beaks. You had some of them that had long beaks. Well, my jaws are smaller. You had some of them that had longer beaks. Right. So remember these here, same species. Kind of like how you have German Shepherds and Chihuahuas. Both of them are dogs. It is different breeds of dogs, but they're still the same species. Now, let me think about what the birds could eat. Right. So some of them could eat nuts. So these birds here, the thick-beaked birds, ate fruits, seeds, and nuts. The thin-beaked birds ate insects and worms because they could have pecked into the bark and the ground. So the point is here now is that if me and my friend, or me and a group of people are alone on an island or something, right? And all are we eating the same thing, all are we eating the same fruit, well, what's gonna happen? Eventually the fruit's gonna run out, and we're gonna have more people than fruits, and we're gonna kill each other for the fruits. In other words, we're gonna have competition for the fruits. But since these birds now, since they had such a wide diet, they didn't have to compete for food. And that's a good thing, because now what happened here is that they could have separate into ecological niches. They could have separate into niches, basically different groups. So what happened now is that over time, so as I said, I had no competition, or very little competition. Now, our next thing that they had was no predators. There are no natural predators. So because of this now, they was able to have a lot of sex and children. Well, offspring. So what happened here now is that eventually the birds now with thick beaks had sex, right? Because there was basically hunting for the same, there's foraging for the same types of foods. And eventually now, they would have made birds with even thicker beaks. These birds with small beaks would have sex, right? Right, they would have bird sex. And eventually, they would form, they would result in birds now. And this happened, remember, this didn't happen from today to tomorrow. This happened over hundreds of years. So what we had now was two types of birds that do not want to have sex with each other. How they have sex? They have male and female reproductive parts. If you look up a duck penis, by the way, a duck penis look like a corkscrew because the female duck have a corkscrew vagina. Okay, so instead what we have here is speciation. We have two different species. Because what happened is that we get the two types of birds differing from each other so much that they don't even want to mate with each other anymore, that they're so genetically different, right? That they become two different species, not different breeds of the birds, but two different species altogether. So this is how that happened. Now, if we look here, and it could be different reasons. It could be that the courtship that didn't, one of them just singing different from the other one. It could be a mechanical thing. One of the penis shaped too weird for the other one vagina, so they don't want to mate. It could be things that it could be just that they have that one of them is awake in the daytime and the other one is awake in the nighttime, so they never get to even meet up. So that is natural selection leading to speciation. Now, if we have two different species, so if two different species produce offspring, which they can, they will likely produce an infertile member, infertile organism. So examples would be like this. See if I could name these organisms there. So what we have here, we have a liger. A mule is our next example. We have a liger, a zedonk, and a jaglion. So these here, yeah, you have zonkies too. It depends on which one is the male and the female. So you can have hybrid species, hybrid organisms, but these here would be infertile. And mules are next example. If you have a horse and a donkey, it produces a mule, and mules are always infertile. So it means that they can't continue as species because they themselves are not a species. Okay, so coming back to the question. With the question here. Name 3 characteristics which distinguish a biological species and 3 factors that lead to the formation of a new species. So 3 characteristics of a species. So what that would be? That would be that they have similar genetic characteristics, similar biochemical or physiological characteristics, like the same type of organs. and they are able to reproduce, well, mate with each other and produce fertile offspring. 3 factors that lead to the formation of a new species. Right, so one of them could be geographical isolation, which is these species are separated by some physical barrier. So, for example now, let me say that the Grand Canyon, right? Which is basically two mountain structures with a big gap in the middle. And you had a squirrel that was around before the formation of the Grand Canyon, long time ago. And what happened there is that when the Grand Canyon formed, those squirrels eventually got separated from each other. So they turned into 2 different species. So they look like on one end of the Grand Canyon, they look very different from the ones on the other end. The next thing that you have is mutations. Random changes in DNA that can be advantageous. So I'm going to show you an example. Alright, let's start looking for something. Alright, so if I look at my stream here. Alright, so this is a legged snake. They still exist. They kind of look like salamanders. But a long time ago, these snakes here had a mutation so that they wouldn't have legs. And those snakes that didn't have legs, actually, that was better for them, but it was a mutation. So those snakes that didn't have legs, they was able to move and burrow faster and escape predators. The ones with legs actually had a disadvantage. So these ones with legs, we assume that they eventually evolved to become salamanders or something like that. So this is an example of a mutation actually being advantageous. So we have very early versions of species that I'm going to show you a next example. Alright, so I'm going to show you a picture here. Alright, so this organism that I'm showing you here, just now, There's the early version of all I could figure it out. What animal or the thing there's the early version of. Nah, so now crocodile, you know, Jaden is correct, is a whale. It's the early version of a whale. And eventually over time, so they had a name for it. This is a Myocetus. Not around anymore. Over time, The ones that remained in the ocean eventually became whales. Lost the fur and gained fins instead of phalanges. Well, it's not phalanges. Instead of webbed feet. The ones that crawled onto land became hippos. So the location of the organism could actually change it could actually lead to speciation, because if we take an organism and put it in a whole different geographical location, it would have to overcome a whole set of other pressures. It would have to overcome a different climate. It would have to overcome different availability of water. It would have to overcome a bunch of different things. So let me come back here. So geographical isolation could lead to things coming out in different ways, because they're basically exposed to a bunch of other pressures, what you call selective pressures. Right, so the last factor that could lead to the formation of a new species is well, basically formation of various courtship behaviors. So various courtship behaviors is like example, the finches that form groups due to having different diets and mating rituals. So it's basically like this: if this one bird singing one song, the next bird singing the next song, they wouldn't want to mate anymore. And eventually, they would just mate with their own kind and they would turn into two different species. I must see what somebody asks this. Somebody asks, what if we really came from monkeys? Where I come from monkeys? We and monkeys have the same ancestors. So we come from a type of organism called a hominid. Or primates. I'll put it there. Okay, so yeah. So I was typing there that the only thing that's really separate in fact, it hypothesized that had 8 different types of humans back then. What happened to the other 7 types? Well, we probably killed them out, honestly. Nobody here really know what happened to them. We probably cut off their head and kill them, so they don't exist anymore. So it is a very, very complex history. It's more than anything that you could imagine. But just to give you the rundown of it, a long time ago, we would have put artificial selection. Artificial selection idea is human control. So artificial selection is not really what you want to look for. It's more natural selection. Anyway, but yeah, a long time ago, humans would live in many different places, and this would of course a lot of humans to have different courtship behaviors for them to look differently from each other, and look differently from their primate cousins. So ones that would have remained in the forest would be like the chimpanzees, the orangutans, the all the other primates that we know, the ones that made it onto flatlands because they was forced to stand on two legs. It would have actually changed their voice boxes, and the voice boxes is actually what led to development of language, and what led to the development of language led to the recording of language. The one difference that we have, humans have, that other species don't, is there is recording of language. Of course, all species could communicate with each other, but they can't actually record language. So same way how I could type something here and only reading it, or people could make cave paintings on a wall, or people could write things down and could draw things, as recording of language. So I could learn something that people learn 1,000 years ago. That's actually what makes humans different. Okay, let me do a few more and then we're done. Okay, so you look at this one. So Hidia there was asking about artificial selection. This is some differences between the two, right? So Spanish stream, nah. Natural selection occurs due to organisms being able to adapt to the environments and changes in the environment and survive over other organisms, right? Artificial selection, basically selective breeding, occurs when organisms are led to reproduce with each other based on desirable traits. It is a human control process. Example, when a cow is forced to mate with a stud at a farm to produce more meat. Genetic engineering occurs when a trait is modified by the insertion or alteration of DNA in the organism. So like example, glowfish are created when genes from bioluminescent jellyfish are placed in them. I will move him just now. So you usually ask me to compare these three. So that's the difference there. So artificial selection is really when you just do it like in a so I'll give you an example. Artificial selection is a human control process, like when Hitler wanted to wipe out everybody who had blonde hair and blue eyes. He wanted to wipe out everybody who had dark hair and dark eyes. That's kind of like artificial selection. It's kind of like, and then he used to have the camps where he used to force women who had blonde hair and blue eyes to mate with men so they could make the Aryan race. So that's the difference, is where you would force the process so you could pass on desirable traits. Not to say that Hitler is a good example of anything of humanity, but as an example. Genetic engineering is different from these two because it is actually a process that involves enzymes and laboratory settings in which you would actually cut out a gene, like you would use like a genetic scissors in a way, and you would paste that gene. Imagine like you cut and paste a gene from one organism and put it in the next one. It's actually a lot more complicated than how I just put it, but that's genetic engineering. So a good example of genetic engineering is this: if to produce insulin, let me say, anybody could tell me where insulin comes from? Well, insulin comes from the pancreas, of course, but you all know where we get today. Also, where they used to get long ago. If all you ever see Jurassic Park or Jurassic World, that is actually used genetic engineering. I mean, it's not real, but they use genetic engineering technology. What they do is that they had dinosaur DNA from a mosquito. Like the mosquito suck the blood of a dinosaur, and they fill it in with frog DNA, and they make dinosaurs from that. It sounds crazy, but the man make millions of dollars from that idea. The author. So anyway, right. So insulin used to come from pigs. But what is meant is that everybody who was Muslim and Jewish and vegans couldn't use it, right? Because it was against the religion. This made insulin very expensive. It's because it basically had a low supply and a high demand, which basically means a high cost. Anybody who doing business. And what we do now is that we use bacteria to genetically engineer insulin. We use genetically engineered bacteria, sorry. So how they do this is that what they'll do is that they will have a bacteria, a E. coli bacteria, right? And you would have this thing called a plasmid. A plasmid is like a circular piece of DNA, which makes it easy to manipulate. And what you'd have is a human cell, right? So you'd have a human cell from the pancreas. And what they do is that they combine the DNA from the two. So basically they cut out DNA from a human, right? So they cut off a little piece of DNA and they put it inside the plasmid DNA. So imagine this, like there's a little piece of this here. That's the human DNA. So this is called, no, they don't use pigs again because they could use bacteria to do it now. So this is called recombinant DNA, which is like both versions, both sets of DNA combined. So they combine the human DNA and the bacterial DNA to make recombinant DNA. So the type of bacteria that they use is something called E. coli bacteria. E. coli stands for Escherichia coli. And what that is basically found in your intestines, is also found in feces, is also found in UE doubles. And so that E. coli bacteria is something that is already found in our bodies, is already found in our large intestines. So it's easy to integrate into the body. So what they do is they combine both sets of DNA. No, they didn't use the yeast. They use E. coli back. Oh, they use yeast now. Okay, but back then they use E. coli. I actually don't know. And they put this back into the bacteria, right? So they put this new into the bacteria, right? So now what we have, yeah, it's in UE doubles. When I was in UE and I was studying microbiology, we did a pathology experiment on UE doubles, and it had basically bird E. coli in it. So maybe it was just that one day a bird fly and poop in the channel, but it had. So anyway, so here we have E. coli bacteria that is able to produce insulin, because what we have here is the insulin gene that they extract from the human cell and they put it in the E. coli bacteria, and now that is able to produce insulin. So the insulin now we don't need pigs anymore. We could just put that bacteria in a fermenter and give it some oxygen and give it some glucose and let it asexually reproduce. We basically get in a very high demand product, a high supply of it at a low price, so that it's affordable to everybody who have diabetes and they don't have to dead. But remember, this is only work for diabetes type 1, not diabetes type 2. Okay, so let me come back to the question here. Alright. You need this stuff for bio? Yeah, you have to know it. It's part of genetic engineering. If you're exhausted, go and sleep, Nobody are keeping you here. Don't come in here and say you're tired. Go and sleep. Wait, your mother tell you to be here or what? You could go and sleep when you want. If all in all is catch hell waking up in the morning, go and sleep. Alright. So if all you look here, this question tends to come up a lot. The distribution of variant forms of the peppered mutt, Biston betillaria, change between pre-industrial and post-industrial Britain account for the increase in the numbers of dark form in post-industrial era. If all you actually look at these pictures here, I'm gonna do a little zoom up on it. It's very bad quality, but you can actually notice both mutts here. So here we have a dark mutt, and this is pre-industrial, where you had all of these like nah, I never eat it again after finding out. Right, so you had so this is pre-industrial, so this is pre. This is before it had all this soot and this smoke and all kind of thing blacking up the trunks. So here now we also have, yeah, I know it's zoomed. I'm showing you the picture. So here we have a white mutt and a black one. We look at it. Now remember what happening here is that it these ones here, these white ones was able to camouflage, and these black ones over here on the right was able to camouflage. So if you look here now, account for the increase in numbers in the dark form in post-industrial era. So this is what you'd see here. In the post-industrial era, tree trunks became darker due to the soot. This allowed dark variants of the mutt to camouflage and escape predators. As a result, they would have reproduced at faster rates than white mutts, which would have been predated, basically eaten by predators. Very simple. Now, this is how you have to think about CXC, right? When you answer any questions tomorrow. When you're walking into this exam, right? You need to think about CXC as a little annoying child. That little annoying child, whenever you say something, is going to say why, and you need to say why. If you know how to say why, say why. Because every little thing in biology is what's going to net your marks. If they say 3 marks, don't write 1 sentence. Think about every little thing to explain this. You're explaining it to an annoying little child so that they don't ask you, but why? You're trying to explain everything. You know it, write it. You know it, write it. You don't have another opportunity really, unless you want to repeat the whole of CXC. That is your fault. You should have deferred if you know you wasn't ready to write. So if your mommy or your daddy make you do it, you should have tell them, you should have stand up to them, because you're your own person and you're probably like 15, 16, 17 years old, and that's too old to be led on by all you're living under people's roof and thing. So if you're ready and you didn't defer, that's your fault. But write the thing, because when else are you going to do it? Next year? That happening. Write the thing. Don't just write 1 sentence, 2 sentences and thing. You need to explain the thing, because what you want to do is to prove to these people, to prove to these stupid, annoying children that you know. Why you have to do that? Why you have to prove to them? That's how society have it. That's how society is. Society is crap. Eventually, nobody will care about CXC. Eventually, nobody will care about CAPE. You really only need it to get into something. Or if you want to be a police, you need 5 CXC. If you want to go with KFC, you probably need like 3 or 4 CXC. But that's not what you want to do, right? Otherwise, CXC don't matter. You're just using it as a stepping stone. That's the thing there. You know it, write it. Don't hold back. You want to get them 1-word answers? Take your 4 and go down the road. That's how it is. Because the thing is, you did not defer. You're going in tomorrow and you're going in to do this thing. You have to put your all into it. It have no excuse really. Nobody really, a year from now, 2 years from now, nobody want to hear about COVID. COVID going to be scrubbed from the zeitgeist of Trinidad memory. In fact, everything that's scrubbed within 7 days. 7 days after the last person get vaccinated, everybody forget what is COVID. So don't blame COVID for the thing. Yes, it is to blame, but if you blame it, nobody care. The thing is, go in there, write the thing, right? Do the thing, write it. Okay. 2 is good. Now, I didn't take the vaccine because, well, I get I don't have any comorbidities and I was not 60 years old. I tried to take it, but the guard hustled me out. As I said, the most powerful person in the Caribbean right now is the guard outside the clinic. The vaccine, the more powerful than CXC, the more powerful than Keith Rowley, the more powerful than Kamala, who is mostly irrelevant right now, the more powerful than the Alsing. Anyway, let's go on to the next one. Okay. Use the theory of natural selection to explain antibiotic resistance. So this for 6 marks. So how are we going to come up with 6 marks here? Now for this, what you need to remember is this. Whatever don't kill you, let's make it stronger. right? Whatever do kill a bacteria does make it stronger. When you do kill a bacteria, it coming back for you, right? So you need to remember this basic thing here. And this is what is the basis for natural selection and antibiotic resistance. So, of course, that's not what we're going to write, right? So we're going to, the theory of natural. First, we're going to define what is natural selection. So natural selection is the adaptation of an organism to pressures in an environment. And its ability to survive despite those pressures, its ability to overcome those pressures to survive. So now you're going to go into it with bacteria. So imagine this. You have a yeast infection, right? Ladies, you have a yeast infection and the doctor give you an antifungal and tell you take this antifungal for 3 weeks. After you take it for 1 week, you find the yeast infection clear up and you say you're not gonna take the antifungal again. So what happens now? You have to remember now that when you take this antibiotic or this antifungal, it is not going to kill all, it's just going to kill most. It's just going to kill the ones that is the weakest, which is the most. Because what natural selection is about survival of the fittest. But think about the ones that are going to survive, the ones that if something say going to kill 99.9% of bacteria, well, we need to worry about that 0.1% because that 0.1% coming back to kill we. So this is what happens with antibiotic resistance, is that the stronger bacteria, right, are the ones that are going to survive. So bacteria that are exposed to antibiotics. Right, let me read this differently. When exposed to antibiotics, only the weak strains of bacteria will die. The ones that survive will eventually gain resistance. Yes, just like sanitizers. Sanitizers is not really a good thing if you will use it or not. So what they really they make we use the sanitizers all the time, and I mean you had to use it, but eventually you're gonna get a bunch of bacteria that's gonna be superpowered because of the overuse of hand sanitizers. Anyway, the ones that survive will eventually gain resistance to the antibiotic or may mutate into a stronger variant. As a result, a higher dosage of the antibiotic may be needed to kill the bacteria. So that's kind of like where you want to go with here. So what we have here is exposure to pressure. The pressure in this case is the antibiotic. Yeah, selective pressure. So a selective pressure could be anything. A selective pressure could be a change in climate. A selective pressure could be the introduction of a predator. A selective pressure could be the invasive species. It could be different things. In this case, the selective pressure is the antibiotic. So the antibiotic in this case is what you're doing is that it's wiping out the weak strains, the weaker strains. So the stronger ones will eventually survive, and those will eventually and the new population will comprise these stronger individuals, these stronger organisms. So what will happen is that eventually they could mutate, and because they asexually reproduce, right? Because they asexually reproduce, this could make mutations even they can make them even more dangerous because of how fast they reproduce. So all of that there you could talk about to get you 6 marks. So, right, so they could end up just being flat resistant to the antibiotic, and you have to get a different antibiotic. An example of this is a type of bacteria that's called MRSA. That stands for methicillin-resistant Staphylococcus aureus bacteria. And that's a type of bacteria that is found in hospitals, because hospitals they sanitize so often, and because they use antibiotics like methicillin and penicillin so often, that there's an entirely new strain of bacteria that's found in hospitals that what they're able to do is they're able to overcome a lot of selective pressures, and when doctors go home, nurses go home, and patients go home, they could spread that bacteria to other people. It's a kind of scary thing, but we found ways to deal with it once it was identified. So yeah, so that's natural selection to explain it. There's the last one. You just studied there's the last one now. Why you don't go and sleep? Just go and sleep if you're tired. You need to stay. Well, Zane, go and sleep. What happened? You look at the next one. Nobody is forcing who is forcing all your mommy forcing all you to stay while you just don't go and sleep. Don't bother me. Imagine you come on a man's stream and ask him when he go down. Alright. Let me find the next one. Me, you know? If it's the last one, I would say it's the last one. Alright, let me see if we could find some more genetics questions here. You do like one more and I feel that could be it. If that answer the question there. Well, I was saying that too. There's a freemium listener. There's the poor man bootleg review listener. No? Okay. One thing we are going through is menstruation. So let me do that one and then we're done. Yeah, well, I mean to be disrespectful to all you, but I'm not obligated to be respectful. Alright, so you look at this one. This is the last one. I have very little patience with children. I don't need to make money. Okay, so alright. Let me explain this menstrual cycle thing, right? So the whole point of the menstrual cycle is this. So the whole point of the menstrual cycle is to produce a follicle, and inside that follicle you're going to have an egg, and that egg is going to get released, and then if the egg doesn't get fertilized, the woman will shed her endometrium, basically a uterus wall, and that will be menstruation. If the woman does get pregnant, the egg will implant on uterus wall, and eventually it will form into an embryo and then a fetus, and eventually she will have a baby. Okay. So we have two hormones that are involved here, right? And there's actually four hormones that are involved in this. So I'm going to list the four hormones here for you. We have four hormones: FSH, LH, estrogen, and progesterone. So let me explain each one. FSH is also called follicle stimulating hormone. Right, so follicle stimulating hormone, and what this does is that it helps form the follicle, which is kind of like the casing for the egg. Now, eventually, the follicle ruptures and becomes a corpus luteum. The word corpus luteum means yellow body. LH stands for luteinizing hormone. LH is used to trigger ovulation into occurring. Basically triggers the release of the egg. I'll make this a little smaller. Estrogen allows the buildup of the uterus wall. It also stimulates the release of LH. Progesterone maintains the thickness of the uterus wall so that implantation may occur. So these are the four main hormones that are involved here. FSH and LH are examples of pituitary hormones. Estrogen and progesterone are examples of ovarian hormones. So these two hormones here are released by the brain, and estrogen and progesterone are released by the ovaries. Although progesterone is also released by the corpus luteum. I don't know what sort of shit the Tiwari doing in here. No, this man talking about gonadotropin releasing hormone and human chorionic gonadotropin and all kind of thing. Or they leave the children alone, man. You're talking about all them things the children on here to learn FSH, LH, estrogen, and progesterone. They need to know about no syncytial trophoblasts and chorionic villi and amniocentesis and yolk sac and alloantois and all kind of thing. They need to know them things. They need to know about the negative feedback mechanism of GnRH and how it is raised FSH and LH and stimulate, inhibit, and testosterone and estrogen and progesterone. And they need to know that when inhibin is released when FSH stimulates the Sertoli cells and LH stimulates the Leydig cells to secrete testosterone, and that testosterone would have a negative. They need to know them thing. Leave the children alone now, man. You're bringing in GnRH and HCG and all kind of thing in here. They just need to know these four things. So anyway, alright. So we have these four hormones here. And let me draw out what has happened here. You already trying to them kill themselves or what with them thing? Alright, so what exactly is hormone X and hormone Y, and what is happening at event Z? Well, this is easy. Hormone X is estrogen. Hormone Y is progesterone. And Z is ovulation, because you could see the egg is being released there. Hey. Well, they're all going through now. If only feel this hard, or they're all going through now. Hey, wait till I'm read about megagametogenesis degeneration and secondary oocytes and antrums and Graafian follicles and all kind of thing. Wait till I read all that. The frighten the children, man. Alright, what is the role of each hormone in the menstrual cycle? Alright, so I just put it there. Estrogen allows the uterus lining to thicken and stimulates LH production. The progesterone maintains the uterus lining for implantation. Alright, so what's happening here? Let me draw it out. Let me draw out the whole cycle for you. Let me draw the whole cycle for you here, right? Alright, that's now, I'm gonna get a picture there. Alright, so forget this graph. And let me get a picture of the female reproductive system. Alright, so show you something here, right? Alright. This is ovulation, right? There's a release of the egg from the ovary. They go into the fallopian tube. So what would stimulate that is going to be LH and estrogen. So LH is going to trigger ovulation. Now as that happens, now progesterone level is going to go up. Because what's happening there is that the follicle is left behind from the egg that is going to start to release progesterone. Now that's going to allow the uterus lining to thicken. So if the egg gets fertilized, it could end up implanting there, but unfertilized egg could end up implanting too. But if we say that the egg didn't get fertilized, basically the woman didn't get pregnant, right? This is what will happen. Progesterone levels is going to start to drop. So let me explain what's happening there. What progesterone does, progesterone allows the uterus lining or the endometrium to remain thick. If progesterone levels end up dropping, basically if the woman does not become pregnant, the uterus lining becomes devascularized, which means that it loses the blood vessels and it constricts. So what the uterus is actually doing, the uterus is actually strangling itself, it's choking itself and depriving itself of oxygen. So ladies, if all you have a period and you find it very painful, this is because the uterus is literally contracting and choking itself. And what it's doing is that it's suffocating itself and the cells are dying as a result. And when they die, they dislodge and they get a muscular layer called the myometrium that helps to contract and push out the dislodged, necrotized or dead tissue. So this happens throughout. However, if the woman was pregnant, what would happen now is that this fertilized egg will start to release progesterone, which will keep the uterus lining thick, because if the baby is forming, the last thing that you want is for a dead baby to fall out of here. So that the embryo, well, basically the placenta, which is what connects the embryo to the mother, is going to secrete progesterone as well. But if no implantation, basically if it never meets up with a sperm, Right, so if it never meet up with a sperm, I don't know why all you have to go through it so much, because I mean, how much times in your life you're going to be pregnant? I don't really know which big words I'm using, honestly. I mean, I understand before, but me I know which ones I'm using now. I'm using normal words now. Yeah, so this is what's happening here. Alright, let me make this bigger. Going by. Right, okay. Right. So where we're gonna have a follicle. So this is a follicle. So this follicle is like a casing for the egg. This thing inside here, that color in blue, this is the egg. There's the egg inside here. So this egg going to eventually get released and go into the fallopian tube, right? And they're gonna leave behind something called a corpus luteum. That's this thing here, corpus luteum. This corpus luteum, this thing is basically this thing allows secretion of progesterone right after ovulation occurs. So this helps to keep this lining here very thick. Now let me say that a woman does get pregnant, right? So she does. What will happen now is that this lining here will get very thick. Right? So we have a sperm cell. Right? So we have a sperm cell. There's the egg cell. And we have a sperm cell. Remember, this always happens in the oviduct, right? And a sperm cell fertilizes the egg cell. So you could have the implantation happening in the oviduct. That's not what you want, because that'll basically bust open your fallopian tubes and you'll die. That's called an ectopic pregnancy. But if it fertilizes the egg and forms a zygote, this will eventually implant here. And from there, it is able to grow structures that would allow it to connect to the mother. Why does it happen every month? Assuming is an evolutionary thing, because it allows the human population to be as big as it is, which is not a good thing, but maybe one day we will evolve to not have the period so much, you know what I mean? In other animals it doesn't really occur as often, but humans, me, you know. Anyway, so let me look at the rest of the question here. And after this, we're done. Okay. Name the organ that produces hormones X and Y. Okay, that's no. I feel I pasted in the wrong thing. Alright. Name the hormones that produce X and Y. Well, that'll be the ovaries, right? For estrogen and progesterone. So this would be. Wait, now let me take a look back at this question. And if X is LH, boy. Well, I feel X is LH. Yeah, X is LH. And Y is progesterone. X is not estrogen. It kind of weird though, like the diagram. Anyway, so let me look at this here. Discuss the fate of the following structures shown in figure 4 if fertilization of the ovum occurs. Alright, so the corpus luteum, or if fertilization occurs. It looked like LH, so I'm not sure. The diagram kind of poor. Anyhow, eventually disintegrates. Well, actually, produces progesterone after ovulation, but eventually disintegrates after fertilization and implantation. You don't have to learn how to draw the graph. We should be able to identify it from the graph. The uterus lining, thickness is maintained. As a result of to allow implantation of the fertilizer. Now the thing is it looked like I feel it's LH, but X and Y.
When day comes, it floats into space like a spirit amazed.
Forget it, yes?
Paradise.
Hopefully there I come. Me feeling to open back the diagram. Alright, so let me look at this last two. Right. Some contraceptive pills contain hormones. Suggest how this may prevent pregnancy. Well, there's just one mark. They suppress ovarian hormones such as estrogen and prevent the release of the egg. Oh, by the way, I noticed that in this exam, people they call anything ovum, or they in for a big surprise if all they do Form 6 biology. Because that thing, a ovum, is a completely different thing than an egg. We're going to get into it now. Alright, so you look at this last one. A drug company is developing a male contraceptive pill. Suggests 3 ways in which this could prevent pregnancy. Alright, so but the male contraceptive pill die out yet. So let me think about ways in which this could work. Well, what we're trying to do is prevent the formation of sperm or the release of sperm. So they've tried to do a contraceptive pill already by trying to reduce testosterone levels, but the problem was that nobody wanted to have sex. So it didn't really make sense. But what they could do is that they could produce immature sperm. So it could prevent the sperm from maturing, from reaching maturation. It can what it could do is that it could prevent the release of sperm through the vas deferens. And 3, it could lower, well, this is not the best one, but it could lower testosterone levels and sperm production. I guess our last one would be it can render the sperm immobile. You know, the sperm have a tail like a flagellum that is whip and carry the sperm forward. It could stop that from producing because that's the thing that is developed last. So it can render it immobile, so something like that. Yeah, so it could lower sperm production, lower sperm count. But you know, I mean, they never pump that much money into producing this thing. But I've read about it, and those are some of the things. They say like one of the best ways to do it is to actually produce a little mucus in the vas deferens. So what has happened is that the sperm does end up sticking up in the mucus when they're coming out. So it though is not like a hormonal thing, it's like a mechanical prevention mechanism now. So it just prevents it from exiting. Okay. Well, well, I can tell you, I feel that's a wrap. I feel like we do real plenty. But let me suggest to you one more time if you want to. I mean, I'm sorry for those who are Spanish, but let me tell you like what I feel might come, although what I say don't really mean anything. I have a feeling all they'll get enzyme graph, and it might have something about something linking enzymes and nutrition. They love to bring enzymes. Um, they will have an ecology question that I feel would link decomposers to carbon cycle. So study carbon cycle and thing, and decomposition linking to carbon cycle, and they will involve something with food webs and thing in there. Transport might be, I really can't say because transport could be plant or animal. But usually they just try to bring, out of 6 questions, they just try to bring 3 plant-oriented ones. So that nutrition one could also have a photosynthesis thing in it. The transport thing could have a transpiration in it. And the only other thing here that could involve plants is ecology. So if I look at it here, that's no, let me see. So remember, I could go through my notes. Again, the links in the description. So they're definitely having a question on ecology, which is biotic and abiotic factors, food chains, predator, prey, decomposers. So the one on disease, the thing that is usually bring most on disease is vaccines, well, immunization, and different types of diseases. So make sure I know the different types. I really can't tell all for reproduction, but since I say this usually try to bring 3 plant questions, I feel all the 3rd plant question going to come under reproduction, which is going to be cross-pollination, self-pollination, double fertilization kind of thing, or asexual reproduction or vegetative propagation of plants. So make sure and get all the rest for tomorrow. As I say, don't drink too much milk, don't eat no curry in the morning. Try to eat something dry so you don't have to go in the toilet. Once you go in the toilet doing an exam, you basically fail the exam because you run out of, well, you fail, you know what I mean? You run out of time. So study my notes, study the checklist that I give there. And yeah, if all you want to do physics is sometime next week, and we're also doing a bio paper one. So this is not really goodbye. Those of you who want to come back, or they could come back, tell all their friends. And as I say, I'm not doing this for non-profit, right? So there's non-profit. It's not really intended to give any lessons or start a channel or anything like that. This is just me saying the pandemic was hard and I'm trying to help all you out. All right, all you. So take care and good luck tomorrow in both bio and what's the other thing, and Spanish.